Okay, good morning and welcome to the last day of the conference. So let's start right away. So the session is Microbiology and Global Health. Let me introduce our moderators. Dr. Manuel Angelakis, who is a research director here at the Pasteur Institute, head of the Diagnostic Department and Public Health Laboratories. He is a clinical microbiologist. He has trained in Crete and also in Marseille in France. He has held positions at Latimon University Hospital. Also, he was an associate professor in clinical microbiology at Marseille University. And his work is about clinical microbiology, public health diagnostics, translational research. He focuses on infectious diseases, antimicrobial resistance, and population-level surveillance. And also we have Dr. Haralambia Boletti, also research director at the Pasteur Institute, in the Department of Microbiology and also the Bioimaging Light Microscopy facility. And she specializes in cellular microbiology, quantitative light microscopy. Also, her work spans intracellular parasitism and host pathogen interactions. Also, they have developed and teach advanced imaging approaches that support biomedical research and diagnostics across the institute here and also other institutes around the world. So thank you very much for moderating this session.
Okay. Good morning, everybody, from us as well. So welcome to the session of Microbiology and Global Health. In a world increasingly shaped by emerging and re-emerging pathogens, antimicrobial resistance and inequalities around the globe, the role of microbiology has never been more critical. And as we heard the first 2 days of this conference, in several talks, viruses and microbes have shaped life in our planet from the beginning. The best we understand them, the more prepared we will be to combat the diseases they caused. And with that said, I will call the first speaker to present her talk. She's Virginia Pichler, do I pronounce the name well?
Well done. Yeah.
From Institut Pasteur de Paris. Dr. Virginia has earned a PhD from the University of British Columbia in Canada, studying Mycobacterium abscessus. And she is postdoctoral fellow at the Institut Pasteur Paris. She's going to talk today. The title of her talk is Beneath the Surface, Unraveling the Morphological Spectrum, Glycopeptidolipids, and Infection Dynamics of Mycobacterium abscessus. Thank you, Virginia. Welcome.
Thank you. Okay, good. Good morning, everyone. Thank you for your determination and commitment to science and being here despite the strikes. And thank you for the invitation to talk today. Hopefully I can kick things off well. Okay, so I'll be speaking about Mycobacterium abscessus in a clinical context. So Mycobacterium abscessus is one of the many non-tuberculous species belonging to the Mycobacteria genus, and it's gained increasing attention over the last 20 years because there's been a rise about 20% in global cases. It's intrinsically resistant to many of the first-line tuberculosis drugs, and only about 50% of the cases are fully treated, so there are a lot of severe and persistent infections that are still poorly understood. So it's especially challenging to treat due to these intrinsic resistance mechanisms, some of which include its mycolic acid-rich protective waxy cell envelope. And with the use of antibiotic therapy, it has inactivating enzymes, expulsion of other antibiotics through efflux pumps, and it also has genome plasticity to onboard resistance genes and modulate environmental responses. So this host of resistance mechanisms paired with infection persistence has earned M. abscessus the moniker of the incurable nightmare. So to better understand how it takes a strong foothold, we can look at its pathogenesis. And we can see that it's actually ubiquitous in the environment, but many of the infections come from nosocomial sources, and immunocompromised individuals are the most affected, like those with cystic fibrosis. So upon infection, innate immune cells flood the area to contain the pathogen, and they form a granuloma, which is a hallmark of tuberculosis infections as well. And so even though it's initially contained by the granuloma, there's persistent infections will see the rupture of this structure, and even though we don't fully understand the mechanisms that drive this, we do see that there are certain phenotypes that have been linked to aggressive and persistent infections. So if we zoom back into the cell membrane and take a closer look, there are actually several molecules that decorate the surface, including immunostimulatory PIMs and lipoproteins. And unique to the non-tuberculous species are additionally these glycopeptidolipids that sit proud of the other molecules, and we refer to them often as GPLs. So expression of these GPLs are variable between species, but they've been observed within the same bacterial population, and this phenotypic variability has become recognized as a key characteristic of M. abscessus, with some bacteria showing a complete absence of GPLs. And this is even being observed within the same infection and impacts host immune response. And so that's because the presence of these GPLs creates this masking effect, thereby shielding the bacterium from detection. So when they're not expressed, phagocytosol-like receptors readily interact with the underlying immunostimulatory molecules, and this triggers an immune cascade to clear the pathogen. And this dynamism is actually really advantageous because it allows M. abscessus to take hold in an early infection by avoiding detection. So when we take a step back and we look at this macroscopically, we actually see this phenotypic difference in the colonies as well. And as it stands, the canonical framework classifies M. abscessus as either smooth, which is the browned, shiny, uniform version, or rough, which is the rough, textured, irregular version. And you can see that these captured images are a pretty classic representation of that. And the feature most closely linked with these differences is the presence or absence of GPLs. But the understanding of these features has been developed primarily from lab strains, and so there's a paucity of clinical infection data, and we really don't know how well these current models apply to a clinical manifestation. So that brings us to my work, my PhD work, and I had the invaluable opportunity to partner with St. Paul's Hospital in Vancouver, Canada, and the BC Centre for Disease Control to work with the Adult Cystic Fibrosis Clinic, where 19 patients provided 46 samples over the course of their infections, and we saw all of the subspecies of M. abscessus represented. So to understand the abundance of each morphotype in these infections, the first step was to subculture them to gain pure stable colonies. And colonies were scored based on those classic characteristics that I just described. But however, it soon became apparent that these elements failed to capture the full morphological spectrum of these clinical strains, like we can see in this progression. And these subtypes were observed across numerous strains and were often independent of transient variables like colony age. So what this really highlights is that to properly qualify morphology, an entire separate sub-study really needs to be done because we currently don't have a standardized protocol or rubric. So given the limitation during the course of my study here, colonies adopting any smooth-like traits were conservatively scored as smooth. The distribution across the 46 isolates showed that both morphotypes were represented about equally, and about half the isolates possessed both types. And just going forward, any smooth and rough strains that were separated from a mixed isolate are referred to as an isogenic pair, which really just means that they came from the same isolate and are genetically linked. So then when we look at the morphotype composition, the original sizable 46 has now increased to 75. So next, I looked if GPL expression matched the colony categorization. So to do this, I extracted lipids and separated them using thin layer chromatography. And as in this example, we can see that there's the distinct GPL banding at the top of the TLC. And this is what we see in the smooth morphotypes, whereas it's absent in the rough. And all GPL profiles matched the previously assigned categories, with the exception of a notable few. Which include number 70 and number 80. They were categorized as rough, however, you can see that they have that GPL banding. And this is how they looked, their colonies looked, so you can see they look pretty classically rough. But then when they were actually plated more densely in the image on the bottom right, they actually approximate more of the smooth morphotype. And then when we captured in color, they have that pretty classic brown hue, which is also reminiscent of the smooth morphotype. So these strains seem to adopt a mixture of both morphotypes, and so we consequently just labeled them as a putative submorphotype R-. Now, with the understanding that clinical strains deviate from this two-category system, we wanted to evaluate how each isolate behaved in an infection environment. So we did this with the THP1 macrophage model. So what's already been established in the literature is that a difference does exist between smooth and rough strains intracellularly, with the propensity of the rough strains to clump, and then they're brought in what is known as social phagosomes, whereas the smooth at the top are brought in loner phagosomes. Lysosomes attempt to fuse the loner phagosome, but the smooth actually alters the phagosomal membrane, thereby halting acidification, and this allows it to persist in the macrophage. Whereas with the rough, they actually are fully acidified, but they're resistant to degradation. The smooth also downregulates reactive oxygen species, thereby preventing apoptosis, whereas in the rough, we actually see that it enhances this, and we think this is so it can be re-released into the surrounding tissues and form these large extracellular clumps that are then difficult to clear by future macrophages. So I went ahead and I used a high throughput and sensitive platform to track intracellular infection over time, and I did this for 3 days, and what we can see here on the left is the smooth lab strain, and then on the right, oh, it's hard to do this, the rough lab strain. And we can see that the smooth actually grows much more rapidly with this dark red color at 72 hours post-infection. But then if we look at the clinical strains, this is actually reversed with the rough strains, clustering on the right, and they're actually more reminiscent of the smooth lab strain. Whereas the clinical smooth strains replicated much more slowly, which was more reminiscent of the rough lab strain. So this was an inverse relationship. But because one aim of this work was to understand the relationship between isogenic pairs, these data were also plotted to show their comparison. And while most rough replicated about 2 to 3 times higher than their smooth counterpart, we actually saw 4 isogenic pairs where this was reversed. And this is really reminiscent, again, of what we see in the lab strains. And then lastly, while extracellular bacteria was not explicitly quantified, we did observe these extracellular cords forming. And here what you can see is an isogenic pair with the smooth being internalized in individual bacilli in one macrophage. And here you actually see that serpentine cord structure spanning 2 macrophages, so it gets quite large. And then if we look at this longitudinally over time, because we had many isolates from the same patients, we do see a transition from smooth to rough, especially in patients 1 and 2, and this is often a feature of worsening infections. And given these are clinical data, we also had access to metadata to track lung function capacity of the patients over time. So we see this transition, and then if we look at this plot on the bottom left here, these 2 metrics, FEV1 and FVC, are quantification of lung capacity, and we see a tight inverse relationship between the decrease in lung function and the increase in bacterial replication rate. And this seems to be more a function of the changing rough morphotype over time. And so while the mechanisms of this shift in GPL expression haven't been fully elucidated, it's been suggested that permanent mutations to the GPL locus cause an interruption to synthesis, transport, or regulation. And we can see this reported in these studies here. And from a handful of other studies, we actually see that the transition to the rough variant is not exclusive to genomic changes in this GPL locus, but can actually also be a function of other environmental factors like sub-inhibitory antibiotic stress. So what we do know is that the canonical framework of smooth and rough, developed primarily from lab strains, is really insufficient to fully characterize clinical results, with the rough strains possessing the most variability within the clinical isolate population. So overall, we do see a relationship broadly exists between morphotypes, intracellular behavior, and clinical infection outcomes, but the current classification system is really insufficient to predict intracellular replication rates and more broadly behavior due to this diversity. And rarely do we see that clinical strains match their lab counterparts, and of course existing studies are mostly done on lab strains, which has limitations to applying this to clinical manifestation. And despite some morphotype association with infection stage, MSS's infections show higher heterogeneity, and other factors like environmental, genetic, or immunological response really do need to be explored to better understand infection pathogenesis and progression. And I will just briefly touch on this. So moving from global to granular, I'm now building upon this work in my current postdoc position in the microbial individually and infection unit, looking at host pathogen interaction at the single cell level. And we heard a little bit about organs on a chip before, but with the alveolus on a chip platform developed in our lab, we can recapitulate the lung environment with complex cell organization, modulate environmental conditions like stress and nutrient availability, and even recreate respiration mechanics. And this will help unpack the specific drivers of this diversity between smooth and rough and better understand the dynamics of subpopulations in infection progression. And with that, I want to thank the many, many contributors to this work and the funders, and thanks for your attention.
Any questions from the audience?
So one slide you had the images of, and then there was this long red. Yes, what was it again? I didn't really understand what it was. The long serpentine structure. I didn't catch that.
Maybe I rushed through it too quickly. This one?
Yeah, exactly.
Yes. So what we have here, so this is one isogenic pair, so it's been separated into smooth and rough. So this central image here, this is just a more a broader view, and then this is actually a confocal image. So we have the smooth bacilli. They're a little overlapped here, but they actually are in individual loner phagosomes, so they're internalized individually, and you can kind of see them as individuals here. Whereas in this case, this is actually a little later in time, and these rough bacilli have escaped macrophages and have formed these large clumps, and they're sometimes called serpentine cords because they look like a snake. And these are actually 2 macrophages here, so you can just see how large they get, and this prevents clearance by future macrophages just because they become so big. So that's one of the strategies for why we believe that these infections are so persistent and challenging to treat.
But those bacteria then continue to infect other cells, or they just stay like?
So they stay like this, but then what you can do is you can have individual bacilli break off and then infect future ones, yeah.
Thank you.
And may I ask here in this image, what are the white dots in the macrophage?
Oh, these are stress macrophages because they're stressed, okay. And so they're nearing the end of their life. I actually didn't take this image. A colleague of mine did, but it was also just to try to get a little bit of better resolution. But I think they're sorry guys, I'm not an immunologist. They're what are they called? Something bodies. I can't remember, but I know they're a feature of stress.
Okay.
That's that. Thank you very much. Yes, that's what it is.
What happens to the macrophages on the right or your left?
Sorry, this one?
Yes. What happens to the macrophages?
So we do see some cell death, but the experiments we usually run for I only did up until 72 hours. I think the longest I've seen anyone do an experiment is up to 5 days. And then at that point, we're not replacing the medium or anything, so the macrophages are just stressed from that anyway. So, but we do see, there are some in vivo studies, and we do see cell death with really aggressive persistent infections.
So it might be an interest, questions because we're in the same lab, but did you ever try to compare RNA-seq, the gene level expressions in genes responsible for glycolipids formation and everything, just between the smooth and the rough, and maybe seeing their changing levels over time?
So I have not. There was a study a while ago done by Alexandre, actually, in Roland's lab. And they compared the lab strains, the smooth and the rough, and what they did see was a huge downregulation in certain genes in the GPL locus. So that has been well characterized. I don't think anyone has done it over the course of an infection where they've tracked it over multiple days. But if I go back to, and I kind of touched on it quickly, but this slide here, this is just a handful of the studies that I found that show that there are sometimes smooth and rough have been observed in clinical context, and they have nothing to do with the GPL locus whatsoever. They find expression level changes in other parts of the genome. Sometimes they don't find mutations at all. because it's suggested that it's the most responsible is this huge deletion in the GPL locus, but that's not true for all cases. And so right now we have this overly simplistic view of what drives this shift from smooth to rough, and it's really just not comprehensive.
It would be interesting to have some more molecular markers, like some protein markers. You showed that it's not clear
Yes.
which protein upregulation leads to the rough morphology, but that's a very interesting aspect to investigate.
Absolutely, and I think I'm going to have that opportunity to do that in my postdoc a little bit more, because we're looking at things at the single cell level, and because of the microscopy alveolus on a chip platform, we can have a more complex infection environment, and I'm going to be using a series of reporters and dyes to try to distinguish those aspects.
Maybe we don't have time. We are a little bit late, so...
Can you keep your question for the coffee break? So...
Thank you so much.
Okay, so our next speaker, thank you very much, Virginia. Our next speaker is Justine Ostermeyer from the Institut Pasteur Paris. Yes, no. Justine.
We changed it.
Yeah. Jasmine is a molecular biologist and geneticist, and he's conducting a PhD at the Institut Pasteur, where she is investigating the coordination of replication in pseudo-alteromonas. So the title of the talk is Two-stir determines replication direction of chromosome 2 in pseudo-alteromonas haloplanctis. Oh, what an exotic name, eh?
Thank you. Does it work? Yes? Okay, perfect. Sorry for the font. I didn't choose that. That just changed now. But, I mean, you can read it. That's the important part. I would like to start with a question. And I was wondering how many of you have heard of bacteria with a multipartite genome before? Yay. Few people. That's good. So for the others, a quick introduction. So bacteria with multipartite genomes are bacteria that have, in addition to the main chromosome, they have one or more large extrachromosomal replicants. This could be either megaplasmids or chromids or anything that is very big and domesticated. And you can see that this is found in about 10% of bacteria. So everything that is not red on the slide are bacteria that have a multipartite genome. And this includes pathogens as well as non-pathogenic bacteria. And over the years, these large ERs, they have been domesticated by the bacteria, meaning that they originated from plasmids that over time became bigger through the acquisition of more DNA. They acquired some regulatory sequences that helped them to be integrated and to coordinate the replication of them. In addition, the copy number for small plasmids is often quite big, and for them it's often reduced to the same number as the main chromosome. And in addition, the replication is synchronized with the bacteria, and with the synchronized replication, I mean it's synchronized to finish at the same time. And here you can see different bacteria, and you can see they all synchronize. So this is a plot where you sequence the bacteria during exponential growth, and then you plate the reads, and because replication is a duplication, what is replicated first is duplicated first, so it's more abundant, and that way you get sort of this gradient where, like, usually what's at the origin, so where the replication starts, is at the tip, so it's most abundant, and then towards the termination side, it becomes less. And you can really see, like, everything sort of finishes at the same time by, yeah. So this synchrony seems to be a hallmark of this extrachromosome replicants. And why it's very well studied in Vibrio, so the causing agent of cholera, Vibrio cholera. We were wondering, how is it in pseudo-Alteromonas? And the reason we are interested in this bacteria is because in the 4 clades there are, in most of them, the chromate, so the large ER, is replicated unidirectionally, and only in one very small clade it's replicated bidirectional. And the bidirectional ones, they tend to have a bigger size. This comes from the integration of DNA between the ori on the diff site, which made the bidirectional replication not only possible but necessary, because in the end the replication is always synchronized to finish at the same time. And today I would like to talk about the mechanism of the unidirectional replication and how this works in pseudo-Altromonas. And essentially there are sort of 2 ways this could be. Either it's a completely different mechanism that initiates replication, or it could be a mechanism like a secondary mechanism, like the blockage of one of the replication forks that causes this unidirectional replication. And in E. coli, there is this TUS-TER mechanism, and there it's very well studied. And the way it works, you have this TUS protein that binds to a TER sequence, and this TER sequence is found multiple times over the chromosome. It's repeated, so it's the same, and it has sort of a polarity. So here indicated by light blue and dark blue, meaning that when the replication fork approaches from the permissive side, which would be light blue to dark blue, the replication fork displaces this TUS protein and the replication just continues. However, if it approaches from the non-permissive side, which would be dark blue to light blue, the unwinding of the DNA from the helicase causes cytosine to flip into a pocket of the TUS protein, and that way it locks it into place and the replication is blocked and cannot continue. TUS is also found in pseudo-Altromonas on the chromate. So you're wondering, maybe if we delete this protein, there would be no protein to block, and if there's no blockage, the replication would continue, and that way maybe we see a change in the direction. So that's exactly what we saw. So this is the wild type. In blue, you have the chromosome, so the main chromosome, and you can also see it's bidirectional, like you have the point and then you have 2 slopes. And then in green, you have the chromate, which you can see it's unidirectional, so you have like 1 point and then only 1 slope. And now in the mutant where we deleted TUS, you can now see it's became bidirectional. So what we actually think is happening, that replication starts bidirectional, and then one replication fork is immediately blocked by TUS, and therefore the other replication fork has to sort of finish the replication, and that's why it looks unidirectional. And then again, like in the deletion, because there's no blockage, now it's bidirectional, because the replication fork is not blocked. So it seems like the unidirectionality comes not from a change in the ori itself, but more from an accessory mechanism, this TUS-TER mechanism that blocks one of the replication forks. So next we wondered, what does it bind to? And again, like I said, in E. coli it's very well studied. There is this sequence that is known to be the target for TUS. And like I said before, like there are multiple sequences. So we started to look for repetitive sequences in pseudo-Altromonas. And we found the sequence, which at first doesn't look very similar, but if you take the reverse complement, actually it's almost the same. So this is very promising. So we took this a step further. We took this sequence and we blasted it against all the complete genomes that were published on NCBI. And as you can see, again, like for the unidirectional strains, you can see them mostly near the origin, where you expect them to be if it's unidirectional. And then in the few strains where it's bidirectional, you can see now actually it moved to the opposite side of the origin. So again, this is very promising, and it looks like this might be the actual sequence that is bound by TUS. Yes. Okay, so almost at the end. Yes, so just to summarize, it looks like these TER sites are controlled by this TUS. The unidirectionality is controlled by this TAS-TER mechanism, and we also found a putative TER site that might actually be the target for this sequence. Yes, and so why we wondered why is this mechanism, why does it have to be unidirectional? And we think, again, this is because the replication has to stay synchronized for the maintenance of the genome in these bacteria. And then with this, I'm already at the end of my talk. I would like to thank my lab and the funders and so on, and you for listening to me. Thank you.
Thank you, Jasmine. Open for questions.
Hi, thank you for a very interesting talk. I had a question about the ERs, the exosomal replicons. I was wondering if you are aware of any biotechnological application of them. Is there any yet, maybe similar to how plasmids are used, for example?
So in the case of pseudoatramonas, I know that some people are interested in using it for technological applications. On the one hand, because you can actually express quite big and quite complex proteins with it. And on the other hand, also because the growth temperature is a bit lower compared to E. coli, so you sort of can save some energy by not having to set your incubator at 37, but only at 25 degrees.
Thank you.
Is there any way of targeting this, maybe? Because you mentioned there are also some...
Okay, thank you very much, Jasmine. And now we move to our third speaker, who is Stefania Kapsetaki. Stefania is a biologist with master's and PhD degrees in zoology from the University of Oxford. And then she proceeded with postdoctoral research at the American Universities of Arizona and Tufts. And now she is working as a postdoctoral fellow at the Institute of Molecular Biology and Biotechnology in Crete. Stefania is going to speak about the bioelectrics of immortality and mortality in cold-sensitive Hydra oligactis. Another very interesting microbe. No, this is not a microbe, it's a multicellular organism. Yeah.
Thank you for the very kind introduction. It's great to be here. Thank you to the organizers for organizing such a great event. Today I'll talk to you about one of my projects at Tufts University on the bioelectrics of immortality and mortality in Hydra. So first of all, why should we care about this project? We all age, and there are lots of diseases that are related with aging. There's a high prevalence of arthritis, heart disease, cancer, diabetes, and chronic obstructive pulmonary disease as we age. And recently in a review, some letters are missing, Recently in a review by Lopez and Levin, they described aging in a different way. They describe aging as a progressive failure of biological information processing and a decline in the ability to maintain anatomical homeostasis. So what does that mean? They drew a picture to try and help us to understand what that means. And essentially, they suggest that young individuals would have a sharper bioelectric pattern, whereas older individuals would have a blurrier bioelectric pattern. So what does that mean? How do we measure the sharpness or the blurriness of an image? Imagine you have like a black pixel and the surrounding pixels are all white. Well, this picture is sharper than another image where we have a black pixel and the surrounding pixels are all dark gray. So this is how the sharpness or blurriness of an image is measured, and I'll tell you more about this later. So what are we going to do to solve the problem of aging? So instead of genetic approaches, our lab back in Tufts uses bioelectric approaches. And specifically in this project, we want to find the bioelectric pattern of aging and immortality. How are we going to do this? What are our methods? Well, we used a membrane voltage sensitive dye, which shows us the relative membrane potential of every cell. And this dye is called Fluorovolt. It's available commercially if you want to buy it. And, wow, lots of things missing. And we use a specific software here called FLIM, Fluorescence Lifetime Imaging, and it's associated with confocal microscopy. So essentially, when a laser beam is emitted from the microscope, it hits the Fluorovolt dye, and then photons are emitted. And the longer it takes for the photon to be emitted, the more depolarized those cells are. If it takes a shorter time for the photon to be emitted, then those cells are relatively more hyperpolarized. And so essentially, when we look at the literature, different cell types, like cancer cells, different kinds of stem cells, or quiescent cells, in the literature they're reported to have certain membrane potentials. But now with this dye, we can actually see this. We can see that the depolarized cells are relatively more like red, and the hyperpolarized ones are more the green-blue region. So we have our dye, and now what kind of animal are we going to use? We decided to use the cold-sensitive strain of Hydra oligactis, and that's because it's transparent, so that's great for microscopy, and it ages at 10 degrees Celsius, whereas it's immortal at 22 degrees. And for many of you, this might sound strange, because usually things age or get stressed out at higher temperatures. So I think this is quite strange model organism. But there are studies showing that actually this animal ages like this. It has mechanisms and phenotypes, hallmarks of aging at 10 degrees Celsius. And so what do we expect? We expect that the old Hydra would have a blurrier and more depolarized bioelectric pattern than the younger and immortal Hydra. I told you what sharpness and blurriness is. Now, why would we expect older Hydra to be more depolarized? Well, if you remember in one of the previous slides where we had the vertical, like an image of which cells are relatively more depolarized and hyperpolarized, you notice that cancer cells were at the top. So in the literature, lots of the cancer cells are mostly depolarized. So, and we know that Hydra also have tumors, some of them, when they age. So therefore, we thought that old Hydra would have a relatively more depolarized bioelectric pattern. What did we find? We found that Fluorovolt can be used in this system to see the relative membrane potential of cells. So here in green, we have lots of hyperpolarized cells. In red here, lots of red cells here. Those are relatively more depolarized. And here I wanted to show you this video because it's important to emphasize that Fluorovolt can only be used in live animals. You can't fix these. You can fix them, but for Fluorovolt to work, you need the animal to be live under the confocal microscope. Another thing we found is that not every Hydra has the exact same bioelectric pattern. So all of these Hydras have been grown at 22 degrees Celsius, so they're relatively immortal, but we do see quite a lot of variation in the bioelectric pattern. Another thing we found that they seem to have a consistently depolarized foot. So let me explain these figures. So on the x-axis, I've aligned all the Hydra. So from the left, we have the tentacles, and the right is their foot. And with image J, I can simply split the image into percentiles. So we have 0 to 25%, 25 to 50, 50, 75, 75 to 100%. And in each of those percentiles, I can take, from image J, I can take like the vertical line. And in each vertical line, I can take the average membrane potential of all those cells. So that's what each dot is here. The colors are how many months after the experiment these images are from. So this is 2 months, 3 months, and after 4 months. And in parentheses, I have how many Hydra I imaged per time point. Next, we found that old Hydra are relatively more hyperpolarized and bioelectrically sharper than immortal Hydra. And we can see this in these results here. I'll explain a bit more. So each dot is a different biological replica, a different Hydra. And the y-axis shows us the average membrane potential, the average voltage of all the Hydra. So that's why it's just one dot. So on average, we see that across all these hydra, which are old mortals, they seem to be more hyperpolarized than the immortal hydra. And here in this figure, we've measured the bielectric sharpness. So it seems that old mortals have a sharper bielectric pattern than immortals. And I have a question mark here because this is opposite to what we expected. And this is quite strange and possibly open for discussion. But then when we looked at the foot, we found that this did follow our expectations. The older hydra have a blurrier foot than immortal hydra. And there's lots of future directions here. For example, we don't know yet what kind of molecular mechanisms are regulating all this bielectric pattern. There's lots of studies to be done regarding, like, how do we switch the old bielectric pattern to a young one? What kind of bielectric drugs can we use that target different kinds of ion channels? And this system can be used in many different model organisms that have to be transparent and they have to be kept alive under the microscope. So with that, I'd like to thank all these people, co-authors in our paper. Bridget, sorry, the surname, Galliot. She's based in Geneva, and she sent us the cold-sensitive strain of Hydra legactis. And thank you for your attention.
Thank you, Stefania, for this very interesting talk. And questions? Yes, Jasmine.
I have actually 2 questions. The first one, do you have an idea why the foot is depolarized?
Why the foot is depolarized?
Yes.
We think that bielectricity is related to function, and for sure, hydra use their foot to stick to different surfaces and use the tentacles to grab prey. So maybe this depolarization is related to stickiness, to releasing some sticky substances, and maybe that changes with aging. Maybe they lose some of their functionality and they like float. So I think it's related to functionalities, but I don't know exactly.
Thank you. And can I ask a second one? So I don't know anything about hydra, but are they symmetric? Like, so or do they have like a dorsal and a ventral side? So yeah, you have the foot and the tentacles.
If you put the foot down and the tentacles up, if you split them vertically, yes, they are symmetrical. But if you split them this way, they're not. They have
okay. Yeah. So there's no, they don't have a dorsal and the ventral side, something that could explain why the pattern is different.
No.
Okay.
And it would also be difficult in that, anyway, the way we've put them flat under the microscope, so we wouldn't be able to, anyway, yeah.
Okay, thank you.
This is a little bit not firmly related, but I came across a study that is actually investigating bioelectric patterns in cancer cells, and you mentioned them in an earlier slide. It was suggested that there's almost a loss of bioelectric memory, which causes cancerous cells to form, and you can actually, if you can restore that bioelectric memory or patterns, it can be used as a therapeutic option. And I don't know if there's any application here. You mentioned, there are a lot of potential follow-up studies here. So have you thought, or is there a potential to try to modulate the bioelectric or memory potentials to see if these features can be restored or changed?
Yeah, there are I shouldn't say too much because it hasn't been published, but there are collaborators in Tufts University who are starting to use a similar approach in flies, dissecting the gut of flies and playing around with them. There's certain ion channels that you can change and possibly switch the old bioelectric pattern to a young one. So they're trying to use those channels and see whether, if they overexpress them at different time points during lifespan, they don't know exactly when would be the good time point to kind of upregulate those genes. But yes, they're trying to make the bioelectric pattern sharper. And it's a good point about cancer cells because here, as I said, hydra sometimes have tumors, and nobody yet has looked at, they could use this system to look at possibly tumor cells in these animals and see whether, they can use a bioelectric drug that reverses aging, but also reverses those kinds of cancer phenotypes to healthy cells. So there's lots of potential there, yeah.
May I ask a question following up that? Do you know of any apparatus that could modulate the bioelectric membrane potential of cells?
I know something simple. So the longer you leave these hydra under the confocal microscope, because of the laser beam, they get heated up and they get stressed. So I know that if I take these images for like over 30 minutes, I start to see all the cells being red, so depolarized. So with simple, by stress, they do become more depolarized. But there's lots more there. Like you could change their diet, you could change, I don't know, the chemistry of the liquid and see lots of things. But I know for sure that like under stress, they tend to like become depolarized overall, the whole body. So I have to be quick with imaging, like within 30 minutes, and that's it. Bye-bye, Hydra. Yeah.
There's another question over there.
We are a bit late.
Just one question.
Thank you. So this might be like less of a scientific question, but I'd like to know your insight on that. Especially in the past decade, we've seen like this huge focus, this huge movement in anti-aging in the pharmaceuticals. I'd like to know what you think about that and how much science is actually backing that up, and if it's diffusing into basic science as well or no.
Okay, I would say I don't know the whole field of aging, but there's like, there's always in science, there's some studies supporting some drug that helps reduce aging or increase health span, but then in different studies you find the opposite. So I think we always have to be careful, maybe listen to many doctors. I know that I've been doing mostly non-human stuff, so I've looked at, for example, lifespan across many different species, and it doesn't always correlate like with diseases. I've been looking at how does cancer correlate with lifespan. It doesn't always correlate. So there's lots of pathways and diet. It doesn't correlate with like longer or shorter, like eating healthier. We saw it like yesterday. So we have lots of room to go forward. Yeah.
Thank you.
Okay, thank you very much, Stefania.
Okay, we're going to move on with Anastasia Lambropoulou from the bacteriological laboratory of the Hellenic Paris Pasteur Institute. Anastasia is going to give us a talk for the central law of predatory myxobacterial evolution and history of metallo-beta-lactamases. Anastasia Lambropoulou is a biologist of the Public Health Laboratory of Hellenic Paris Pasteur Institute from the Department of Medicine at the University of Patra. He is currently a PhD candidate in the microbiology laboratory of the Hellenic Pasteur Institute under the supervision of Dr. Kotsakis. Anastasia, thanks a lot.
Good morning. The subject of my presentation is the central role of predatory myxobacteria in the evolutionary history of metallo-beta-lactamases. The multi-resistant problem is mainly due to beta-lactamases, which deactivate even the large resort of beta-lactams, such as neurocephalosporins and carbapenems, which are drugs of choice for this infection. Metallo-beta-lactamases are of great concern with the respective enzymes NDM, VIM, and IMP, hydrolyzing almost all the drugs, and there are no clinically available inhibitors. Identifying the organisms where MBLs evolved may provide novel therapeutic breakthroughs, as would enable the identification of adaptive mechanisms leading to efficient expression and the high catalytic efficiency of MBLs in gram-negative. Moreover, we could narrow down the organisms where natural inhibitor molecules can be found. In order to identify the native hosts of MBLs, we screened bacteria genomes using specific hidden Markov models. We observed that certain bacterial groups hosted the identified enzyme families, with MBLs being part of the core genome. As we can see, Bacteridota have the highest carriage rates, shaded in blue color. MBLs are part of the core genome from several families from Chytophagia. The gene was also part of the core genome from Bixobacteria, with the enzymes of the latter forming monophyletic lineage sharing a common ancestry with clinical NDM and VIM, as shown in the subtree. Based on the above, it's possible that MBLs were originated in Bacteroidota, and they did not jump immediately to Proteobacteria, as these two groups are phylogenetic distant, and they will face expression problems. It's possible that Myxobacteria was the intermediate host, and especially the predatory members of it. Myxobacteria are abutant soil organisms. They have complex life cycles. They prey on other bacteria, forming fruiting bodies containing myxospores. They are also potent producers of secondary metabolites, some of them having antibacterial activity. Studies in the model organism of Myxococcus indicated the production of beta-lactamase in certain stages of their life cycles, and our data indicate that this beta-lactamase is an MBL. These findings are in line with their role in the evolution of MBLs, and hence we studied the hypothetical gene transfer from Myxobacteria to Proteobacteria by studying their expression in E. coli. Initially, we isolated 4 strains from Greek soils, representing 3 genera, and they were sequenced with ion taurine whole genome sequencing. They were all producing MBLs, which was confirmed with hydrolysis assays of imipenem and inhibition assays with VTA in cell-free extracts. The open reading frames of these genes were cloned in expression vectors, along with the gene of the model organism Myxococcus xanthus and the clinical NDM, and they were expressed isogenically. The beta-lactam resistance phenotypes were determined with minimum inhibitory concentrations. In the graph, we can see the difference between the environmental enzymes and the clinical one, as expressed in serial dilution differences. As we can see, the environmental MBLs have a weaker phenotype compared to the NDM. Deviations in the amino terminus containing signal peptide may have a tremendous effect on expression of difference. Beta-lactamases should be secreted in the periplasm in order to provide resistance in gram-negative, as the targets of beta-lactams are located there. Most of them are secreted via sectorase lock-on using type 1 signal peptide for soluble expression. The NDM is a rare case where it has type 2 signal peptide, and it's located bounded to the inner leaflet of the outer membrane. Myxobacteria have both type 1 and type 2 signal peptide, with the ribobox differing from that of the NDM. In order to compensate the signal peptide differences, we expressed both mix and NDM under a common type 1 signal peptide. The replacement of the signal peptide reduced the resistance levels for NDM, while it had no effect on mix. And this indicates that Myxobacteria MBLs are possible that are secreted as soluble proteins in E. coli, despite the fact of having a lipobox. Studies have shown that the membrane localization of NDM protects in proteolysis, and this is why we see the weaker phenotype. Assuming they have the same sensitivity to proteases, the weaker phenotype for environmental MBLs may be due to failure of attachment to the membrane. And in order to answer this, we perform localization of the MBLs in comparison with type 1 and type 2 signal peptide. We conduct fractionation of E. coli cells, and at each step we measured where the majority of beta-lactamase is located. Our data indicate that Myxobacteria MBLs have the same properties with enzymes secreted via type 1 signal peptide and not as NDM, which the sole enzyme is located in the outer membrane. So it's possible that only type 1 signal peptide is functional in E. coli for these MBLs, which is why we could purify high amounts of them in their native form. Even under a common signal peptide, the phenotype of the environmental enzymes was weaker compared to the clinical. In order to determine if this is due to catalytic properties, we measured the kinetic constants of different beta-lactams during hydrolysis. As we can see from the K to Km ratios, the environmental MBLs are potent beta-lactam hydrolysis with catalytic properties similar to those of the NDM. The differences were observed in oxymenocephalosporins. While they could hydrolyze efficiently cefotaxime, they turn over poorly ceftazidime and cefepime, with NDM being above 10 times more efficient. Ceftazidime and cefepime have a positively charged R2 side chain, which may impair the binding with the catalytic center of the environmental enzymes. In order to map the enzyme moieties where adaptive evolution took place, we tried to regenerate NTM mix properties, NTM properties to mix. For this reason, we conducted direct evolution to native mix using aeroprobe PCR, and we observed that the mutations increasing the resistant levels to all beta-lactams are located in the amino terminus of the protein. The mutations in the mature enzyme were rare, and the majority of them were increasing the resistance to all the beta-lactams, except of some that increased only the levels to ceftazidime. Among these, the glutamate to lysine located before the catalytic 120 aspartate, which decreased significantly the resistant levels to ceftazidime. We then crossed over the recombinant forms of mix and NDM with using non-homologous recombination, utilizing in the H technique. We obtained two libraries. The first had the mix first and then the NDM, and the other with the opposite arrangement. We used these libraries in order to select the evolved hybrid enzymes and in order to select the enzymes yielding the beta-lactam resistance. We used ampicillin in concentration higher than the two enzymes, and this corresponding to our naive library, and newer beta-lactams to concentrations higher than the environmental enzyme, corresponding to our evolved libraries. Selected clones were analyzed phenotypically and Sanger with Sanger sequencing, and the whole the libraries were used for were sequenced with nanopore sequencing. Analyzing the contrast of the hybrid enzymes by determining the coverage against the two genes, we observed that in order to provide resistance, the N-terminus should not contain more than 30% of the environmental enzyme. But when NDM was in the N-terminus, high percentages of the environmental enzyme in the domain 2 could be contained. Phenotyping the isolated enzymes from our selected clones verified our findings, indicating that when the amino terminus domain containing the HHF motif belongs to NDM, then we can say the hybrid enzymes exhibit high resistant levels to cephalosporins. And finally, we assessed if bacteria could produce molecules against MBLs using crude extracts from our xocoxazolate. We observed a synergy with cephalothene against Nicolai clones producing beta-lactamases from different classes. the synergy was not specific for MBLs, as the inhibition was also detecting the E. coli clones producing serine beta-lactamase. But these preliminary data indicate that Myxobacterium may produce bioactive molecules that act against MBLs in Enterobacteria. Finally, our data indicate that adaptations leading to the emergence of MBLs mainly concern efficient expression in the new hosts rather than functional differentiations. Myxobacteria may integrate MBLs in their physiology, with their dual peptide being the main facilitator for their expression during the cycle. Secondary metabolites may control this phenomenon, and our data indicated the pivotal role of in terms of MBL from their functional properties, but this work is ongoing, haven't finished yet. Thank you.
Thanks a lot, Anastasia. Do we have any question? Okay, thanks a lot. We're gonna. We will move to the next presentation. It's Olga, ποιος σας έχω. Δεν ξέρω. Okay, we'll continue with Mrs. Olga Papageli from the Hellenic Paris Institute. She's going to talk us about the assessment of viral activity of photocatalytic surfaces for a clean and safe environment, Apogeon. Olga is currently pursuing a master in infectious diseases and international medicine. She conducts her master at the Hellenic Pasteur Institute, focusing on the antiviral activity of photocatalytic nanomaterials against HSV1.
Thank you for your introduction. Good morning, everyone. I would like to thank the organizing committee for giving me the opportunity to present this work here today. Our work is a result of a multi-institutional collaboration between Democritos Institute, Hellenic Pasteur Institute, and also a Nanophos company. To give you some introductory information, photocatalysis is the procedure where a semiconductor is activated through photon-induced excitation, leading to the creation of strong redox reactions that have the ability to oxidize air pollutants, to purify water from organic ones, and also to exhibit strong antibacterial and antiviral activities. Titanium dioxide is a well-established and highly active photocatalyst, but has the limitation that it's only active under UV light exposure. However, there are other photocatalysts like graphite carbonitride and silver phosphate that are visibly light active, and here in our work we assess the antiviral activity of their combination. A few words about the workflow of our project. Our colleagues in Democritos Institute were the ones that synthesized the photocatalytic materials to be tested. Our colleagues in Nanophos scaled them up into ready-to-use specimens that we received here in Hellenic Pasteur Institute and assessed their antiviral activity against herpes simplex virus type 1 at the Laboratory of Molecular Virology and against Bacteriophage Q-beta at the Laboratory of Medical Microbiology. Our specimens are divided into two groups, and only one of them contains titanium dioxide. Both of the groups contain a specimen that is modified only with graphite carbonitride, and also another one that is modified with the composite of graphite carbonitride and silver phosphate. Now about our biological models. We have two very different viruses tested. Bacteriophage Q-beta is a small, non-enveloped RNA virus with single-stranded RNA genome that exclusively infects bacteria, while HSV1 is a large enveloped DNA virus with double-stranded DNA genome and constitutes an important human pathogen. Both of them are well-established laboratory models, and Q-beta is a safe surrogate for studying single-stranded RNA human viruses. Also, both of them are lytic viruses that gives us the opportunity to measure their infectivity easily by plaque assay. The cells we used in our experiments include Escherichia coli for the Q-beta viral system and Viro ATCC for HSV1 viral system. Moving on to our methodology, we first wanted to exclude any possible cytotoxicity of our specimens. So in order to do it, we incubated Viro cells or E. coli cells with neutralizer that had previously came in contact with each one of our specimens. And after a specific period of time, we tested their viability. The antiviral activity evaluation protocol was based into 2 ISO standards. Briefly, our virus came into contact with each of our specimens under 2 different conditions, either kept in dark or illuminated by under visible or blue light. And then the viral mixture was retrieved, and its titer was calculated either by double-layer agar method for Q-beta or by plaque assay for HSV1. Finally, for the expression of our results, we used two mathematical formulas given by the ISO, with delta V being the antiviral activity and has a minimum significant value of 0.3 that corresponds to 0.3 log reduction of our virus and subsequently to a 50% decrease in viral infectivity. On the molecular detection methods, we evaluated 3 qPCR systems as for their accuracy of detecting Q-beta genome, and we also tested 2 alternative primer pairs during these protocols. Moreover, we investigated the effect of blue light activated graphite carbon nitride to HSV1 life cycle. To do so, we detected the late glycoprotein C gene in the supernatants of infected Viro cells, and we also studied the expression of GC gene and the early thymidine kinase gene intracellularly during the infection. Moving on to our results, no adverse effects of any specimens was observed either on E. coli cells or Viro cells, since their viability remained intact. Regarding the antiviral activity against Q-beta, we observed that there are multiple delta V values. Titanium dioxide had the strongest antiviral activity against Q-beta, followed by the newly synthesized graphite carbon nitride and silver phosphate, whose antiviral activity enhanced by the presence of titanium dioxide. In contrast now to the Q-beta viral system, for HSV1 we have a viral reduction approaching 50% only for the specimen containing graphite carbon nitride. Given these results, we decided to also examine the effect of blue light, which has higher energy inside the visible light spectrum and probably stimulates our photocatalysts more effectively. The results showed a tremendous increase of the viral reduction, reaching 90% on both specimens containing graphite carbon nitride, which was unrelated to the existence of titanium dioxide. Moving on to the molecular detection of Q-beta, 3 different qPCR systems were efficiently standardized. 2 primer pairs were used, but only one of them, primer pair 1, was able to successfully detect the Q-beta viral genome. A representative results from probe-based qPCR shows that for both 2 steps or 1-step probe-based qPCR, we have the same efficiency either on diluted or undiluted samples. Finally, we examined the effect of graphite carbon nitride on HSV1 life cycle. The results showed a significant reduction in extracellular viral load 8 hours post-infection between the blue light condition and the controlled dark condition. These results may indicate possible changes in the expression of HSV1 genes. So we decided to study also the expression of the late GC gene as well as the TK gene during different time points of infection under both blue light and dark control conditions. Both viral genes seem to be downregulated at 6 and 8 hour post-infection under the blue light condition compared to the dark. In conclusion, the newly synthesized complex of graphite carbonitride and silver phosphate has adequate antiviral activity only against bacteriophage Q beta when activated under visible light. Moreover, graphite carbonitride itself exhibits mild antiviral activity when activated under visible light against HSV1, but a strong antiviral efficacy when activated under blue light. Three different qPCR systems for the detection of Q beta viral genome were established, making it easier to detect viral genome changes in future experiments. While the specimen containing graphite carbonitride appears to cause reduced viral particle egress and also affect the expression of early as well as late HSV1 genes. Overall, antiviral activity of each specimen highly depends on the type of viral system being tested. I want to thank all the members of my lab and also all of you for your attention.
Thanks a lot, Olga, for your interesting presentation. Do we have any question?
May I ask a question? What are the prospects for using these materials? Actually, is this certainly there are applications?
Yes, these materials can be, these photocatalysts can be coated on surfaces that, on public places or in hospitals in order to purify air, to purify the air and also to prevent of
spreading.
Of bacteria or viruses to be spreaded.
So we saw that for different viruses, different materials are active.
Yes.
Are more effective. So then there should be a combination at the end.
Yes. There is ongoing research for this subject, and also we think that the size of each virus plays an important role on why it is activated, why it is.
One material is more effective in rather than another. Yeah. Thank you.
You're welcome.
Okay, we will finish the session with Dimitra Yalama from Alexander Fleming Institute. She will present us the discovery of novel antimicrobials, inhibitors of microbial functional amyloid aggregation linked to biofilm formation. Dr. Dimitra Yalama is a chemical engineer with over 10 years of international research and experience in biochemistry. Her work focuses on protein aggregation inhibitors, synthetic biology, and translational applications in neurodegeneration and antimicrobial, leading to two patent applications and several peer-reviewed publications. He is a postdoctoral research associate at Euroscreta laboratory.
Thank you. So today I will talk to you about the discovery of novel antimicrobials and the discovery of inhibitors of functional amyloid aggregation linked to biofilm formation. So as you all know, AMR is a growing threat worldwide, and the next generation antimicrobials are virulence compounds that reduce selective pressure, promising to minimize the chances of resistance evolution. Thank you. And E. coli is a bacterium that is mostly responsible for the most deaths attributable to AMR. For this reason, and also because we can use easily E. coli as a model system for our drug discovery platform, we choose to target its E. coli, its biofilm matrix, and to target the functional amyloid CSGA. So as you know, depending on the environmental conditions, bacteria constantly alternate their lifestyles. One of their survival strategies is to form biofilms. On surface, they often organize themselves in multicellular communities, and they're enclosed in a matrix. Usually, one main matrix component is Curly. So Curly is composed of functional amyloids, and one major Curly subunit is CSGA. So the role of CSGA is very important because it acts as a shield for the bacterium. As you can see here, this is the bacterium, and this is CSGA. This is a cryo-EM image. So it offers protection against antimicrobial agents and the immune system. So it's much harder to kill bacteria when they are in a biofilm than when they are not forming this enclosed matrix. Also, they're responsible for recurrent infections. So to use our drug discovery system, we want to produce CSGA in the cytosol of E. coli in order to monitor its aggregation. For this, we delete the signal sequence of CSGA and we produce it from a plasmid. The next thing we had to do was to find a reporter for CSGA aggregation, and we tried to use GFP, which has been successfully used by our lab previously for pathological amyloids. So once you form a fusion of an amyloid to GFP, then GFP changes its conformation and it does not fluoresce. And the question is, does this work for functional amyloids as well? And the answer is yes. So for slowly aggregating CSGA variants, this one and this one, we have higher fluorescence, and for more aggregation-prone CSGA variant, we have less fluorescence. So now that we have our reporter system ready, we can target CSGA using cyclic peptide candidates. We have these libraries in our lab. We use the Cyclops technology, which is based on inteins. And so far I have tested random tetrapenta and hexapeptides. And they are composed of 10 to the 7th library members. So this is the screening cycle that I used. So initially, you transform E. coli with a reporter plasmid, and then with the plasmid library, you can use huge libraries, as the ones I just mentioned at once. And the bacterial library is then plated on an agar plate, and in each colony, you have two plasmids, the reporter plasmid and the plasmid of the library. Each colony has a different member of the library. Then after culturing and induction, using FACS, you isolate the most fluorescent bacteria, you isolate their plasmid, and then you do the cycle again and again until the fluorescence is saturated, and then in collaboration with the lab of Dr. Hadjis at Fleming, we can do NGS and identify the cyclic peptides that are promising. So these are the actual data. So as you can see, after rounds 3 and 5, we had enrichment of twofold enrichment of GFP fluorescence. And after NGS, we focused on the most frequent cyclic peptides. So let's move on to heat validation. I co-expressed the plasmids encoding these peptides along with CSGA in the E. coli cytosol, and I could see that, as you see here, for CH1, 2, and 3, we have higher ratio of soluble to total CSGA compared to the random plasmids, the random peptides, I'm sorry. This gave us some hope. So the next step was to do the in vitro assay. So I purified and refolded CSGA, and using the well-known THT assays, so thioflavin T is a dye that actually fluoresces only once bound to amyloid fibrils. So you can see that for the wild type we have the signal we expect, and for CH1, 2, and 2, we have less fluorescence, less THT fluorescence, meaning we have less amount of amyloids. Yeah, and for peptide, cyclic peptide in 7, we have the same result, and cyclic peptide in 7 was isolated from a modified screen. The next step was to inhibit, to check if we inhibit endogenous CSGA amyloid formation, so CSGA that's actually found on the surface of E. coli. So I used the Congo red staining, and as you can see in the wild type sample, we had this red color indicating that we have amyloids on the surface of E. coli as we expect, whereas for delta CSGA we don't have this color. I hope you can also see that for CH1 and 2 we have decoloration as we expect, showing that we have inhibition of endogenous CSGA amyloid formation. Finally, for cyclic peptides were tested for their ability to inhibit early biofilm formation on the surface of a 96-well plate. So I did crystal violet staining, which only stains the sample when you have bacteria attached on the surface of a 96-well plate. So for peptides CH1 and 2, and in 7, we see that we have reduced signal, showing that we have reduced biofilm formation. However, when I couple this with microscopy, we can see that the wild type is, these are color bright field images. This is quite characteristic of what I see in the microscope. And for CH1 and 2, we see that there are some cell aggregates that are large, as you can see, and they are not as well stained as the wild type. We don't know why. So this means we don't have this inhibition that we were hoping for. However, for IN7, we have the phenotype, exactly the same phenotype as delta CSGA, so we have successfully inhibited the early biofilm formation, at least on the plastic plate, the polystyrene plate. So to conclude, novel cyclic peptides that inhibit CSG aggregation and biofilm formation of E. coli have been discovered. These findings highlight the potential of the discovery platform for other functional amyloids that perhaps also form biofilm. So I would like to thank my supervisor, Georgos Kretas, and the rest of our lab, our collaborators, Padilis Hadis for NGS, and we have a new collaboration with Ioannis Rabias here at Pasteur Institute to test pathogenic strains. And I would like to thank the organizing committee and you for listening to me.
Thank you very much, Dimitra. Any questions? Yes?
I'm wondering if you've had the opportunity or if there's a plan to test this in other biofilm-forming species or bacterial strains.
Yes, so for now we are checking, we are aligning other CSGA genes from other organisms to check if there's any similarity and if we can use what we have with other bacteria as well. So, for example, Salmonella CSGA has a high similarity, and we are searching for more, and then we plan to test these bacteria as well.
If I can just ask one more question. Sorry, maybe you said it, I missed it. Where did the library come from?
So we have it in our lab. We have constructed it in the past. So it's random cyclic peptides. So the first amino acid has to be a specific one. It can either be cysteine, serine, or threonine, and the rest are random amino acids. So the theoretical diversity is 10 to the 7th.
Thank you.
Thank you for your great presentation. My question is partially covered by the previous one. If I understood right, you were talking about some cyclic oligopeptides.
Yeah, yes.
So their function in interrupting the biofilm formation, it's not a secondary structure feature, it's basically a sequence feature. Is the amino acidic sequence feature, or is it there some secondary structure elements that are?
We don't know why they bind. So we have this random library, and we do the screening to find some of the members of the library that actually do have the desired properties. And we don't know why they bind, but we see the effect. So in the future, hopefully we can determine why they bind, if it's just the sequence or a structural thing that makes them bind. Do I answer your question?
Yes, thank you.
Another question? Yeah, Dr. Zur, here, yeah.
Thank you for the presentation. I will follow up that discussion for a minute. So these cyclic peptides usually adopt a particular conformation. Are they structured? Are they unstructured in solution? Do you know?
So I don't know. So we haven't done any experiments to check yet, but hopefully in the future.
And the other question is, do you know, are there other molecules that do have similar activity, prevent biofilm formation, and do you know how yours compares to that, if you're taking it forward?
Yes, there are other chelicide, as they are called, and we don't know yet how they are compared to ours, because we cannot find them commercially available. So they're not commercially available. So we can only make some guesses from the amount of the peptide that is needed to have an effect, maybe we can understand if they are more potent.
Okay, thank you.
And the last question by Dr. Soures.
Very quick question. I don't know if there is a connection. Do you see any other effects on quorum sensing activity of this bacteria from the
Yes,
peptide?
we don't know yet. We haven't tested this. We see these huge aggregates. We don't know where they come from and what this means. Yeah, it's possible that there's some communication between the cells.
Okay. If there is no other question, we would like to call this session to an end. And we thank all the presenters and the participants and invite you to continue conversations in the breaks. There is the next session now.
Okay, so we now have small talks by our biotechnological companies, so please stay. They are very interesting. Also, please visit their stand outside. They have small gifts for you, and also they're friendly people. Don't be shy. And we will start with Vespina Tuda from Bioanalytica.
So hello everyone. First of all, I would like to thank the organizing committee for giving us the opportunity to present our solutions, Bioanalytica solutions, in this congress and sharing the stage with such accomplished scientists. So my name is Despina Tuda and I'm working in the MDX and Life Science division of the Greek company Bioanalytica SA. Just a few words about our company. Bioanalytica SA was founded back in 1989 and was the first company that introduced the Greek market molecular biology technologies. And since many, many years of experience in different applications like extraction, PCR, real-time PCR, digital PCR, and next-generation sequencing, now we are leading the way to the multi-omics era.
So our company is working for more than 35 years in the life science part, more than 30 years molecular diagnostics, already 15 years in next generation sequencing, and 13 years in flow cytometry. And our company is working as a distributor of globally known brands like QIAGEN, Illumina, Mission Bio, NanoString, Seer, among many, many others. And our vision is, of course, to contribute to a scientific community of our country with the most innovative technologies, and, of course, to contribute to the improvement in human health and the quality of life. So today's talk has to do with the multi-omics approaches, and actually I would like just to remind you what is multi-omics. Of course, you already know. When we do research to a single layer of these four, genomics, epigenetics, transcriptomics, and proteomics, this cannot tell you the whole story. So multi-omics is actually the approach where you do research to more than one of these molecular layers. And the aim actually is to do these multi-omics in the same sample at the same experience. And that way you can avoid misinformation, measure directly the causes and the consequences of everything, and maximize discoveries with data-rich and unbiased approach. So I would like to start by Illumina. Illumina, here in this slide, you can see the core instruments of Illumina, the sequencing instruments. And no matter what your application is, if it is whole genome sequencing, whole exome, transcriptomics, methylome, you can use this sequencer that they say are the same, XLEAP, SBS chemistry, and give high quality result. And you can choose through different high throughput system. So Illumina goes beyond genomics and has already integrated multi-omics, accelerating its multi-omics strategy. So it has new library prep kits with bulk, single cell, spatial, metagenomics, and more, connecting them with powerful sequencers and with state-of-the-art bioinformatics solution. So you have end-to-end solution with Illumina. And actually, it has recently integrated the connected multi-omics software, and this platform has the ability to connect proteomics, single-cell, spatial transcriptomics, methylation in one platform, and actually bringing all the data to one common framework. So at this point, I would like to share with you, and I'm really excited about it, the new update from Illumina, the 5-base sequencing. So now Illumina, apart from the 4 bases, can also read the methylated cytosine in a single assay with a single library preparation. So it has the ability to combine genomics and epigenomics with a single assay. Illumina has already acquired another company, the Fluent Biosciences, and has integrated the PIPSEQ workflow in its portfolio. Actually, this is Illumina's single-cell 3' RNA technology that uses the PIPSEQ, its barcoded particles, that actually partitions the samples, a million of cells, in working with them and give ability to do single-cell RNA sequencing. And this is done with a simple vortexing. These PIPSEQs, there are in different kit sizes that have different capability of capturing different number of cells, and they can go from hundreds to millions of cells without using microfluidics or any instrumentation. Another update from Illumina is that Illumina has acquired SomaLogic, so now has the ability to do proteomics. This way, Illumina accelerates its proteomic approach and builds its multi-omic strategy. So SomaLogic and Illumina have created a new library preparation capturing thousands of proteins. So we said that Illumina now has single-cell transcriptomics and also single-cell proteomics. So single-cell multiomics, because we are doing research in two different molecular layers. But why single-cell is important? Single-cell is important, of course, because you have the ability to have a better understanding on cellular heterogeneity and uncover population, rare cells population. So it gives you deeper insights from hundreds of thousands of cells and single-cell resolution. This approach has also been adopted by BD Biosciences, another company that we also distribute for many, many years. So BD Biosciences now has the BD Rhapsody system. The BD Rhapsody system actually works with transcriptomics and proteomics in a single cell level and has the ability to capture both surface proteins, mRNA, and also receptors from T cells and B cells, giving you insights in the immunology, oncology, and beyond. So by this system, which is a microwell-based single-cell capture, you are able to capture from 100 to 800,000 cells and then prepare them and actually to do sequencing from this system for RNA and proteins. And actually, the BD Rhapsody system includes also the BD Rhapsody scanner, which is actually a quality control in different stages of the workflow. You can check how many cells are viable, and then during the process, you can check how many cells you have in every step. And also, BD Rhapsody adopts the CytSec technology. And CytSec technology is a single-cell assay capturing both RNA and proteins using antibody oligonucleotides. So this is also incorporated in the BD Rhapsody system, and it's really important for a single-cell transcriptomics and proteomics. The readout of BD Rhapsody is NGS sequencing, and the workflow is fully integrated with NGS sequencers from Illumina. So, BD Rhapsody has the ability to give you and bring you all the applications available, from targeted RNA-seq, whole transcriptome, CRISPR screen, protein profiling, BCR and TCR, clonotyping, antigen specificity, and ATAC-seq. Similar story is coming from the company Mission Bio, which we also distribute. Mission Bio has a system, the Tapestry platform, that does at the same time genomics and proteomics in a single cell level. So BD Rhapsody was suitable for transcriptomics and proteomics, and this system is suitable for genomics and proteomics. And it uncovers a genotypic and immunophenotypic insights simultaneously from single cell, and you can detect single nucleotide variants, deletions, translocations, loss of heterozygosity, and surface protein expression simultaneously at the same cells. So this is this make it a powerful tool for applications like hematologic malignancies, solid tumors, genome editing, biomarker discovery, and cell and gene therapy. So Mission Bio and Tapestry offers an end-to-end solution with different targeted panels, and the readout again is sequencing. So this is also integrated with NGS sequencing platforms. And actually with Tapestry system, what we do is not only to detect mutation, but also link them to protein expression at the same cell. Then we have a collaboration for many years now with NanoString, which now it's a broker company. NanoString integrates a full, complete portfolio for spatial biology research. And has the GeoMX DSP system. It's a digital spatial profiler which actually captures proteins and RNAs from a tissue slide and has a region of interest selection that is really precise. So after that, the molecules are captured, are prepared, and they're going for sequencing, and has many different panels available in RNA and protein level. So if you don't want to do NGS and you have a more targeted panel, you can use the encounter analysis system, which counts up to 800 molecules in a very easy and short setup, and can give you powerful results and very reliable. So either after the GeoMX system, you can either choose NGS or the encounter to count specific molecules. Sorry. And then NanoString created the Cosmic Spatial Molecular Imager. This system takes the spatial biology in the next level because it does high resolution imaging, reaching single cell or subcellular level. So with this system, we can capture the whole transcriptome in a single cell and in subcellular level, as mentioned. This has also many panels, RNA and protein. And now also NanoString has added to its portfolio the cell space precise spatial proteomics system, which does in a single level again proteomics analysis after imaging. So the Cosmic system is single cell spatial transcriptomics, while the cell space is single cell spatial proteomics. So, and since we discussed about proteomics, I just want to show you this slide to just highlight the complexity of human protein. And I'm sure that you already know it, but it's really impressive that from 20,000 genes, we have over 1 million of proteins. And this is a result of alternative splicing or post-translational modification and dynamic expression. But here we see the enormous complexity of the proteome. The proteome is really rich, it is really complex, and is always changing. So, Bioanalytica has already collaboration with companies with purely proteomic solution, and this is one of our updates. So at this point, we are collaborating with SEER. SEER has a way to do deep, unbiased, and high throughput protein detection at scale. So actually, it has a very growing catalog of that reaching more than 10,000 proteins across 1,900 pathways to discover proteins associated with more than 100,000 possible biomarkers. So SEER has the Protograph XTSA kit with full automation. This is a preparation of different types of samples for many proteins with many different panels. And actually, all these preparation results to molecules that can be used by LCMS, the existing LCMS systems, and then SEER give us the Protograph analysis suite that brings all these data into sense and can give you insightful results. So last but not least, this last for this presentation because I said too much, we have a collaboration with a company, Quantum-Si. Quantum-Si has the Platinum system, which is next generation protein sequencer, and offers a simple workflow in a space-saving benchtop instrument with the precision to accurately detect protein variants and modification. It works into single molecule amino acid resolution and gives direct protein, it's a direct protein detection method and can interrogate proteoforms and modifications. So you can do protein barcoding, protein variant detection, antibody sequencing characterization, protein identification, post-translational identification analysis, and biomarker identification. And this is a system designed for real-world applications. So at this point, I would like to thank you very much for your attention. I gave too much information for this short time. But of course, feel free to visit our booth and learn more if something is really interesting to you. And of course, our expert can guide you to the best solution for your own research. Thank you so much.
Thank you very much to speak. And now we welcome to the stage from the Zafiropoulos company, Androniki Tasiopoulou and Marios Atanosopoulos. Sorry. Thank you. Oh, and they will talk about uncover the mysteries of personalized medicine, from deep immune profiling to the dynamic landscape of gene therapy. Thank you very much.
Good morning. Thank you very much for the invitation. My name is Androniki Tasiopoulou, and here is my colleague Marios Athanasopoulos. We're both sales and application specialists at Zafiropoulos SA, and today we're going to guide you through the fascinating world of personalized medicine, from deep immune profiling to the dynamic landscape of from deep immune profiling to the dynamic landscape of gene therapy. I'm going to present you BioLegend's TotalSeq platform, which is a solution that combines protein profiling with single-cell sequencing, DNA and RNA sequencing. At first, TotalSeq antibodies, since 2018, they have been the gold standard antibodies used for the detection of proteins by single-cell sequencing. Unlike classical flow cytometry antibodies, they are not bound with a fluorochrom. Instead, they are bound with an oligonucleotide, which consists of a PCR handle, an antibody barcode, and a capture sequence. This capture sequence is what makes them compatible with sequencing applications. One of their main benefits of using TotalSeq antibodies is that they allow scientists to discover and characterize cell states by immunophenotype, using also cell surface markers and also intracellular markers. Additionally, their usage is very important because they enhance the resolution of cells that have similar transcriptome, and also enhance the identification of cells with low or poor RNA quality. Finally, their staining procedure is very simple, and they very easily and seamlessly be integrated into new and already existing protocols and 10x genomics single-cell assays. Regarding their formats, there are 4 formats, TotalSeq A, B, C, and D. The first 3 of them, TotalSeq A, B, and C, are used for the combination of proteomics and single-cell transcriptomic analysis. And finally, TotalSeq D is used for single-cell genomic analysis. Regarding the first 3 of them, TotalSeq A, B, and C, the 3 of them are compatible with 10x genomic platform, and their main difference is their capture sequence, which makes them compatible with different assays. TotalSeq A has a poly-A tail and is compatible for 10x genomic 3' universal gene expression, and TotalSeq B and C are generated in collaboration with 10x Genomics and are compatible with 3' universal and flex gene expression assays and 5' universal gene and flex gene expression assays, respectively. Regarding TotalSeq products, products can be broken down into 4 different categories. We have single antibodies, we have cocktail, hashtag antibodies, and secondary reagents. regarding single-cell antibodies, BioLegend offers the largest selection on the market of different antibodies, which can be used to build your own antibody pool or as spike-ins to our already existing cocktails. Regarding cocktails, I'm going to talk more at the next few slides. So we're going to hashtag antibodies. Hashtag antibodies are very important because they can be used for sample multiplexing, or they can be used in order to increase the output, or to reduce the overall cost of a single cell experiment. And then we have secondary reagents that include products such as streptavidin conjugates or anti-isotype specific conjugates. As for total-seq cocktails, there are three basic categories of total-seq cocktails regarding the level of their complexity and the level of antibody multiplexing. The first category is the least complex cocktails for basic immunophenotyping with 9 antibodies. Then we have more targeted cocktails, more complex, such as essential cocktails with up to 100 antibodies, and then we have our most complex cocktails with more than 150 antibodies for deep immunophenotyping. One of these cocktails is Human Universal Cocktail version 2. It's the newest product at the total-seq portfolio. It has 175 antibodies and 9 isotype controls. What makes this product very important is that it can be used for deeper characterization of cells. It can be used at more tissue types, and also it has many antibodies that are highly relevant with translational research. Here you can see the results using the previous cocktail at the multi-omic analysis software provided by BioLegend. At the figure on the left, you can see how different cell populations are separated regarding their surface and intracellular markers. And on the right, you can see how RNA expression is differentiated regarding different cell types. And then we have a next generation tool for single-cell proteomics and transcriptomics, which is Phenosite. What makes Phenosite so unique is that it gives us the opportunity for instrument-free cell partitioning using the Scale Biosciences quantum barcode technology. It's also high throughput and scalable, and we can process over than 1 million cells at each experiment. Here you can also see the results using Phenosite with multi-omic analysis software. On the left, you can see data filtering and flow-like gating features using the mass software to define different cell populations. And on the right, the difference in RNA expression regarding cell types. To sum up, total-seq reagents make advanced protein characterization via single-cell sequencing very easier than ever, and also total-seq portfolio is expanding with new cocktails, new antibodies, upcoming tools for nuclear and phosphoproteins to help you with your multi-omic research. Thank you.
I will continue. My name is Marios, and I'll continue talking about the AAV gene therapy solutions. In the dynamic landscape of gene therapy, understanding potency and immunogenicity is paramount, and we know that developing AAV capsids that go unnoticed by the immune system is challenging, and getting the right tools. Sorry. Now can you hear me better? I apologize. So getting the right tools to support your design is very important. At PISA Firopoulos SA, through our long-lasting partnership with SVA Life Science, we know that no AAV vector is the same, and neither are your development needs. That's why our platforms for immunogenicity and potency offer flexibility and full vector-specific customized solutions to support the safety of the therapy and the efficacy. SVA presents two main types of assays: the custom development of potency assays, as well as assays for immunogenicity. With potency assays, you need to make sure that your AAV vector is working properly. And you do this by detecting three things: the transduction, whether the AAV enters the cell, the translation from DNA to protein, and the functionality of the protein. This is an example of a potency cell line. The iLite cGMP GC has been developed to facilitate the therapies for genetic diseases, and we use human retinal cells because they express firefly luciferase under a mechanism that is similar to the AAV vector. With this technology, you can have valuable data regarding potency, regarding vector similarity, and vector optimization. And especially, the thaw-in-use format enables you to have results faster, that are more robust, and more reliable. Using this technology, you can compare the different AAV lots, you can compare the different manufacturing methods for AAV capsids, but also optimize your capsid. As indicated previously, further than the potency, we have the immunogenicity assays. So let's start about the assays for the antibodies. This is a two-cell setup system, the iLite platform. The cell on the left are the packaging cells, the ones that produce the AAV vector serotypes shown in blue. And the cell on the right is the responsive reporter cells that detect whether your AAV vector can be transduced in the cell. If this happens in the reporter cell, you have activation of firefly luciferase. But if in the serum of the individual you have antibodies against the AAV capsids, then in this system the transduction is blocked or reduced, and thus the luciferase signal is also reduced. You can see that in the neutralizing graphs, where if in the patient serum you have high concentration of neutralizing antibodies, you completely block the transduction of the vector. This is also true for lower concentrations, but in very low antibody concentrations, you don't have any inhibition. And this assay takes overnight. SVA Life Sciences also has a simple ELISA to detect the antibodies, the total antibodies against the AAV vectors, independent of their functionality. Because previously I said about the neutralizing antibodies. This is total antibodies. And we currently offer solutions for IgG and IgM. Finally, let's talk about the complement. In clinical trials and therapy solutions with AAV vectors, high doses of AAV are systemically administered, and this may lead to the activation of the complement, which is critical for the safety and for the efficacy of this therapy. SVA Life Science, with over than 30 years working with the complement, has developed an assay that can detect the risk of a given individual, a given serum, or in other words, to see whether an AAV construct can activate the complement. To briefly explain the method, so AAV capsids of your interest, tailored to your needs, are coated, and then, when treated with a serum of an individual, if antibodies against the AAV capsids or other complement activators are present in the serum, then if they're incubated with the same serum that is treated with a complement activation buffer, finally you get a signal that is proportional to the level of complement activation. At PISA Firopoulos SA, we offer innovative solutions to support your laboratory needs. Make a step forward to advancing life sciences. We would like to thank our team at PISA Firopoulos SA. We would like to thank the organizing committee and welcome you to our booth so we can discuss about all your questions, and thank you for your attention.
Thank you very much, both of you. Now we welcome to the stage Theocharis Eftimiropoulos from the Anticell company, and he will talk about seeing biology in 5D multiomics, rich data, true dynamics, full context with AVD24.
Say my name.
Okay, thank you.
Hello everybody. So my name is Haris from Anticell, and I'm very happy to be here again to share with you the latest updates from Element Biosciences, technologies that they grow rapidly. Actually, I will talk about AVD24. It's a platform that can allow you to see biology in 5D multiomics to uncover mechanistic insights into your cells that are well hidden. It is exciting, and you will see how rich data this system can generate in a very simple way. That makes me want to go back to the lab and use it. And it's not only myself that found the system very innovative, but also from the Scientist magazine, AVD has been awarded as the breakthrough platform in biotechnology for this year. And there is two reasons for this award. AVD24 provides the best sequencing in the market, but at the same time provides in situ multiomics capabilities, both in one platform. So talking the first part about the sequencing characteristics, and then we will move to the cool stuff, to the in situ multiomics. So AVD sequencing offers four main reasons. The best accuracy. Generate Q50 data, it means that makes only one mistake in 100,000 base pairs that incorporates. It's cheap, significantly lower cost than any other mid-throughput platform in the market. Flexibility, it has two flow cells with two lanes inside the flow cell, so you can run 4 experiments with different parameters in the same time. And last but not least, compatible. You pick up your favorite sample preparation kit, and you load it to the flow cell. This will be circularized, simplified by rolling sang amplification. This is not prone to errors like PCR-based amplifications, and sequenced by avidites. This is the star molecule who won the award. The avidite-based detection offers high specificity because by stronger binding affinity with the nucleotides to the sequence that is newly synthesized. Due to this technology, avidite-based chemistry, we see Q45s and Q50s with our Cloudbreak and Ultra-Q kits, and this comparable to the other systems. AVD can offer clear improvements in read quality, optical duplicates, index hopping, and perform better in homopolymer-rich regions, difficult regions in the genome. This quality affects nearly any NGS application. That's why we see a tremendous increase in publications referring AVITI, not in peer-reviewed, in preprints. And it's not only the number of publications, but it's the quality. As you see, these studies were published in high-impact journals, and reflecting the relevance that on the data that are working, the data that have produced from the AVITI sequencer. So now, I switch gears, and we expect even more publications to accelerate since AVITI 24 is outside. This is a platform evolution from AVITI, so it has all the strengths of the sequencing that I mentioned before, but it's also, it introduces also this Teton cytoprofiling kit, which is actually the tool to make the in situ multiomics. This functionality offers the high resolution of measurement of RNA, proteins, morphology, in a subcellular spatial resolution, and can do this only with one assay, only with one run. So this multi-features that Teton provides opens the way for new experiments that you can answer complex and multi-layered questions with one experiment. You can answer which genes are overexpressed and which proteins are abundant. What is the cell morphology looks like? Where the gene of interest it's located inside your cell during your functionality, and how molecules and cell morphology changing during time upon stress. If you are able to answer all these questions, you will find the function of your gene that you are looking for. The coolest thing, this is not that you can answer all these questions, but you can do it very easy. AVITI 24 achieves this from sample to biological insight with library prep-free multi-omic approach. You need only 45 minutes hands-on time. You take your cells, you put it on this plate, Teton plate, you permeabilize, fix, and you assemble it with a flow cell, and you load it to the system. After 24 hours, you co-detect 6 morphological markers: the classic cell brain, nucleus, actin, ER, mitochondria, Golgi. You detect 350 RNA targets and up to 138 proteins and phosphorylated ones. So all these only with one assay. But you can also have a dynamic response, and this is a preprint where this is how the data look like. So in here we treated the cell line with an inhibitor, and with the same time we measured morphology changes, we measured RNA expression changes and protein abundances changes. As you see, the cells during the time morphology is changing, the convoluted cells are dying, and also the count per cell are decreasing for these 2 specific targets. So where are we now? What are the capabilities now in terms of sample types? We can load adherent cells, cell suspensions, and PBMCs. In terms of content, we have 4 fixed panels: MAPK, cell cycle, MAPK, apoptosis, immuno-oncology, and neuroscience. Fixed panels of 250 RNAs, 50 proteins, and 6 homology markers. If the gene of your interest is not belonging here, we can provide customization. You give us the sequence of your gene or the antibody that works well for your protein, and we give you back the barcodes. In terms of the configuration, we have 1 well plate where you can analyze 1 million cells, and then 12 well and 48 well plates are good for the dynamic responses, for time point experiments, for different perturbations and different concentration studies. And now we'll talk only now for this for direct in-sample sequencing. This allows you to sequence up to 100 base pairs direct in your sample without library prep. So it means that opens the way for applications like cell typing and CRISPR screens. So the great part of this, first of all, called Teton Atlas, it's compatible with many samples and different species, not only human cells. So this is a fully automated library prep-filled multi-omic approach. You just have to load your cell, and what you get out, it's sequencings up to 100 bases that they come out either from a targeted RNA sequencing or from the whole transcriptome, from a single cell. And on top of that, this is compatible with the previous chemistry that you can do also cell profiling and protein and phosphoprotein detection. The technology behind that, it's simple. I will talk about the transcriptome. We have a Proly-T probe that binds to the poly-AM RNA. So you perform cDNA extension, which encodes the actual target of the gene. This is the emphasis. We actually target the gene of interest. This could be circularized, amplified, and read by the sequencer, by AVITI. So actually, we have a very good specificity by this way. So with this, you can resolve SNPs, gene splicing, and strand orientation. Apart from the specificity, the problem with these technologies, for example, for whole transcriptome analysis inside the one cell, is very difficult because there are too many spots. The optical crowding, the image-based platforms, they have a problem of find all these spots in a very small area. So what is the answer here? Don't forget that this is a sequencer, so we have reagents for 300 cycles. So what we do, we split the whole transcriptome in 6 batches. So in other words, these 600 counts in the first batch are different from these 600 counts in the second batch. So at the end, in this experience for the HeLa cells, we have 6 counts, 5 batches, which means we take 3,000 counts at the end of the experiments, that if you will try to do this on the same time, it would have been not possible to distinguish. So this is the protocol to support that. So you use this direct sample sequencing to do cell typing. And here we just co-culture 4 different cell lines. We mix them. We load it to the well, fix, and on the machine. And the next day, we were able to distinguish these 4 cell lines only by reading the RNA. Again, no library prep, no cell partitioning. Just load your cells, put some cell paint markers, and next day you will be able to see in your samples what is your cell types that you have there. And last but not least, I like this one. These you can use it for targeted sequencing, and this is for optical pool CRISPR screens for knockout. So here is a collaboration with Gentech. They have an AVITI24, and they do studies for biomarker detection and drug discovery. So the experiment here is that you have a cell line with a Cas9 and the nuclease that makes the cut, and a pool of infected with a pool of 500 guide RNAs, and these cells were cultured on the one well plate, the load to the system, and after 48 hours, that took more time, what you get out is you get the cell paint, you get detection of 50 proteins, the detection of 350 RNAs, and you can actually now detect where the guide RNA is in which cell. So this is the first time that you can link genotype to phenotype in order to understand the functionality of the gene. So I talk about this, and that already for the optical screens is available. Spatial transcriptome will be available at the end of the year, and next year, all these capabilities, they will move to tissue. Early next year, they will be commercial also for FFPEs. So I hope I convince you that AVITI24 can actually see the true complexity of your biological data. You can check deeper with 5D. And of course, with that, I would like to thank you for your attention. And we are outside in the booth. Whoever has a question to please join us.
Thank you very much. So now we have the last sponsor who's actually doing it online because she's in Paris, and she is the main sponsor of the Stepper people here. So she will present Corning, the hyper technology, a cell culture scale-up solution for cell therapy processes. And her name is Annette Funfank, and we are very happy to have her here. Can you hear us? Yes, we can hear you. She's not on now. No, we see you. Not your slides yet. Yeah, I think it's the internet. Because now we don't have internet again. Okay, we have some technical problems. I'm so sorry. So, if you want to get a coffee first, could take a while. No?
So, okay, thank you very much. Well, I don't know if there was an introduction meant.
Yes, I
but,
introduced you already.
Okay, thank you. I didn't see that. Okay, thank you very much. Yeah, happy to have the opportunity to speak here at this event. So before I go a bit more in details on the Corning hypertechnology and how it's used for cell therapy, I thought it may be interesting to share some information on Corning. So Corning was founded in 1851, and actually everything started in a small town called Corning near New York City. So the base industry was glass, and we as Corning, we have core value of innovation. So that what we did in the last 170 years, innovated a lot of products. So now we have 72 plus manufacturing sites, 10 plus R&D facility, that's globally, and 54,000 employees across 40 countries. We are ranked Fortune 500, 324, and we have a core sales in 2024 of 14 billions. Now, maybe you know Corning Life Science Technology, but actually we have much more major businesses. So besides Corning Life Sciences, we have optical communication. So these are the optical fibers. We have display. These are the flat TV screens, mobile consumer electronics. So you use every day. So it's the glass of the iPhone, and also the automotive, where more and more very thin glass surfaces are integrated. For Corning Life Sciences, so here we offer products for you, for R&D, on the standard core cell culture, but also 3D. So we have a big portfolio of 3D cell culture, everything to make organoids and spheroids. And then we have a big part of bioproduction, so products actually adapted to produce a lot of cells or products from cells. So one sector is the cell therapy, but also the classical bioproduction, that would be vaccines, antibodies, and proteins. So this is when you go more in the process development, clinicals, and manufacturing. And then another part is the clinical and diagnostics. So here we have two products are depicted. For sure, there are more. There's everything for diagnostics like tips and plates, and then also at the end, products for drug storage and delivery. So the fill and finish, basically here depicted in the picture, is our glass vials. So now let me just introduce the field of cell and gene therapy. Just a quick overview. So the global market in 2025 is calculated as $25 billion, with an annual growth rate of 19% from 25 to 34, and then in 34 the market is expected to be $97 billion. At present, we have 43 cell and gene therapies approved by the FDA. I listed some. That's from the FDA list, starting with the very first released up to the more recently released. But for sure, there are more, as you see, 43, just to have some names and also the diseases they intended to. And then approximately 75 approved therapies are expected by 2030. In a very high level, cell therapy involves the introduction of new healthy cells into patients to replace diseased or damaged ones. We have these two major approaches, which is autologous and allogenic. So autologous is with the patient receives its own cells, and allogenic you have donors which provide the cells for the patients. Now, looking in a general workflow for cell-based therapy, normally it starts with cell isolation activation, and then depending on the therapy, whether there's a transfection, reprogramming, or transduction step. So here the viral vector production, the field of viral vector production is very important. And then what it needs is to produce a lot of cells. So we spoke about clinicals, about manufacturing, so that these are a lot of patients, so the cells have to be expanded and grown in a large amount. And then for sure we have the harvest and wash step, and then everything what is downstream, QC testing, cryopreservation, cell characterization, before we have the final product which goes back to the patient. From the perspective of producing a lot of cells, it's actually not easy, and I think maybe you have this experience in the lab as well. You suddenly have to produce more cells, you have to use more T-flasks, and fast you have maybe a crowded incubator. So we as Corning, we offer adapted platform to grow this amount of cells. Now you have this two points you have to consider when you choose the right platform for a process. It's one is operational and the other is the biological aspect. And from the operational, it would be something like availability, facility space, time to optimize the process, platform flexibility, media use, process control, automation. So all that's placed together to decide for the right platform. And that brings me now to the hyper technology. Hyper stands for high yield performance vessels. So we have the three here depicted in the picture. We have the smallest one is my cursor, is the hyper flask. And I want to explain the technology using the hyper flask as an example, and then also speak a bit about the bigger vessels. So what you have here in the picture is the hyper flask next to a T175. I think T175 you regularly use in the lab. And you see the hyper flask has the same size, same special footprint. But the big advantage is that you actually, when you use one hyper flask, you actually work with 10 175. So produce the cells of 10 175. And what are the big advantages here is for sure, I try to give you already the picture of a crowded incubator, as you can see here. Actually, you save incubator space, but you also save storage space. And on top, handling 10 against one flask, I think it saves also time and cost at the end, and it's also more sustainable, seeing the plastic. So it is a bit different technology, and I want to explain you how it works. So in standard cell culture, you need a vent cap, you need a big headspace for a good gas exchange for the cultivation of the cells. So this all you don't need anymore in the hyper flask or hyper technology in general, because you can imagine you compress basically a flask on a thin layer. You see my red cycles here, so this is basically an illustration of that. So you have one thin layer and you assemble then several layers together. In the case of the hyper flask, it's 10. And they are not connected, so the air can render. They are only connected in what we call the manifold, so on the top here. And on the bottom of each of these layers is a very thin film. So the thin film allows the gas exchange. So basically that's how it works. So the gas exchange is now on the bottom where the cells are located. And for sure for the handling, for the cell culture, typically what you do in a flask, you add a minimum up to a maximum volume of your flexible in a certain range to add your media or cell suspension. So for the hyper flask and in general for the hyper technology for cell suspension, for seating the cells, you have a fixed volume. But when you look at the volume per centimeter square, it's in the range what you typically use for a T-flask. So what we recommend is 0.2 to 0.3 milliliter per centimeter square, and that you work with 0.3 in a hyper flask. So it's not much more media you have to use. It's in the range. For the microscopy, you can put the hyper flask under a standard microscope. You cannot image all the layers, but you can image the first two and the last two layers under a standard microscope. And as it's no vent filter, you can also turn it around. Now for, I mentioned it, the clinicals. For the manufacturing, you need bigger vessels, you need closed system solutions. So there we have the HyperStack. They offer even more cell culture surface. They are, the technology is the same than the hyper flask, so I don't explain it again, but they come now with closed system solution. They are GMP compliant, so they can be implemented easily in a GMP process, and they are scalable products. Now from the application, and it focused on cell therapy for sure, that was the topic today. They have a range of application, but the largest I will have highlighted here So typically is the viral vector production. I mentioned it in the workflow already. So AAV and LV production with normally the classical standard producer cell lines like HEC or Vero, but also others. Another big field is stem cell expansion or stem cell production, MSC in this case, mesenchymal stem cells, from various sources. And then a bit more recent field is the extracellular vesicle production, for example, from MSCs. That's also one field of application. So this is more focused on adherent cell culture, but I also want to highlight that this technology is used for suspension cell culture. Here in this case, it's more for antibody production. These examples here for CHO cell and hybridoма cell expansion and antibody production, but more and more recently also used for T cell expansion, so immune cells. And I think that's basically what I wanted to show you in my time I have. Just at the end, I want to say that we have a very strong field application and scientific support team in Europe, but also globally. We work also very closely together globally. So we are here to support you on any scientific, technical questions. So if you want to know something, want to discuss with us, always reach out. So then that's it from my side. Thank you very much. And I'm happy to take some questions.
Thank you very much, Annette. Are there any questions from the audience? Okay. No, I think you explained everything very well. Yes, so if you want to reach out to Corning, please do so. And thank you again very much, Annette. Yes, there was supposed to be a booth outside, but unfortunately there's not. But you can find all the information you need on their website, I'm sure. And yes, thank you very much.
Exactly, and always reach out.
Yes, thank you.
Okay, thank you very much, Leah.
Have a nice day. Bye.
Have a nice day. Bye-bye.
Okay. And we will continue with the lunch break now.
to do a flash talk now. They're going to have to sit on the first row. And each one will stand up when it's their turn to have their 4 minutes presentation. We'll wait 5 more minutes to start.
If you have a flash talk, please come to the first line.
Sorry, I'm trying to see who is missing.
One flash talk is still missing.
Nikos, Isabella, Katerina.
Και εσύ κάτσε από εκεί για να κάτσε και το φλάστο.
Is Dimitra Pano here? Ah, can you come to the first line because you will be one after each other. So welcome all. Now we have a more fun scientific session. First, we will start with the flash talks. The concept is to present your work in 4 minutes. The time will be strict. This is a more interactive way to present your research. We will also include the vote of the audience in this award. So after each presentation, we will have a recording of the decibels. So you can clap and show your enthusiasm in each presentation. Of course, we also have the scientific committee, but we want also to include your vote. And after that, we will have the scientific image contest. So for the flash talks, you will go one after the other, and we can start now.
Okay. Hello, everyone. I'm Nikos. I'm a first-year PhD student working with Dr. Nikos Vakirlis here at the Evolutionary Genomics Group. And today we're going to be talking about antimicrobial peptides and our efforts to discover them in the animal kingdom. So these peptides are short amino acid sequences contained in larger proteins, which we call precursors. This is a precursor, and the green part here is a mature peptide, which when enzymatically cleaved, it acquires an antimicrobial activity. So this set of peptides is really diverse, and they can be found in all organisms as part of their innate immune system. And they're killers. What they do is that they kill pathogens by causing cell lysis. So we really love them, firstly, because they pose ideal candidates for alternative antibiotics, and also their diversity makes them a great space to study complex evolutionary relationships, try to understand how all this novelty arises, and if we're lucky, maybe find some cases of de novo gene emergence. So this is the current picture of the animal lamps in the databases. We strongly believe that this is an underrepresentation, so we built a custom pipeline to try to discover even more and all the novel ones that we could find. So what we did is we got MadeDB2, a curated database which contains almost 1,000 genomes and transcriptomes from metazoa, and we gathered all the available proteins. We filtered for smaller ones in order to get our precursors of interest, and then using cleavage site prediction, we were able to isolate the mature peptide amino acid sequences. Since they're so divergent from one another, we have to rely on machine learning classification. So we used 4 different tools, and we got predictions whether they believe that they are AMPs or not. As you can see here, yep, more than 200,000 cases have at least one positive prediction. But for now, we have decided to go forward only with the strong candidates, the set with a little bit over 10,000 cases. This is their normalized distribution across the different species. We can see that some species are hotspots, some are deserts, and an interesting case is the mollusca phylum, which contains more than 3,000 candidate AMPs. So we compared these novel sequences, new sequences, against the known ones, and we found almost 6,700 novel sequences, meaning they do not match anything in the databases. And when we cluster them into protein families, we were able to almost double the AMP family space, which is really exciting. So as our project keeps going, our future goal is to, first of all, fine-tune the pipeline, apply it to more data, and also integrate the 3D structure in order to find structural homologs, novel structures, and how these small molecules evolve during time. Thank you all. Thanks to the team. Thanks to our funding agent, and hope you liked it.
Thank you very much.
Hello everyone, I'm Isabella, and today on this flash talk I would like to share with you how we combined deep learning tools with transcriptome analysis to uncover cryptic splice sites in breast cancer. So, alternative splicing is one of the ways our cells create diversity. From a single gene, it can produce many transcripts. But in cancer, this process might go wrong. Somatic mutations can destroy splice sites and activate cryptic ones, or even create new cryptic exons within introns. So this study was aimed to systematically identify such mutations in breast cancer to confirm whether they really change transcripts, and to ask whether any of them might have clinical relevance. So, to address this, we analyzed data from 89 breast cancer patients in the Cancer Genome Atlas. For each patient, we have variants derived from whole genome sequencing, matched RNA-seq, and clinical information. From the variants, we focused on those in intronic and exonic regions and annotated them with Splice AI, the deep learning model that predicts whether a variant is likely to disrupt splicing. We also assembled transcriptome to identify novel isoforms. And bringing two layers together, we compared expression between patients carrying mutations and those without them. Lastly, we matched predicting splicing disruptions with observed transcript changes, classifying events by their type and directions, and asking whether any of them were associated with patient survival. And to ensure reproducibility, the whole pipeline was automated with SnakeMake. As a result, we began with the huge space. It was more than 13 million of single nucleotide variants and indels. But after applying filters and Splice AI predictions, we narrowed this down to a few thousand candidates that might alter splicing. On the transcriptome side, the assembly revealed more than 400,000 transcripts, including thousands of novel isoforms, and yet only few hundred variants showed significant expression between carriers and non-carriers. Interestingly, that just 18 SNVs and 49 indels showed direct agreement between predictions and transcript changes. For example, in H6PD, a cryptic acceptor splice was activated, and in AF4, an indel produced novel transcript site. And when we turned to survival, we found only two significant associated with patient outcome indels, but they did not overlap with isoform changes. So what does this tell us? Indels stood out as having stronger and more interpretable effects on splicing than single nucleotide variants. Loss of F events was linked to transcript downregulation, which is consistent with exon skipping and nonsense-mediated decay. Not every predicted size had transcript support, but this may reflect technical limitation or RNA decay, not necessarily false predictions. Overall, this framework bridged deep learning predictions with transcriptome evidence, offering a mechanistic view of how mutations disrupt splicing in breast cancer. And looking ahead, it provides a foundation for scaling to larger cohorts and for integrating long-read sequencing. And thank you very much.
Thank you.
Hi everyone, my name is Katerina, and today I will present you my project titled AI-driven characterization of CNS cells, classification, morphology, activation state, and interactions. If you have ever looked at the night sky full of stars, you can understand how impossible it feels to understand each tiny point of light. That's how it feels when we're studying the brain. There are millions of cells with different characteristic morphologies that constantly interact with each other. But how do we make sense of them in order to understand brain function and pathologies? We need to understand what the cell is doing, what they are, how they interact. And up until now, we've been trying to do that the hard way, by hand, like trying to map an entire galaxy with a magnifying glass. So, the gold standard manual immunostaining protocols are what we currently rely on. Sectioning, immunostaining with antibodies, imaging and analysis that can often lead us to pictures that look something like this, confusing, blurry, and packed. This causes some barriers that mainly have to do with human limitations like objectivity or bias, technical dependencies like the resolution of the images, workflow challenges like the quality of the antibodies that can lead to time-consuming and costly solutions. But what if we could teach a brain to see and analyze the cells for us, as better as or even better than a human scientist? So that's exactly what I try to do. I try to develop an AI-driven automated pipeline for the classification, identification, and characterization of CNS cells in confocal fluorescent images. We begin again with the same pipeline, with a key difference that we don't rely on selective and expensive antibodies, but we can use genetically encoded fluorescent markers like GFP that can save us both time and money. And the main advantages of this approach lie on its objectivity, robustness, consistency, speed, and cost effectiveness. When I first applied this tool in fluorescent confocal images from brain tissue from different regions, experimental conditions, and resolutions, the results were striking. First, this AI tool managed to identify glial cells, microglia, astrocytes, and OPCs with clear-edged masks and bounding boxes, and generated cell type and cell count reports with the confidence scores for each prediction and the visualization of metrics in charts. Then it pinpointed the interaction sites of cell-cell interactions, and beyond all, let's say, these static measures, it also managed to classify with color-coded masks the cells in a per-activation state, normal, intermediate, or activated phenotypes based on well-established morphological features, and generated cell morphology and cell interaction reports, activation state per cell and per image reports, and the confidence scores for its predictions. And like that, we can say that the static snapshot of cells was turned into a dynamic image. So let's see the whole road that led us here. First, we begin with the manual self-classification methodology that we currently rely on that can be subjective, slow, and costly. Now I believe it is time that we are trying to develop more automated ways that will lead us to faster, scalable, and more accurate solutions. But the journey doesn't stop here. The next steps of this tool contain the expansion of the data sets to different cell types and cell tissues, the incorporation of 3D cell analysis, the implementation of explainable AI techniques for transparent decisions, and some usability improvements that mainly have to do with user-friendly graphical interfaces. My goal here was not only to automate the workflow, but to provide an accessible and powerful tool that will contribute in reshaping everyday neuroscience research and development. Thank you very much.
Thank you.
So, hello everyone, I'm Bulgari Despina, and I have recently joined the Bioinformatics Lab here in Pasteur with Professor Hadjigeorgiou, where I will carry out my master thesis. So, today I will present you the first step for my master project, which focuses on microRNAs. So microRNAs, last year they even got a Nobel, but we know almost nothing about them. And I will try to fill a little bit this gap. So currently we have two resources, the MIRGEN-DB and the MIRBase, but they only contain information about the precursors and not the genes that microRNAs are come from, produce the microRNAs. And also they don't even have the same kind of information, and sometimes they even use different names for the same microRNA. So this inconsistency slows down progress and makes it difficult to compare results. We need a more universal, more accurate annotation scheme for microRNAs. And we decided to compare the data from these two resources with a new sequencing technique, which are called CLS, or capture long read sequences. And CLS gives us high quality of the full transcript. And that means from the start until the end, as one piece. And this is important and different for the short read sequencing techniques, because we have the whole information in one piece and not in fragmented small pieces that we need later to assembly. And in this way, we miss information. So we compare this data from the CLS with the gene code annotation, and to see if there is an overlap, and if so, with which genes, like with protein coding genes, long non-coding RNAs, pseudogenes, and so on. We also checked if this overlap is happening on the same or on the opposite strand. And if we have no overlap at all, then maybe these microRNAs originate from unannotated regions. So what did we got? We can see here for the MIRBase, for example, the landscape is a little bit different between them. For the MIRBase, we have 605 microRNAs with no CLS coordinates, and that means that we don't know something about their primary transcript. And the 595 of them have an overlap with protein, mainly with protein coding genes and long non-coding RNAs, and always on the same strand. The situation is different for the MIRGEN-DB, because here we can see that we have some overlaps with the opposite strand too. And this means that MIRBase suggests that the microRNAs might have the same transcription unit as the genes, and MIRGEN-DB that microRNAs might have an independent one. So the key point here is that we managed to have an overlap for like 80% of the microRNAs, and this is an info that we didn't have before. So to conclude, we did manage to provide a genomic context for the microRNAs that had no CLS coordinates, and we saw that the majority of them are not orphans, but they actually indeed they are within known genes. And as we refined this process, we made a solid step to have a more universal, more accurate annotation system that will improve not only communication but also reproducibility and ultimately our understanding of the role of microRNAs in biology. Thank you very much for your attention.
Thank you.
Hi everyone, I'm Dimitra Panu from Alexander Fleming Biomedical Research Center, and I will talk about LLM farms for biomedical question answering. So the motivation here is there is an exponential growth of biomedical literature. We don't have time to read so many papers, and the experts, the biomedical experts, need assistance. To be able to answer biomedical questions and to be able to be in the front line. So we don't need systems that handle domain-specific terminology, but also to know how to respond where we want one answer, exact answer. For example, is this related to cardiological diseases? yes, no, we need a strict answer, and where to have multiple perspectives. So this is our work. We try to answer biomedical questions. So we separate the questions to 3 categories. For example, this, we want a strict answer, so it's a yes or no question. This needs a quick answer, short, like 5 things, so we call it factoid. And this one is a list of items, which are inhibitors. We want a list of items. And the process is, we have the query, we have PubMed abstracts, so we find the abstract and that is related to the specific question, and extract the related text. After using this related text, we extract the final answer. Usually, the most of the systems try to experimenting with different models and try to find the best model to build for the system, to have the best answers. But we work on groups of models. The key idea is not to have one model to answer a question, to build a ChatGPT, but to build multiple ChatGPTs, to say it like this, to extract like a farm the answers. For example, the genie asks what are the common side effects of imatinib. So we have edema, nausea, and rash, and collects the answers and try to respond a more full answer. We tested 13 different large language models, the state of the art, and evaluated more than 8,000 combinations. For each question type, we built a different strategy, and this is the key idea. The fun fact of this is that they work better in smaller groups. You expect, okay, we have 13 models, all of them to answer better the question, but it's like humans. They work together in small groups of 3 to 6 models. In factoids, we see that the group of 6 are better, and in list type of questions, the groups of 3. And here is the best union model, and each model individual, their accuracy. We submit these results to a competition, and we won first place in some categories. This is the summary of models. And to conclude, we want to expand this technique to build a web application in order for the people to try it. This is our team. I want to thank my supervisor, Martin Rechko, and thank you very much, all of you.
Thank you very much.
Hello everyone, I'm Irina Karamichali and I'm going to present the LIPS project. Okay. The main goal of this project is to integrate multidisciplinary expertise in a learning and adaptive European pandemic preparedness system. It is important to detect, understand, react, and respond to a future outbreak with a well-prepared strategy using real-time genomic data. This project aims to achieve this goal with the research from 5 European countries, as you can hear here, including Institute Paris and our laboratory that participates at the level of the countermeasure screening and development. Our main goal is to develop viral-like particles for vaccine development, drug testing, molecular and immunodiagnostic assay using viruses with pandemic potential. Now, VLP is a nanostructure with self-assembly feature that contains structural viral proteins, and the molecular morphological characteristics of this VLP are similar to the authentic viruses. Due to the lack of the genetic material, the virus is not able to replicate and it's not infectious. The aims of our project was first to produce SARS-CoV-2 and West Nile VLPs using a cell culture system, and second, to validate the functionality of these VLPs through cellular assays. So we performed for this reason, we performed transfection experiments with plasmids expressing the structural proteins, as you can see here, and we have collected these supernatants, and we performed ultracentrifugation on sucrase gradients. The results from the biochemical characterization indicated the presence of all the structural proteins, as you can see here, and for both VLP types, and furthermore, NT analysis, nanoparticle tracking analysis, showed that the size of both VLP types goes in the expected range for each virus, here and here. We are currently now checking the morphology of both particles with transmission electron microscopy. Next, we have proceeded with a series of cellular and molecular assays to evaluate functionality and immunogenicity of these VLPs. And here you can see that when incubated with neuron-like cells, both VLPs can cause cell death. From here, and incubation with VLPs. So, concluding, we have a successful generation of both VLP types that was ascertained by the presence of the relevant structural proteins. We have the characterization with NTA, and both VLPs enhance cell rounding, disappearance of neurites, cell death following incubation of cultures with VLPs, but West Nile exerted faster. We know all that is a neurotropic virus. And I would like to thank all the members of molecular biology, the bioinformatics and applied genomics unit, diagnostic departments, the LIPS consortium, and of course all of you for your attention.
Thank you very much.
Hello everyone, I am Georgia and I'm going to present your work on 3D bioprinted models for the study of viral infections. Thank you. So to begin with, hepatitis B and C remain highly prevalent worldwide, while clinical outcomes are highly influenced by endogenous and exogenous factors. And can potentially lead to severe liver disease and even hepatocellular carcinoma. Research is limited by oversimplified 2D cell culture models and animal models that lack range of hosts and biological relevance. To overcome these barriers, our virology laboratory partnered with the Material Science Laboratory to create 3D bioprinted tissue-like structure that provides a more physiologically relevant environment for our studies. To do so, we used Hook 7.5, a hepatoma cell line that supports the entire life cycle of HCV, and NF2, a genetically engineered Hook 7.5 cell line that supports the complex viral life cycle of HBV. As viral stocks, we used concentrated supernatants and patient sera of each virus, and as biomaterials, we used biofunctional hydrogel matrices that integrate biocompatibility, mechanical strength, and bioactivity. To mimic infection of liver tissue, we used 3D bioprinting to assemble the matrices with the selected cell lines into 3D tissue-like structures, which were then inoculated with defined HCV and HBV strains. And finally, the viral replication was quantified by qPCR. Moving on to the results, the two first graphs on the upper part show the viral titers measured by qPCR in supernatants collected at defined days post-infection. And as shown, our model supports robust HCV and HBV infection with high viral titers confirming successful replication. On top of that, on the third graph, we can observe higher infectivity in a 3D setting compared to the traditional 2D. To conclude, our model supports the complete life cycle of HCV and HBV and can serve as enables the study of hepatitis viral infections in a more tissue-relevant environment. These biofabricated platforms can serve as disease models to study hepatitis and other viruses and host interactions, to assess antiviral strategies, and to support drug development and screening. I would like to thank every member of the team who contributed to this work, and of course all of you for your attention.
Thank you very much.
Good afternoon. Hello. I'm Irini Kolya, a bachelor student from Dr. Vasilakis Molecular Virology Lab, and this presentation is about the implication of RNase kappa in Flaviridae infections. Flaviridae are small envelope positive strand RNA viruses with 9 to 13 kilobase genome, flanked by unrelated regions encoding a single polyprotein. Today, there are known 110 species, divided into 4 genera, with human pathogens belonging to orthoflavivirus and hepativirus, the most prominent being dengue, Zika, West Nile virus, yellow fever virus, and of course HCV. RNase kappa is a small conserved protein of 137 amino acids in length. It has 2 transmembrane domains. In vitro, it exerts endorubonuclease activity, but its cellular role is still unclear. Recently, via structural analysis of mammalian brain V80-based proton pump, it has been identified as being subunit F of the V0 region of the pump. This way being implicated in bioregulation of the cell and endosomes, critical in autophagy, cell death, and lysosomal degradation. So far, RNase kappa is known to be necessary for receptor-mediated endocytosis of multiple kinds of cargos, including viruses. Silencing of RNase kappa results in inhibition of the regular canonical clathrin-mediated endocytosis pathway, but also a different non-canonical pathway linked to viral infection, probably through inhibition of the V80-base activity, thus hindering viral entry and replication. In our lab, we specifically tried to see the effects of RNase kappa function activity in Hook 7 cells in a Flaviridae infection. Our first results showed that in Hook 7 cells, dengue but not HCV infection, it did reduce the expression of RNase kappa 48 hours post-infection, as showed through RNA levels and protein levels. Then we decided to test the effect of RNase kappa silencing in infection. We indeed saw what was expected in entry with reduced entry 1 hour post-infection. Although in replication kinetics, the result was not the expected one, as we saw increased infection as seen through RNA, luciferase activity, and used as reported, and protein levels. Next, we sought to investigate what is the stage of the viral life cycle that is affected through going over entry through electroporation. We electroporated both full-length DVR2A genome and also subgenomic clone lacking structural proteins. Again, in both cases, when RNase kappa was silenced, replication was increased. We also tested the infectivity of the infected culture media in second route of infection, and again we can see that when RNase kappa is silenced, the supernatant of the culture is more infectious. Well, because RNA-seq has also been proposed as a positive regulator of immune responses at apoptosis, and these two pathways are heavily regulated by dengue, we next examined these pathways. What we saw was that IC56 and IC15 they were induced as expected when RNA-kappa was not silenced, but when RNA-kappa was silenced, IC56 was heavily impaired, IC15 was expressed but at lower levels. Regarding proliferation, by judging cell viability through ATP levels, we can see that dengue doesn't lower ATP levels so much in silent cells as much as control cells. The same effect was shown in cell proliferation imaging. Now, regarding autophagy, we saw through Western that the RNA-kappa silencing seems to block the autophagic flux. We can see this because of through the increase of the lowering of RAB7 expression and the increase in LC3B2, showing a blockage at the autophagosome stage. This is an effect also exerted by dengue itself, so these conditions favors dengue. Regarding apoptosis, infected control cells showed higher BACs and lower procaspase 3 and 9 levels, which means that in silent cells, apoptosis pathway was less active than control cells. In conclusion, our takeaway is that dengue downregulates RNA-kappa expression in its later stages, while RNA-kappa reciprocally seems to not favor dengue replication because its silencing seems to inhibit apoptosis autophagic flux and promoting viral replication. Our goal for the future is to better understand the mechanism and of RNA-kappa implication in dengue virus and also in immune regulation and cell death. Thank you very much.
Thank you.
So, hello everyone. I wanted to ask if anyone here has ever been in a toxic relationship. Because there's one going on at the moment between HIV-1 and the mitochondria. So you might know HIV-1, it's a retrovirus that's been working at Pasteur since 1983, and it is a master evader of the immune system. It really likes to romance the mitochondria in the beginning of their relationship because it wants to shut down a protein system called MAVS that helps the cells detect the virus and send in the appropriate responses. But why the mitochondria are very interesting to the study is because when they're damaged, they can act as key signaling hubs that tell our cells to send in the proper defenses after viral infection. And one really interesting protein that they send in is called ApoBec3A. And I like to think of ApoBec3A as the messy best friend in this toxic relationship, because while it's meant to help, it can sometimes go ahead and make things a lot worse. But before I get into this, I just wanted to go through why I think it's a toxic relationship. Because there were red flags. So the first red flag was that there was just bad energy in the cell. So looking at the oxygen consumption rate over time, with our infected cells in green and our non-infected in black, we can see that at the basal and at the maximal level, that the infected cells were able to produce a lot less energy. So yikes, not very good. And the second red flag was the mitochondria began to change how they look. So using confocal imaging and a machine learning pipeline that I've built and I'm happy to share with anyone, is we can see that there's a higher number of mitochondria infected cells, that these mitochondria are bigger in size, and that they decrease in their spherical shape. So they're forming these kind of longer tubule-like networks, which is highly indicative of stress. So that leads me to the biggest red flag of them all. The mitochondria begin to call in for backup from the best friends. So when mitochondria are stressed, they release mitochondrial DNA into the cytosol. And this sets off a lot of different DNA sensors, which ultimately results in the upregulation of type 1 interferons. So these are kind of like the best friends texting in the group chat saying, hey, you're in danger, you need to send in the appropriate responses. And this is where we see the messy best friend arrive. And while the messy best friend is supposed to help by restricting the virus, It actually can cause a lot of chaos in the cell by editing genomic DNA of the host, causing apoptosis in cell. It can also damage the mitochondrial DNA and prime it for degradation, which is going to suppress our immune response in the cell. So I was trying to figure out, as my next step in my PhD, of why is this all happening? So using a doxycycline-inducible system that expresses A3A and submitting these for mass spectrometry analysis, I've identified some really interesting proteins and interactors that I'm doing some functional analysis on at the moment. And if you want to find out more about this, you'll have to talk to me over wine later on in the garden. So thank you.
Thank you very much.
Hello, everyone. Today I'll tell you how this tiny insect, the fruit fly, helps us understand the sleep problems in neurofibromatosis type 1 or NF1. The title is When Fruit Flies Can Sleep, What NF1 Teaches Us. So, neurofibromatosis type 1 or NF1 is an autosomal dominant multisystemic disorder. It's one of the most common genetic conditions that affects behavior and cognition, and it's inherited by, it's caused by inherited or de novo mutations in the NF1 gene. The NF1 gene encodes for neurofibromin. As you can see here, it's a large multifunctional GTPase activating protein, and it's a negative regulator of RAS through the GRD domain. Most of people know that NF1 is the syndrome with the neurofibromas. As you can see here, is the tumors of the PNS and the CNS. But most of the patients exhibit many sleep, attention, and learning deficits. Among the sleep problems in NF1 patients, the most significant is sleep fragmentation. So the big question is, why does the loss of NF1 leads to poor sleep? And here is the, sorry. Okay, this is a sleepy fly. So you use the Drosophila melanogaster to study this question. So we know that flies have their own NF1 gene called DNF1, which is similar to the human one, and is broadly distributed in the brain, as you can see in this image. And we know that flies love to sleep. They sleep around 10 hours per day, just like human. But the sleep in Drosophila is based on some other behavioral criteria, like an activity of at least 5 minutes. So we aim to study the NF1 deficient sleep phenotypes using the Drosophila model and the Drosophila activity monitors, which is the smartwatches for flies. And we found that the NF1 null mutants have decreased sleep, decreased about number, decreased about length, which is translated as sleep fragmentation. And then we answered the question, which neurons require DNF1 to mediate normal sleep? After a large screening, we conclude that DNF1 is required in cholinergic neurons to mediate normal sleep. So then, we continue with the expression of only the GRD domain, the catalytic domain of the protein, within the cholinergic neurons, and we found that their sleep is improved. And then when we abrogate the RAS1 protein within the cholinergic neurons, we found that their sleep also improved in a lower level. So we conclude that neurofibromin regulates sleep homeostasis by modulating RAS signaling in cholinergic neurons, and is RAS1 dependent. And the take-home message is that better sleep could mean better life for NF1 patients. Special thanks to Dr. Skoulakis, Dr. Irene Maria Georgada. Don't miss her talk in the next session. The organizing committee for let me do this talk, and of course all of you for your attention.
Thank you very much. And the last one.
Hello, everyone.
I'm Ellie from Hellenic Paris Pasteur Institute, and today I will talk to you about mechanisms of early synaptic dysfunction in A53T pathology. We often think that Parkinson's disease appears late, but today I will prove you that the first alterations happen much earlier at synapses. The most strongly PD-associated protein is the alpha-synuclein, a small presynaptic protein that its normal function is still unknown, but we know that has a role in synaptic vesicle maintenance and plasticity. Aggregated and misfolded alpha-synuclein accumulates gradually in presynaptic terminals and leads to synaptic dysfunction. Our team studies the most common mutation of alpha-synuclein, the A53T, which causes early onset Parkinson's disease. Transcriptomic analysis of human-derived neurons and neural progenitor cells revealed a dysregulation of genes involved in synaptic signaling, suggesting that early defects happened in synapse formation. And this raises a key question: how this mutation leads to early synaptic dysfunction? To address this, we employ both a transgenic mouse model that overexpresses the human A53T specific neurons and human-derived neurons bearing this mutation. These systems allow us to investigate early changes in molecular, cellular, and network level. To see whether changes happen early. Before any sign of neurodegeneration, we isolated synaptosomes from 6-month-old mice, and proteomic analysis revealed that presynaptic proteins are clearly dysregulated and are related to synaptic vesicle cycle, endocytosis, and recycling. These results are further reinforced by analysis of the synapse ultrastructure, and as you can see in the bottom figures, in the right side of the presentation, the A53T synapse has diminished number of synaptic vesicles and distorted PSD formation. Then we asked whether these defects affect synaptic composition and network properties. Unexpectedly, the A53T neurons form more inhibitory synapses and fewer excitatory, and in fact, both in vitro and in vivo, we observe more GABA-positive cells, indicating a stronger inhibitory drive. At network level, the dendritic architecture is less complex with reduced map to positive area, and looking closer to dendritic spines marked with the antibody drebrin, we observe that the alpha-synuclein and the drebrin are abnormally close and highly colocalized. When we checked the activity of our A53T neurons, we found that they fire less and they may have shorter and more frequent bursts and irregular synchronicity. So, this altogether point the excitatory-inhibitory imbalance that affects network function and complexity. Can we restore it? The answer is yes. Using a compound called nitrosynapsin that blocks the extrasynaptic NMDA receptor, we achieve to rebalance inhibitory and excitatory synapses to wild-type levels, reduce the number of GABA-positive neurons, increase the complexity of our network, and decrease the colocalization of alpha-synuclein and drebrin. To make the long story short, today we see that dysregulation of synaptic proteins in the A53T brain happens really early prior to neurodegeneration. The A53T synapse has a distorted organization, and the A53T neurons present an aberrant synaptic and network function. This synaptic imbalance is largely restored upon treatment with nitrosynapsin, which suggests that early synaptic dysfunction could serve as a therapeutic in Parkinson's disease. Thank you.
Thank you very much. All of the flash talks were very creative and very exciting. Now we will move on with the scientific image contest. The audience will vote for the best image. And after that, we have the neuroscience session. the plenary talk of Professor Gravanis, and at 6 o'clock is the ceremony of the awards. So there will be a Best Oral Presentation Award, a Poster Award, a Flash Talk Award, and the image. So stay with us, and then we will have the closing party in the garden. Hopefully it won't rain. So yes, we're ready for the scientific image session.
Hi everyone. So the procedure for this session is simple. Me and Anastasia will be presenting each image separately. We'll be calling the person responsible for each image to the podium so that they could say us a thing or two about the picture. And at the end of the session, everyone will vote for their favorite image. We will gather the votes and announce a winner. And hopefully the winner this year will be the image that poses on next year's handbags. So we will be starting with the first entry. It's by Eleni Paximadi. The image is called The Flirt of Nature. Unfortunately, Eleni couldn't be here with us today. So Anastasia, could you read the description of the photo?
On my balcony, I have a flower pot where I put breadcrumbs for the sparrows to eat. Shortly after the birth of the newborn and after they start flying, the mother stood with her baby in the flower pot, took pieces of bread with her beak and fed her little one. It's not the obvious gifts of lovebirds. That's why in research we often have a different way of thinking and interpreting.
Okay, so we'll be moving on to the next picture. It's by Angeliki Giannouli. If Angeliki is here with us and would like to share a thing or two about her photo. Okay. Moving on to the next one. It's called Neural Stem Cells in Neuroimaging by Irini Portokali. Irini is here with us and would like to say a thing or two about her photo. Where is everyone?
Hi. So this is my figure. It's the dentate gyrus of the hippocampus. And with the red, we see the astrocytes all over. And the blue structures is the granule neurons of the hippocampus. And when I first saw this picture, it reminded me of space nebula, that there are particles all over. So I named it dentate gyrus nebula. That's it.
Okay, we'll be moving on to the next one. It's called Neural Kaleidoscope. So if Elpiniki would like to join us on the podium.
Hi. So this is a totally processed image. Actually, the original image is 1/4 of it. It's from embryonic mouse brain, day 16. With pink, you can see the nuclei, and with blue alpha-synuclein, and with green, some microglia cells. That's it. I would just like to thank the imaging unit of Hellenic Pasteur Institute, which is great and expanding. And we spend a lot of hours there, so when the results are at least beautiful, we feel a little bit better about it. Thank you.
So just a quick tip. Everyone that has submitted a photo, we would like you after the session so we could all get pictures of you with your respective photo. So don't leave. We'll be moving on to the next one. This one is called microglial attack, and I would like Makis to join us on the podium.
Hello, everyone. We observed a cerebellar tissue. We have stained tissue with IBA1 for microglia and PSD95 for synaptic marker. So we observed during experimental autoimmune encephalomyelitis models, animal model FMS, how microglial cells aggregate and migrate to PSD95 positive Purkinje layer, and it's highlight the role of microglia in the progression of the disease, and we'll discuss more about this later on my talk about microglia and multiple sclerosis. That's all from me.
For the next one, I would like to invite last year's winner to the podium. Olivier, if you would like to join us.
Hi. So the title of this picture stems from the fact that when I was looking at it, the greenish astrocytes lurking in the back seemed a bit slimy to me, and it was very Halloweenish. So with green, we see a mutant astrocyte, and with orange, we see the mutant neurons from our Parkinson's disease model. And basically, when we first obtained this image, I was kind of looking at it as neurons attacking the astrocytes. It was looking a bit, poor little astrocyte, but little did we know then that this astrocyte was the actual hidden murderer lurking in the back. So, yeah, that's it. It's obtained with a standard immunofluorescence protocol with confocal imaging here in the Hellenic Pasteur Institute. That's pretty much it.
Okay, the next one is by Elisabeth Kalliopi-Akrioti. It's called mouse hippocampal neurons.
Okay, indeed, it's mouse hippocampal neurons at the in vitro 20. They were stained with MAP2 and alpha-synuclein. As you can see, it's a processed image. However, I love it because it gave me motivation. It was one of the last images I took before our institute closed for reconstruction works, and one of the first images I came into after a personal journey. And it motivated me to begin again science, start dreaming, and love the magic of science. That's it.
The next one is called astrocyte embracing a blood vessel. And if Evangelia is with us, I would like her to join us on the podium.
Okay. Hello, everyone. This image shows an astrocyte which embraces a blood vessel with its end feet. And this image was it was acquired with 2-photon intravital microscopy inside the brain of an alive anesthetized mouse, which it is also transgenic and expresses the CFP in astrocytes. And it shows the close proximity of the astrocytes to the vasculature, And with all this close interaction, which helps to maintain the blood-brain barrier integrity. And I found out this image while I was doing the image analysis. It was very impressive. And it looks like, I don't know, someone who is running. And his hair is blown by the wind, maybe. So it was very impressive. Okay, thank you.
Okay. The next one is by Stefania Kapsetaki. It's called the Bioelectric Atlas of Aging in Hydra.
Hello everyone, I'm Stefania Kapsetaki and this is Hydra oligactis. Hydra is a small invertebrate animal that lives in freshwater environments like ponds and lakes, and it has a remarkable ability to regenerate. So if you cut it in pieces, each of its little pieces can grow into a whole new Hydra. It also has a remarkable ability to age or not age, depending on the temperature. Wouldn't we all like to live forever just by changing the temperature? So until then, we can study and learn more about aging and immortality in these Hydra. This is an image from a confocal microscope. It was quite difficult to take this image because the Hydra were moving all the time. And it's the first time we've been able to look at the bioelectric atlas of a whole organism. So we've used a dye here called FluoVolt, and we can see the relative membrane potential, so Vmem, membrane voltage, of every single cell. So green means that those cells are more hyperpolarized, red means that they're more depolarized. And why is this useful? Well, we can see the bioelectric pattern of old versus young individuals. We can try and find bioelectric drugs that reverse the old pattern to a young one. And I would encourage you to use this system in your model organisms and see whether the bioelectric atlas of aging is common or different in model organisms, and whether we can reverse aging. Thank you.
For the next one, I would like to summon Anastasios to the podium. It's called Lord of the Cells.
Hello, everybody. So, yes, this is Lord of the Cells. I hope you get the reference. We just found a really old tube that was written body pie on it, which is a dye which dyes lipid molecules, phospholipids. And we said, okay, let's use some spare cells to see what it dyes. And as I was navigating, I found this huge Parkinsonian astrocyte, which has this really nice eye perspective. And I don't know what is this hole in the center. Maybe it's where nucleus stands, but there is no autofluorescence. Maybe it is a big vacuole, I don't know. Maybe it's the Mount Doom in Mordor. So this is it. Thank you very much.
The next image is number 11, When the Barrier Breaks, by Nicoletta Riga. Nicoletta, please join us.
Hello everyone. This is an image of a spinal cord tissue from a mouse which experimental autoimmune encephalomyelitis was induced. At the onset of the disease, Evans blue dye was injected through IV to the mouse. Evans blue dye binds to the serum albumin, which and normally, under normal conditions, the dye does not cross the blood-brain barrier. As we can see in the image, the visible red spot represents the presence of the Evans blue dye that leaked into the tissue due to the breakdown of the blood-brain barrier, which also means that there will be immune cell infiltration. Thank you.
The next image, number 12, is Butterfly by Ekaterini Voulia.
Hello everyone, my name is Katia and I am a PhD candidate at the University of Crete.
I want someone to tell me what this image looks like. I mean, it's obvious. Yes. A hand, please. Okay, I will tell that this is a butterfly. And I always think about butterflies like a symbol of freedom. That's why I choose the title Freedom. And for the reason why they can fly from one topic to another and explore the universe, exactly as in research, where our minds can fly from topic to topic and hypothesize and make conclusions and regressions and go from the beginning again. And the irony about this image is that it illustrates a mouse prostate with cancer, and it's taken during confocal microscopy using the cytokeratin markers that target the epithelial cells of the prostate. And we found that we have tumors because we have the aggressive and excessive proliferation of the epithelial cells into the lumens, which is the characteristic of tumors. But I always try to find something good in every bad situation, and the only advice that I give to myself and to every young scientist is to try to find the good in every bad situation, bad day, bad experiment, etc. Thank you very much.
Moving on to image 13, that's Our Pollen Fox by Sofia Maka.
Hello everyone, this is my image for this photo contest. It was taken on a scanning electron microscope called SEM, yes, and it was taken with the support of my colleagues and my friends who, by the way, were very dedicated and support and helpful by bringing this project into life. And what we are looking here are, okay, obviously pollen grains from different kinds of flowers, such as hibiscus, Relia, and morning glory. And that's why it is dedicated to people with allergies. So before starting this project, I have never observed by myself pollen grains on SEM. And when I first captured on SEM the images, I was literally captivated by also the shapes, the different shapes, the geometry, and by the fact that this was the moment that I realized that, okay, different flowers have also different pollen grains, obviously, and by the fact that they look so alike bacteria and viruses. And when I have the images, I asked my colleagues, my family, and my friends their opinion of what they think this could be. And the majority of them told me, okay, yes, viruses. So that was the moment that I realized that this was the image that I have to choose. And briefly, just to mention some things, here in the corners we see some pollen grains from hibiscus. And you look in the right corner pollen grains that they have a very thin fiber, like a sticky thing, like a bridge, like they're holding their hands. And also in the left corner, you see how they are like a ruler, aligned all together. And also in the upper part, we see, by mistake also, they stack on the top of each other like a cluster. And generally all around, we'll notice that there are different shapes and texture, spiky, spherical, and each one suits to the plant that comes from and how it spreads. And yes, just to mention that images of SEM are grayscale, but this is with the help of software. And that's why I choose this photo, because I think it captures the beauty of biological sciences. Thank you.
Image 14, Memory is a Cabinet of Curiosities by Irene Thano.
Another hippocampus. This is a confocal image of dentate gyrus of hippocampus, which is a neurogenic niche. It's depicting newborn neurons in cyan, as you can see, microglia in yellow and nuclei in magenta. And the title comes from several called Cabinet of Curiosities in Art, because it seems to me that memory is our living museum, our cabinet of curiosities. There is always room for something new and strange and marvelous. It's the way to stay interconnected with our collective past and the imagination of our common future. Even though brain was considered up to 30 years ago unchanged, the discovery of hotspots that keep producing brain cells reflect our ability to adapt and evolve as organisms, hopefully also as societies. Thank you.
Moving on to image 15, a neural comet by Nikos Kokorakis.
Hello, everyone. This comet is actually a neurosphere that is a three-dimensional spherical structure consisting of neural stem cells, and these stem cells are derived from and isolated from embryonic murine spinal cord. This is an important system if someone wants to study the action of drugs on neurogenesis or other factors that have any impact on this system, of course, upon their differentiation into neurons or glial cells. That's it. Thank you.
Image number 16, Brainiverse by Maria Margariti.
Hi, everyone. So, if someone asked me today what this is, I would tell that the structure that looks like a worm is the choroid plexus, which is, by the way, one of the favorites of my colleague Irini. I mean, she's obsessed with it. But I brought this picture here today to tell you why I choose this particular one. Because when I started neurobiology, I was completely clueless about this scientific field. But almost 7 years ago, I can say that I've learned so many different things about this remarkable system, the brain. And I mean, let's be honest, all of us have a lot to learn. Just like the functions of the choroid plexus. Do we know all the functions of the choroid plexus? I don't think so. So today, I just wanted to introduce you to my Brainiverse. Thank you.
And this is the last image, number 17, Must Be Love on the Brain by Katerina Tsibuki.
I don't have much to say, but this image shows a cortical neuron from primary culture, from the model day3, which is an Alzheimer model. And it can tell us that even in the impaired brain, love is always present. Thank you very much.
This is the time for voting. I would like if everyone has a piece of paper to vote. So, how many don't have?
Yeah, if anyone doesn't have
Can you raise your hands, please?
So each one of us is each one of you is allowed to vote for one picture because someone has already made this mistake so far. Yes. So we will start from the beginning and Τώρα ή μετά?
Only one.
Yes. Τι τραβηγμένο. Είναι στον όγκο γι' αυτό. Δεν ξέρω. Μάλλον όχι. Πόση ώρα.
Να το βάλουμε
Ναι,
όλα
βάλτε.
μαζί. Είναι 17.
Ναι.
Για 2 μέρες 9. Μισό λεπτάκι.
Θέλει να τις βάλει όλες σε ένα τώρα.
Αλλά γρήγορα. Ναι, ναι, ναι.
Η κάλπη φοράει γραβάτα και περνάει από εκεί.
Αχ, μικρά είναι. Τώρα θα τα βάλουμε.
Περιμένετε λίγο να τα θυμηθείτε.
Ναι, άργησα μάλλον. Αυτό προσπαθώ να βάλω. Αυτό φτιάχνω τώρα. Τι όμως. Γιατί?
Γιατί δεν φαίνονται, ναι. We have a short break and then we're coming back for the neurobiology session. If you want to take photos with your photo. Ναι. If you want to come and take photos with your photo. So we are ready to start our last session, which is the neuroscience session. I would like to introduce Dr. Rebecca Matsas. She's a neurobiologist. She has a long-standing research experience in Hellenic Paris Institute. She has served as a general director and is an elected EMBO member, and also a member of the Hellenic Society for Gene Therapy and Regenerative Medicine, and a governing board member of the Network for CNS Transplantation and Restoration. Also, I would like to introduce Dr. Florentia Papastefanaki. Is a research staff scientist in the Laboratory of Cellular and Molecular Neurobiology and Stem Cells at the Hellenic Pasteur Institute, and she's running the Human Embryonic and Induced Pluripotent Stem Cell Unit. It's an honor to be here with us today. And after this session, we have the plenary talk of Professor Gravanis, and after that, the award ceremony. So thank you.
Thank you, Athena, for the kind introduction. And indeed, we've reached the final session, which is the second session of neuroscience. So it seems that there is a lot of neuroscience going on in this institute. I'm happy to see, I'm very pleased to see. And without any further, I would like to introduce our first speaker, who is Paskalis Theotokis from Monash University, who is also a researcher, as far as I can understand, in University of Thessaloniki, and he's going to present his work on thyroid hormone mimetic DITPA enhances remyelination and oligodendrocyte preservation under CNS neuroinflammation. And this is a sort of collaborative work between the Monash University and the University of Thessaloniki, and also South Korea, in Seoul, etc. So, Paskalis, the floor is yours.
Thank you very much, Professor. Hello, everybody. I'm super happy to be here, and I'd like to thank the organizing committee for giving me this opportunity. Today, I'll get you through the synthetic thyroid hormone analog and how it can promote the myelin repair in conditions of neuroinflammation. Briefly, I would like to state that I'm affiliated with a company that developed a drug for progressive multiple sclerosis. It's called dioprotectome. And currently it's under the development, as I said, but the next year we're going to be in clinical trials phase 2 in patients. So while I'll focus on the science, just keep that in mind, the potential in the translation of this molecule. We also have 2 patents. The last one was filed earlier this year. So multiple sclerosis is basically, as you probably know, an autoimmune disease where the immune cells target the myelin sheath. So the myelin sheath is stripped away around the axons. The myelin is produced by oligodendrocytes. And in patients, this can be a very subtle symptom, like fatigue, up to a very debilitating one, which is motor and vision problems. Globally, 3 million people live with multiple sclerosis, and here in Greece, if you go on the demographics here, it's 20,000 people. Our fellow people, 20,000 people live with multiple sclerosis. So the drugs that we do have in our hands that are called disease-modifying drugs, they do target specific aspects of the disease. However, they only stop or halt the progression and cannot repair the damage done in multiple sclerosis. So a key molecule is the thyroid hormone. We do know that T4 is the molecule that runs in our bloodstream, but it needs to be converted in T3 in order to be functional. This is done by iodinases, and as the name suggests, it takes out iodines. Then T3 is picked up by cells. There are monocarboxylic acid transporters. There are a lot of them, but in today's talk, we're going to be focusing on MCT8, specifically in oligodendrocytes. And why in MCT8? Because we do know that mutations in the gene can bring out a very debilitating disease in children. It's called Allan-Herdon-Downley syndrome. You can see that here, which is basically mental disorder and other motor deficits. So imagine you are T3 and you get into the cell. Normally what T3 does, it goes into the nucleus, it binds into receptors, and the genes are transcribed and so forth. But there is also another way, the non-genomic one, the non-canonical one here. It binds into the integrin receptor. It activates a specific pathway. We're going to be talking on that later on. And eventually, it will activate mTOR, which basically is a protein that is related to proliferation of cells. And in our case, we talk about the precursor oligodendrocytes becoming mature myelinating oligodendrocytes. So we hypothesize that in multiple sclerosis, we do have a downregulation of MCT8. Okay, so this pathway is out. So a nice strategy to convert that would be to give some thyromimetics. We call them thyromimetics because they act like the thyroid analogs. And then we activate the non-canonical pathway, that one. And we did that, we actually tested that in vitro with a thyromimetic called, hold your breath, 3,5-diidothyropropionic acid. Let's call that DITPA or DITPA to make our lives easier. So we've knocked down the MCT8, and what we saw with the use of this compound, we saw that the OPCs were preserved and matured and also formed myelin. You can see here, this is a newly formed myelin in those cultures. So we set out with two goals here. The first one was to take a look to investigate the lipid synthesis in the cells, and more specifically in postmortem tissue, in multiple sclerosis patients, postmortem tissue. And the second one was to capture that in a translational way. So we utilized in two preclinical trials, we wanted to test that this drug, this compound, along with some others that I mentioned later, if they have any kind of neuroprotection in those cells. The hypotheses were that that MCT8 is downregulated in oligodendrocytes, and also that this specific action, the mechanism of action, the pathway, is basically the ACT-mTOR signaling. Okay, those were the hypotheses. We started with the human tissue, as I said, the postmortem tissue. Basic stains, who is familiar with that, the Luxol fast blue to test demyelination, and Bilsovsky stain, which is basically the silver stain, to test for axonal loss. But we also did some other more specific stains, more specific analytic ways to the lipid synthesis, which are those things here, the infrared spectroscopy. At the same time, we did immunofluorescence to check the levels of MCT8. And the levels of MCT8, if you can see here, in the progressive multiple sclerosis, downregulated. So, first question is this only specific for multiple sclerosis? That's the first question. And the second one, since this is downregulated, what about the rest of the molecules downstream the MCT8? So, in order to do that, we went and tested other postmortem tissues, again from human postmortem tissues, from neurodegenerative diseases such as Alzheimer's disease, frontotemporal dementia, Huntington disease, and of course multiple sclerosis. You can see that all of the molecules here downstream of this specific pathway, they're downregulated, but they are more downregulated here in the multiple sclerosis. And we also did some proteomic analysis as well. In order to identify the molecular underpinnings of this, we went ahead and did untargeted metabolomics between the MS tissue and the control one. Okay, and what we saw is the downregulation of 16 metabolic pathways. If you look closer here in those pathways, one of the most prominent one is the one that implicates the formation of pantothenate and the coenzyme A biosynthesis. All right, so there is a specific enzyme, it's called pantothenate kinase 2, or PANK2, which actually converts the pantothenic acid, this is vitamin B5, into the coenzyme A. So it's very critical to understand that because that connects directly the mitochondrial dysfunction to lipid biosynthesis directly. That's very, very direct. Now, we wanted to capture that also in vivo. And our in vivo models, so if anyone here is not familiar with what experimental autoimmune encephalomyelitis is the EI model where we did that in mice. We have an onset of the disease, a peak, and the chronic phase. Here is the clinical score of the animals. We started again with luxolfast. We actually saw that specifically in the lumbosacral area because that's where we can see most of the inflammation happening. We started with the luxolfast blue. We did the silver staining. We did the Western blots, and also we did triple immunohistochemistry to identify the special expression of those proteins. Out of all those outcomes, what we see is that, again, a downregulation of the molecules of the non-canonical pathway. So we went ahead and target that specific mechanism with 3 independent drugs. One of those drugs, so those are the thyromimetics. You can see that they are they're similar together. The first one is dependent on MCT8, so this binds actually to the receptor. It's basically a synthetic T3, whereas the other two are the triac, it's called triac, and the last one is our DITPA, which are independent of MCT8. Keep that in mind. And those drugs were delivered through the esophagus. I did that. I know it's gross, but consider me doing that every day for a whole month. Yeah, it's for the science, right? So it's a very low dose, actually. Okay, so it's a low dose. And we also did all the blood work that were needed for the animals, the liver function, the immune function. We went up to check the stools, the frequency and the morphology. Again, all for the science, right? So out of these 3, yes, the DITPA was the one out of the 3 that actually increased, as you can see here in the last graph, the PANC2 enzyme. This is the clinical outcomes of the 2 independent trials that we got approvals to do here in Monash and this one here in Greece. And this one, the difference is basically we administer the drugs here in the onset, whereas here we did that in the peak of the disease and went ahead up to the chronic phase. In both situations, we saw that all the drugs did, they improved the outcome. However, the DITPA went below 1, which is basically very clinically relevant, so means a functional modification. Now here we did, again, untargeted metabolomics in mice now, not in humans, like the things that we did earlier. And as you can see, the same kind of pathways were activated here. We checked those biochemical pathways, but we also did functional analysis, such as the measurement of NFLs with Simoa, which basically measures a specific, it's a biomarker of axonal integrity. We did the locomotor recovery, how the animals were, the feet of the animals, and also visual evoked potentials, all of them in the mice. But we didn't stop there. We did MRIs again in the mice. We utilized diffusion tension imaging, and the metrics that we utilized were axial diffusivity and fractal anisotropy. They're both metrics that are taking a look into the integrity of the myelin. So those were upregulated, and in order to check that, that's the last one. In order to check that in the ultrastructural level, we utilized transmission electron microscopy, and we've measured the G ratio. So the G ratio is the diameter of an axon to the total diameter of the fiber, and this denotes a lower number, denotes thicker myelin. Okay, you can see that here, and this is the newly formed myelin. Taking all together, what we've shown is that DITPA, the analog that we promote, rescues the oligodendrocytes in those kind of situations. The pathway is this one that is seen here in this schematic through the PI3K, ACT, and PANC2 specifically signaling. We saw that there is oligodendrocyte preservation. We saw that axonal and also axonal preservation and also remyelination. So, all in all, this is a very potential candidate for treating multiple sclerosis. Right. So before I finish, I would like to acknowledge all the collaborators here in this work, a big one, as you can see. Steven Petratos is the lead PI of this work. I'm here with him in Melbourne, and this is where all the magic happens, in AHEPA and in Monash University. And with that, I would like to thank you all for your attention, and if you have any questions, I'm more than happy to answer. Thank you.
Thank you, Paschalis. This is a very nice work that you have done. Questions, please, from the audience. There is quite a few people working here in multiple sclerosis. Yes. Vasily.
So thank you very much for the presentation. I really liked it. I wanted to ask about the lipid metabolism. If you try to use lipids to see this pathway, let's say, what happens? I don't know. For example, in multiple sclerosis, maybe also diet plays a role sometimes, Mediterranean diet, and omega-3 fatty acids, or what is your opinion about this approach to target the lipid metabolism?
Thank you for your question. Well, if you mean administration of any kind of lipid, it's not in this work, but we do know in patients that yes, diet really plays a critical role actually to the progression of the disease. So no, here in this work, we just utilize those analogs because those are going, as I said, into clinical trials.
Yes, Achille.
I'm speaking very loud, so I will...
Okay.
Okay.
Okay. Thank you very much. Very good work, actually, very interesting. Did you ever try to see the effect of your compound in animal models with demyelination, like the Cuprizon, model?
Yes, yes.
So?
It's also in this big work, but unfortunately I didn't have the time to show that because EIE is a very good example of how demyelination happening in the CNS. But as you suggest here, the Cuprizon model also was involved in this process, and we did see improvement with the use of our treatment. Yes.
And a suggestion. Another neurodegenerative disease with demyelination processes is Alzheimer's.
Yes.
We start to realize that the last few years. So it would be interesting to use Alzheimer's rodent model to find out if you can reproduce what you've seen in these models, actually.
We're open to suggestions. If you do have a model, yes, we can try that. Absolutely. Thank you very much.
Last question. No, one before.
Congratulations on your talk. Do you have any idea what this compound has on the axonal damage? Because EAE is a model that is associated also with axonal damage, maybe from your electron microscopy studies.
Yes, it's very direct. It does axonal preservation. We've seen that. We've tested that head-to-head with we've benchmarked actually this drug with other compounds that they present to be axonal preservers. And we also seen that with the control tissue. So yes, we do know that there is axonal preservation with the molecule.
Okay, thank you.
Fotini, you have...
Okay.
But we need a microphone, I think.
Yes. Actually, I will follow up on Dr. Gravani's question. Given that this pathway is quite ubiquitous, are what you see, your data, the phenomena, the results, are they specific? First question. And if so, how is it specific? How do you explain the specificity, given that it is ubiquitous, the pathway is very ubiquitous? And also it's part of the same question. In EAE, there is demyelination and destruction of the axons and all that, but this is mediated by a very strong inflammation. And I really like that in your title, you put inflammation as the target of your drug, that it's against inflammation. So can you relate inflammation, again, a very ubiquitous phenomenon, to the specificity of your results? Thank you.
Yes, thank you very much for your questions. They are actually very good questions for a reviewer, so we're ready for that. Let's take it step by step, shall we? The first one, you said about that this pathway is ubiquitous. Yes, I agree. But the key molecule here is the MCT8. We do have MCT1, we have MCT2, we have MCT10. We have a lot of actual MCTs in all our cells of our body. But specifically for the neuronal, the nervous system, it's all about MCT8 and 10. MCT8 and 10. Why did we picked MCT8? Because it's most clear, because we do know the syndrome. We have the syndrome, the human syndrome, that produces hypomyelination. So that's 1-to-1, right? We've also, as I said, did the blood work, the liver function. We tested that. Stool analogy, we went crazy ahead because this one will be, as I said, translated to humans, right? So it's very important to know exactly. Second question was about the immune part. Yeah, the immune part. Well, of course, there is microglia there, and I'm expecting a question about microglia, what it does. Yes, we've checked that, but the mode of our drug is not targeting microglia. Our drugs that we have in our hands, they do act on B cells, right? They do act on T cells, the disease mode, the DMTs that we have today. There's only one drug actually that targets microglia, which are the BTKIs. That's a very new one. What I'm talking about here is one that we are trying to develop that targets oligodendrocytes, right? So that's why we've picked up this specifically one. Yes.
Thank you very much. I'm sure there would be more questions. I would have more, but we have to carry on and discuss with you later on. Thank you very much.
My pleasure.
Okay, let's move on with our next speaker, Irini Maria Georganda. She works with Dr. Skoulakis at the Hellenic Institute in Bari, and the title of her talk is Cross-Species Analysis of Behavioral Deficits in Neurofibromatosis Type 1.
So, hello everyone. Thank you very much for the introduction, and I would like to thank the organizing committee and congratulate for this very nice scientific event. So, in this presentation, in fact, I decided to give an overview of the neurofibromatosis type 1 project running in the lab, rather than one story. However, you'll see that they converge, our data converges in one conclusion. And this regards cognitive and behavioral deficits associated with this disease, neurofibromatosis type 1. So to begin with, a small introduction. Sorry for the repetition for those who heard Calliope before. So NF1, neurofibromatosis type 1, or NF1, is a multi-system, multi-symptomatic neurodevelopmental disorder that is inherited in an autosomal dominant manner. It results from mutations, either genomic or de novo mutations in the neurofibromin gene, and it affects approximately 1 in 2,000 to 3,000 individuals. It is characterized by a highly variable clinical presentation, and also it is the most common monogenic disorder that affects behavior and cognitive function. So, as I already said, the main hallmark of the disease is the great variability in the manifestations, as you can see in the figure. So, people with NF1 display some skin pigmentation and other cutaneous problems, optic pathway gliomas, skeletal abnormalities, but we as lab focus on the neurocognitive impairments, because NF1 patients very often face challenges in learning and memory, in attention, in sleep, as you heard from Calliope, autism spectrum abnormalities, social behavioral deficits, and so on. So based on, neurofibromin, as you can see here, is a very large molecule. The NF1 gene is a tumor suppressor gene. The encoded protein is a multifunctional, mainly cytoplasmic protein that is involved in several cellular signaling pathways, with the most well-studied being the RAS-MAPK pathway. So neurofibromin is a negative regulator of this pathway through its GAP-related domain, but it also affects several other pathways. NF1 is expressed everywhere, but is very abundant in the cells of central and peripheral nervous system. And NF1 gene has one of the highest mutation rates, probably due to its very large size. And this probably results to the variable clinical outcomes that we see. So till now, we know thousands of mutations that are distributed over the different apparent domains of the protein. And based on all this, our hypothesis is that these different mutations in NF1 gene contribute significantly to the variability that we observed in NF1-associated symptoms. So to test our hypothesis, we used two models, Drosophila and mouse. The most widely used models for NF1 till now are either the null Drosophila mutant or the knockout mouse, either conditional or total knockout. So both these models recapitulate key aspects of human symptomatology, meaning cognitive deficits, reduced growth, metabolic alterations, neuronal activity, social behavior deficits, and so on. Now, our aim here is to generate novel mutants, especially point mutants, which are most common in NF1, in both organisms, to behaviorally characterize these mutants, to investigate the implicated molecular mechanisms and neuronal circuits, aiming towards more precise treatment of the affected phenotypes through genetic and pharmacological approaches. So far, we have generated and we use 4 different mutants in Drosophila, a null mutant and 3 point mutants. So these mutations, as you can see, reside in different domains of the protein outside the prominent GAP-related domain. They are all viable and fertile, and thus you will see they share both common but also unique characteristics. So if we look here at this graph at the phosphorylated MAPK levels of the mutants, you can appreciate that only the null and this M1114 mutation, which resides closer to the GAP-related domain, so statistically significant increase in the phosphorylated MAPK levels, showing hyperactivation of the RAS pathway. Now, if we look at the lifespan analysis here that Kayopi did, we see in all mutants tested decreased survival rates, but to a different extent. So different mutations affect differentially the cellular signaling and also overall organismal health. We also tested the effect of these mutations on sleep and learning, 2 behavioral processes that are broadly affected in NF1 patients. Here in the sleep graphs, down here as you can see, Kayopi has very nicely shown that all mutants show decreased total sleep time, but to a different extent. And if we look at their patterns in the sleep chronogram, you also can see differential patterns between the different mutants. So there are some mutants that are completely arrhythmic, and other mutants like the arginine one, which retains periodicity, but with a small shift in the transition phase from light to dark, as you can see here, which mean a difference in the initiation of sleep, a delay there. Now, if we have a look at the learning deficits of these mutants, which were assessed using the olfactory associative learning paradigm, we see decreased learning performance in all mutants tested. And here we have identified a very strong learning mutant, this specific mutation, this cysteine 2 tyrosine in this region, which shows the most robust effect. So we can see that different mutants, in fact, show a different effect in the extent of behavioral phenotypes. And also we see that there is a heterogeneity in these point mutants, which are not just partial knockouts. One of our main questions is to identify the molecular mechanisms and the neuronal circuits that are affected by this mutation. And here I show you an example that shows that the null mutant and the E4 point mutant, the strong learning mutant, do not affect the same molecular mechanisms for associative learning. So here we have used ALK inhibitors. ALK stands for anaplastic lymphoma kinase. It's a receptor tyrosine kinase that broadly colocalizes with NF1 in Drosophila brain. it functionally interacts with NF1 both in Drosophila and mammals, and it is a negative regulator of learning and memory. So if we abrogate genetically ALK, or if we pharmacologically inhibit ALK, we can rescue the learning deficits of the null mutant, but not the ones of the E4 point mutant. So even if we have similar behavioral outcomes in the two mutants, the underlying pathways can differ, and this is very important for designing targeted interventions. And in order to obtain some mechanistic insights into the molecular consequences of these mutations, we have performed whole brain proteome analysis in these mutants. Here you can see in this heat map that illustrates distinct patterns of differentially expressed proteins of the mutants compared to the control, which is translated to differentially regulated pathways, as we have seen. So based on these results, we have already validated, and we still validate some of these processes that we found in order to confirm their role in the observed phenotypes. And in the last couple of slides, I would like to focus a little bit more on this specific mutation, this arginine 1809 to cysteine. It resides in this region here, the plexin homology domain, which, as you see, it's highly conserved among species. We have chosen this mutation because it is one of the very few known genotype-phenotype correlations in humans, and it is associated with behavioral deficits. So patients with this mutation display mostly learning disabilities, a shorter stature, but no neurofibromas or other type of cancers. So we have already generated, through CRISPR mutagenesis, the mutant fly, and we have obtained the same mouse model from Professor Gutmann in Washington University. So now we have started behavioral phenotype of both this model, and I'm going to show you some of our preliminary data. So here I show you the effect of this mutation on organismal size, which, as I told you, is affected in humans with this mutation. In the case of Drosophila, we see reduced size of the mutants, both in the pupal but also in the adult state compared to the controls. And in the same line, in mice, we see reduced body mass of the mutant mice compared to control litter mates. So both mutant flies and mice recapitulate the anthropometric features of identified in these NF1 patients. And lastly, I would also like to show you the effect of this mutation on short-term memory, which is impaired in these patients, largely impaired. In flies, we have assessed associative learning through the olfactory associative learning paradigm, where we teach the flies to selectively avoid a shock-associated odor. And here you can see impaired learning performance of the mutant flies. In mice, we have evaluated their recognition memory through the novel object recognition test, and they also show decreased learning performance. So this mutation consistently affects cognitive function across different species. And what I would like to leave as take-home messages from this presentation is that indeed our hypothesis is correct. Different NF1 mutations contribute significantly to the variability that we observed in the phenotypes. So this shows that it's very important to identify novel genotype-phenotype correlations for genotype-driven specific therapies. And also we see conserved phenotypes across Drosophila and mouse model, which recapitulate human symptoms. And this, of course, strengthens the validity of our results, but also shows us that we can use Drosophila for rapid screening and mechanistic studies, and then transfer this knowledge to mouse, where it provides more complex behavioral readouts. So with that, I would like to thank especially Mikey Skoulakis for our collaboration all these years. The Super NF1 team, especially Kaio Pachonio, which is a very talented and hardworking PhD student in the lab, all lab members, Professor Gutmann and Corina Anastasaki for providing us the mouse model, our facilities in Fleming, funding agencies, and of course all of you for your attention.
Thank you, Irini. We have a couple of minutes for questions from the audience. There, Mr. Efthymiopoulos.
Irini, congratulations for your work. I would like to ask you whether the interactome of NF1 is known,
and whether proteins that interact at different positions, domains on this protein, can explain also the variability.
My hypothesis it does. This is the 1 million question. I mean, yeah, it's very important. We also want to answer this question. We don't know the interactome. We know from in vitro studies several proteins that have been shown to interact with NF1. Not all of them have been validated across studies, and very few of them have been validated in vivo. So from our data, we have performed, previous members of the lab through collaboration have performed an interactome approach to show the proteins the proteins that interact specifically in the domain where the E4 mutation is, because this is a largely uncharacterized domain of the protein. And there we've seen some results that really are in accord with our proteomic approaches. So we see there, as a spoiler, mitochondrial proteins. So I guess that, yes, there are different proteins that interact with different domains, and this is one of our goals, to make this structure-function analysis, which is missing, really, from the NF1 research.
Okay, thank you. Other questions? Okay, if not, Rebecca.
Can I ask something?
Yes. Another one there.
Is it okay if I speak through this microphone? You made the null mutant. Is there in humans there is no null mutant, right? Or
No, there
are null
yes,
mutants.
Yes.
Are there also
They're
half?
Yes, they're heterozygotes.
They're heterozygous.
Yes.
But
Yes.
you didn't do anything with the heterozygous, for example.
Yes, we have. We have in the case of the null, but there is no effect in these mutants. Although in mammals we need haploinsufficiency of the gene, in flies we need a loss of heterozygosity to see the effects. Yes.
So in mammals you do see, but not in the flies. You see in the heterozygous.
Yes, because they are lethal. They are homozygous lethal in mammals. All mutations, not only the null, the knockout, even the point mutants are lethal in mice, in mammals.
In mammals.
Yes.
Okay, thank you.
Okay, we have one last question.
Hi, great presentation. You mentioned that the NF1 mutants have a smaller body size, and they sleep less.
Yes, they do sleep.
I'm wondering whether, I know that there's a growth hormone that's secreted when animals sleep. So maybe the animals that sleep less, they have less growth hormone and there's smaller body size.
Yes, this is a very good point. This could be a very good correlation between the two phenotypes, but I don't have data to support this.
Okay, nice.
Yeah,
Thank you.
Okay, thank you very much, Irini.
The next speaker is Gerasimos Nakas from the Hellenic Pasteur Institute, and in fact from the group of Vasiliki Kirargiri, and they're working on multiple sclerosis as well. And also they have developed models of experimental autoimmune encephalomyelitis, and in this case the title of the talk is Cerebellum-Specific Microglial Activation Correlates with Altered Synaptic Integrity in Early Experimental Autoimmune Encephalomyelitis, EAE.
Thank you very much. Hello, everyone. I will discuss about how early cerebellar microglial activation correlates with pathology in experimental autoimmune encephalomyelitis. Multiple sclerosis is the prototypical immune disease of the CNS, affecting millions of people, and is more prominent in women. Hallmarks of multiple sclerosis include demyelination, axonal loss, neurodegeneration, and excessive synaptic pruning. There are 4 main types, with relapsing remitting MS to be the most common form. Current therapies of MS mainly target the immune-related component and symptoms that are immune-related. However, over half of patients show cognitive disabilities even before the clinical symptom. And evidence suggests that cerebellar lesions are linked to cognitive syndrome and have a poor prognosis in MS patients. in contrast to white matter pathology, in which MRI scan give us the opportunity to detect the pathology, in gray matter pathology, which involves synaptic damage and degeneration, conventional methods did not let us to detect the pathology. However, advances in live brain imaging have revealed that gray matter pathology is present from the early stages of the disease and is correlated to cognitive disability. And microglia are of great interest in efforts to better understand the gray matter pathology, given their roles in at first stages of the disease. Microglia seems to promote demyelination, secrete toxic factors, and pro-inflammatory cytokines. And in contrast, at later stages, microglia seems to have a beneficial role promoting remyelination and by helping oligodendrocyte migration or secreting anti-inflammatory cytokines. Experimental autoimmune encephalomyelitis is a T-cell mediated model of CNS which recapitulates many aspects of MS. On day 0, mice are immunized with MOG peptide, and PTS and pertussis is toxic to enhance further the innate immunity response. A second dose is given on day 2, and the onset of the disease occurs around day 10 based on clinical score. It's important to keep in mind that E research mainly target the spinal cord pathology. However, brain regions such as cerebellum still remain underexplored. So our aims were to study microglial dynamics in distinct brain regions during EE progression and how microglia activation may correlate with cognitive disabilities, especially with brain pathology. So first, to investigate progression alteration in microglia morphology, we performed in vivo 2 chronic imaging. First, we performed craniotomies and head plates in 6-3CR1 GFP mice to expose a part of cortex and cerebellum. After 3 weeks of recovery, we performed in these mice baseline recordings of microglia cells in cortex and cerebellum, and we mark the regions after retroorbital injection of the dyed dextrans Texas red, which let us to visualize the vasculature of brain. And after E induction, we performed recordings of microglia cells in these regions every 2 days until the disease of the onset. And the novel with this method that led us to monitor the same region in each animal during EE progression. So the results show that in cortex, microglia retain their typical ramified morphology during the EE progression. However, in cerebellum, microglia cells show morphological hallmarks of activation, some enlargement, and process drag from the pre-onset phase of the EE. Actually, the hypertrophy index saw a 50% increase at the preclinical stages of the EE. To further assess the tissue specificity of microglial activation, we perform immunohistochemistry in fixed tissues from cerebellum, cortex, and spinal cord, as spinal cord is the most highly affected region in this model. The data generated by Elias Rufagala show that in cortex, microglia remain unchanged during EE progression, while in cerebellum, microglia show morphological changes, findings that were consistent with results from 2-photon imaging. It's important to know that in cerebellum, during pre-onset phase, 2 components CFA and PTX were sufficient to induce morphological changes, despite the presence of MOG was necessary to further increase the index at the onset of EE. And with regard to spinal cord, as I said, it's the most highly affected region in this model. We didn't observe an alteration in microglia morphology until the disease onset. So the early microglia activation occurs specifically in cerebellum in experimental autoimmune encephalomyelitis. So the questions are, what causes this early activation and how does it affect brain pathology? To investigate potential triggers of early microglia activation, we stained decalcified tissues with pan-immune marker CD45 And our results show that in healthy group, CD45 positive cells were detected in the meninges, indicate the baseline surveillance. However, in the preclinical phase of EE, these immune cells were detected in cerebellum parenchyma, and their numbers still increased on the onset of EE. The results indicate an immune infiltration that may contribute to early microglia activation, and now we're working on still further characterization of these immune cells. And we next ask if there is effect of this microglia activation in the brain pathology, quantification of myelin levels of MBP markers, so no difference between group, and we can observe that EE pre-onset phase will have normal appearing gray matter myelin with no lesion. Lesions were detectable at onset of EE. However, when we stain tissues with a neuronal marker MAP2 and IBA1, our analysis show that we have increased interaction between microglia and dendrite elements that detectable already at the pre-onset phase of EE. Given the increased interaction between microglia and dendrite, we next investigate if there is changes in spinal density, and we focused on Purkinje layer of cerebellum to investigate the effect on Purkinje neurons. Purkinje neurons are inhibitory neurons of cerebellum, and using Goldie staining method, our results reveal that we have a spinal loss detectable already at the pre-onset phase of the EE, persisting through the onset. And finally, in order to evaluate if activate microglia phagocytosis synapses, we stain tissues with synaptic marker PSD95, and the results show increased PSD95 engulfment of microglia in the onset of EE. So taken together, our results indicate an early specific activate microglia in cerebellum, and this activation is linked to increased microglia dendrites contacts, spinal loss, and PSD95 engulfment. So our future plans is to investigate if this morphological alteration translates to functional outcome using electrophysiology or behavioral assessment, and the take-home message is a better understanding of gray matter pathology and the role of microglia in progression of disease may shed light on mechanisms on more targeted therapies for cognitive disability that present in MS patient. I would like to thank my supervisor Vasiliki Kirargiri and members of our team Nicoletta and Erikos. Many thanks to Hellenic Paris Institute for supporting my PhD through scholarship of Nostos Foundation. I also would like Dr. Leslie Probert and Dr. Konstantinos Kabas and all members of Laboratory of Molecular Genetics, as well as the member of Department of Animal and the members of Bioimaging Unit, and the greatest thanks to all of you for your attention.
Thank you very much, Gerasime, for your talk. I just have a quick question I'll start with. Can you follow this interaction of microglia and the neurons, Purkinje neurons, in live cell imaging? I mean, you should be able to, right?
Okay, that will be a good choice. We will have a transgenic mice for a marker for Purkinje cells. There is, I'm sure, a mouse model for these cells, and cross-mating with 6-horn GFP mice, so it's monitor the live interaction of these cells with photon imaging.
You plan to do that?
Yes.
Okay, that'll be very nice to see.
Thank you very much.
Any other questions from the audience? Yes, please.
Very nice work. So I guess those CD45 cells were only in the meninges, and then you saw them that they infiltrate the parenchyma, right?
Yes, in the meninges in all groups, healthy, pre-onset and onset, and in parenchyma only in pre-onset and onset phase. In healthy group, we didn't detect CD45s inside.
Okay. Did you check around the area, like in the fourth ventricle, which is close by, choroid plexus? Because I would suggest that those cells would be found, because those are probably the border-associated macrophages, so they will be all around the place, and they might do something else there. That's a suggestion, if you haven't done
Yes,
so.
we're supposed to be infiltrating new cells based on morphology, more round cells, and based on a T-cell-mediated model, maybe lymphocytes, and we have stained for markers. Of course, there are perivascular or choroid plexus macrophages, and there is hypothesis about secrete molecules of these cells inside the parenchyma, or may infiltrate these cells, but we are working on still characterization.
Sure. Thank you.
Any more questions from the audience? If not, thank you very much.
Thank you.
Okay, our next speaker is Irini Portakali. She's a student in the Athens International Master's Program in Neuroscience, and she's currently working on her thesis in Dimitra Thamoidou's lab at the Hellenic Paris Institute. The title of her talk is Alzheimer's Disease Risk Factor BIN1, Linking Microglial Deficiency to Neuroinflammation and Regulation of Adult Hippocampal Neurogenesis. Irini? Microphone.
Now? Yes. So today we're going to talk about Alzheimer's disease risk factor BIN1 and how its deletion links microglial deficiency to neuroinflammation and regulation of adult hippocampal neurogenesis. As you probably already know, Alzheimer's disease is a complex neurodegenerative disorder that is characterized by the presence of A-beta plaques and tau tangle, as well as chronic neuroinflammation that manifests through reactive microgliosis and astrogliosis. The disease can occur in a familial form through specific gene mutations or sporadically through risk factors that increase susceptibility for the disease, such as aging, environmental factors, and genetic variants. One of these variants, and actually the second strongest sporadic AD risk factor, is bridging integrator 1, BIN1. BIN1 is a protein that is involved in cytoskeletal interactions, vesicular trafficking, endocytosis, and it has been mainly studied in neuronal cells and linked to the disease through tau pathology, although lately it has been proposed to be a modulator of microglial inflammatory responses. Other associated features of the disease are the symptoms of memory impairment and cognitive deficits that have been functionally linked to hyperhippocampal neurogenesis, which is also present in AD brains. And interestingly, microglia are crucial regulators of adult hippocampal neurogenesis through the secretion of neurotrophic factors or through phagocytosis control of their population. However, when they secrete pro-inflammatory cytokines, they can have a detrimental effect on the neurogenic process. Now, to link everything together, our goal is to unravel how microglial BIN1 regulates neuroinflammation and non-cell autonomously adult hippocampal neurogenesis, ultimately driving AD progression. To induce that, to address this, we had an inducible double transgenic mouse model, and using the 6-3 CR1 gene and tamoxifen injection, we generated microglial-specific BIN1 knockout. And also we performed LPS injections as a model of neuroinflammation. Previous signal nuclei sequencing data from our team has revealed that cortical BIN1 knockout microglia present an increased interferon type 1 phenotype. Interferon type 1 is basically an immunological response pathway that has been found to be strongly activated in AD brains. Moreover, they found a cluster of microglia with increased markers of proliferation. If you want to learn more about this, you can ask Mary Margariti from our team. So our first job was to assess whether this was the case for the hippocampus as well, and we started with PCR screening of interferon type 1 molecules, and as you can see from the pink bars, these molecules were increased in the knockout animals under LPS conditions, and we went to look further the protein levels of Fe2O4 molecule that was significantly increased in the sequencing. And as you can see, it was increased with its fluorescence intensity, actually was increased with great significance in the knockout animals, indicating that BIN1 knockout increases interferon type 1 gene expression. Now, we needed to address whether this result was attributed to a different number of microglial cells. So we quantified the number of microglia in the dentate gyrus under LPS conditions, and we found that they didn't have any difference in their population, and we went further to look at their proliferation state, either with a BRDU incorporation method or KI staining. And in either cases, we found that BIN1 knockout does not influence microglial proliferation. Now we know microglial cells, cell responses are tightly linked to morphological changes. And for that reason, in collaboration with Evangelia Xigi, we did morphometric analysis of microglial cells. And as you can see from the figure here, we found that BIN1 knockout microglia under LPS conditions present a hyperramified morphology, which is considered an intermediate state of activation and a sign of resilience. We know neuroinflammation enhances microglial phagocytic capacity, and for that reason, we stayed for lysosomal marker CD68. And to our surprise, BIN1 knockout microglia had reduced phagocytic capacity, and in a closer look, they remain on the same levels as the non-LPS conditions. At the first glance, this might seem logical, given the fact that BIN1 is implicated in membrane curvature and endolysosomal trafficking. But given the fact that we have increased interferon type 1 response, it's a bit contradictory, and it's a point we want to further investigate. So we wanted to test more on the inflammatory component, and for that reason we did hippocampal proteome analysis, and to our surprise, most of the inflammatory-related proteins were decreased in the knockout animals. There was only a small cluster that were increased. Some of these, like CXCL10 and CXCL11, were members of the interferon type 1 pathway. And interestingly, interleukin-4 and TIP1 were increased in the knockout animals. And these are actually established markers of positive regulators of adult hippocampal neurogenesis, which was a good point to now jump to the non-cell autonomous effect of microglial BIN1 deletion. So we stained for all neuronal lineages of the hippocampus, and we were very excited to see that radial glial-like cells, as well as intermediate progenitors and committed neuroblasts, were all increased in the knockout animals, and they also presented increased proliferation rates. And of course, we are ongoing for the neuroblast as well, collectively indicating that BIN1 knockout increases adult hippocampal neurogenesis. Now to sum up, BIN1 knockout decreases most inflammatory-related proteins with few exceptions, mainly from the interferon type 1 pathway. It presents reduced phagocytic potential and results in population expansion and proliferation in green in the neurogenic proteins of the hippocampus. And to look more the mechanistic aspect of this result, we have generated an in vitro BIN1 knockout model where we use BV2 microglial cells and siRNA to knock down its expression. Some first screening on inflammatory-related proteins and phagocytic markers show a decrease in the knockdown, reproducing the in vivo results. And the most interesting part is that we are trying to take this condition medium and put it in primary astrocyte, and in collaboration with Dr. Kutmani from BRFFA, we're planning to use this on adult hippocampal neural stem cells. Now I want to give acknowledgements to everyone from the group, our supervisors, the lab members, and our collaborators, and of course every one of you for your attention.
Thank you very much, Irini, for the nice presentation. Questions? Yes, please.
Hello. I was wondering if you know the molecular mechanism on why the BIN1 knockout is affecting the proliferation of the cells and the inflammatory cytokines are reduced, and why there is like this exception between the cytokines are like reduced and the other one that are like still maintained.
So first, for the proliferation, in the hippocampus, it's not actually affected. That was in the cortex. And BIN1 knockout has a domain that interacts with CMYC, which is a marker of the cell cycle. And so it's actually, it blocks. It's, so when you have a loss of BIN1, then the proliferation is more free to happen. But this was not the case for the hippocampus. It was only visible in the cortex. And the other part, what was it?
And about the cytokines that you see, like that mostly are like reduced, but then there is like few that are still are regulated. If you know always the mechanism why you
We don't know the mechanism, and actually it's BIN1 in like microglial cells, and it's now starting to be more researched, and we're trying to find the link as well.
And one last question, sorry. And after I saw that you want to do in vitro model to study more, do you think that changing the model will like impacting your results, or like are you sure that, I mean, I don't know if you use like stem cell from like mice stem cell or like human stem cell, and
I didn't get the question. If it will change the result?
yeah, because I mean after you want to do some in vitro model and
Yes.
experiments, so I was wondering before you're using another model, so if this one can impact your results and perhaps you will see the same things or not.
Well, we're hoping to see the same things, obviously. If I would suggest, the best would be to use primary hippocampal microglial cells and not a cell line that is usually a lot different from primary cells. And this is a startup. I think we will move to primary cells as well.
Okay, well, I'm happy to hear more about it. Thanks.
Nice talk. I have a question here. Sorry. I have a question about like the if you are planning to do some behavioral study, in particular for Alzheimer's disease, is we know that there is impairment in memory. So since like we are talking about hippocampus, and hippocampus is renowned like the neurogenesis is involved with memory, and your protein, when you knock out your protein, like there is an increase in the neurogenesis. Like I was wondering if you are planning to do some type of.
It's not planned to happen soon, but it's something that we want to do, because at the end of the day, this is what we care for, behavior, and like if it changes functionally something. Yeah, we would like to do that as well.
More questions? Professor Efthymiopoulos.
Is there a differential expression of BIN1 between cortex and hippocampus? And another question is, do you see any translational directions coming out of your work?
I don't know if there is a BIN1 difference from cortex to hippocampus, actually. No, I don't know this. Translational work, not the translational direction. Haven't think about something, and actually I don't know how you could mess up with BIN1 that is implicated in so many different processes, in all brain cell types as well.
Okay, for the sake of time, if there are any more questions, they can be discussed with Irene during the party. So we move on. Thank you, Irene.
The next talk is going to be given by Nikos Kokorakis, who is a postdoctoral fellow at the Hellenic Pasteur Institute in the group of Maria Gaetanu. And his talk is entitled Pharmacological Inhibition of Mirk-DYRK1B Kinase Rescues the Early Axonopathy of the Embryonic SOD1 G93A Spinal Motor Neurons. So it's a model of ALS, I guess.
Of course. Thank you for your introduction. Good evening, everyone. I'm going to talk about you the identification of the functional and pathological role of DYRK1B in development and especially in disease in ALS. DYRK1B is a major member of DYRK kinase family that regulate many aspects of cellular processes, and its main role is that of inducer of cell exit of early progenitors and inducer of differentiation. But despite the fact that paralogous DYRK1A function is well studied in CNS, in brain and spinal cord, the role of DYRK1B remained elusive. For example, what about DYRK1B expression and function during CNS development? To fill this gap in knowledge of DYRK1B function, from our previous studies, we tried to elucidate the function in spinal cord development by applying in novo gain and loss of function studies in the chick embryo, in the developing chick embryonic spinal cord. In novo, in egg, gain of function studies with overexpression of DYRK1B by unilateral electroporation, and the loss of function by applying a small molecule that acts as inhibitor of the DYRK1B kinase activity, a selective inhibitor. The first finding was that DYRK1B affects the ventral patterning of the embryonic chick spinal cord, especially ventrally at the motor neuron domain by promoting the apoptosis and reducing the motor neuron populations, motor neuron progenitor populations, and therefore motor neuron population ventrally, as well as a type of ventral interneurons, V2A interneurons. In contrast, the pharmacological inhibition by this specific molecule AZ191 led to the opposite phenotype with the increase of cycling progenitor population in ventricular zone in the developing spinal cord, and especially in the increase of motor neuron populations, as well progenitor populations that give rise to V2 and V3 in the neurons, that is P2 progenitor population and P3 population, and of course the rise of PMN progenitor of motor neuron population. this specific and severe ventral phenotype make us to think about the possibility of the implication of DUC1B with the Sony-Hedgehog signal pathway that governs and architects the ventral patterning of developing spinal cord. And indeed, we have shown and observed that the overexpression of DUC1B leads to the decrease of the expression of Sony-Hedgehog and the decrease of the expression of its effectors, GLI3 and GLI2. And in contrast, the pharmacological inhibition of DUC1B led in the opposite phenotype with increase of Sony-Hedgehog expression, a similar phenotype with what caused by Sony-Hedgehog activation by a small molecule that is an agonist of smoothening, a component of Sony-Hedgehog called SAG molecule. To study further the DUC1B function at a later developmental stage at the cheek spinal cord, we have observed that DUC1B affects motor columns, especially the LMC medial motor column, I call it subcolumn, that innervates the ventral side of muscles in the limbs of cheek embryo. This is from brachial level. And this specificity only in this LMC medial subcolumn is reflected to the surprising fact that the only motor subcolumn that is characterized by the expression of Sony-Hedgehog is the LMC medial motor column, except for, of course, the expression of Sony-Hedgehog by the main source of it, that is the floor plate of the spinal cord. So, to conclude, DUC1B is a potent transcription suppressor of Sony-Hedgehog, directly or indirectly. We don't know the exact transcriptional regulation, but we know that DUC1B controls the generation and survival of spinal motor neurons, as well as their organization at the LMC medial motor column. Collectively, DUC1B is a potential pharmacological target in motor neuron disease. So, our aims of the study is to investigate the Sony-Hedgehog signaling and neurodevelopmental deficits in embryonic spinal cord of SOD1 mice that harbor the mutation of G93-A and resembles the symptoms of ALS, that is a fatal neurodegenerative disease characterized by motor neuron degeneration and loss, both by in vitro and in vivo approaches. So, to answer these questions, investigating Sony-Hedgehog signal and neurodevelopmental deficits, we have observed that embryonic spinal cord of SOD1 mice exhibits neurodevelopmental deficits characterized by reduced Sony-Hedgehog at the floor plate, that is the source of Sony-Hedgehog, and increased DUC1B expression. That is an important clue. Of course, we have observed that SOD1 mice exhibits reduced numbers of P3, P2, PMN progenitor populations indicated by immunohistochemistry. And after these observations, we have developed an optimized 5-day spinal motor neuron primary culture protocol, resulting in high yield of motor neurons. And this is the main system of our experiments. we use the pharmacological inhibition of DIR1B that rescues the early axonopathy of SOD1 spinal motor neurons that is characterized by short fragmented axons. And you can see here, upon administration of AZ191, the recovery back to normal levels to wild-type phenotype. Furthermore, pharmacological inhibition of DIR1B reduces apoptosis in SOD1 spinal motor neurons. At this point, remember that DIR1B kinase promotes apoptosis ventrally at the motor neuron domain in chick embryos from our previous study. Furthermore, pharmacological inhibition of DIR1B in both wild type and SOD1 spinal motor neurons indicated by increased markers of autophagy like LC3AB and decreased the marker P62 expression as indicated by the figure and Western blots. In addition, pharmacological inhibition of DIR1B improves the imbalanced proteostasis in SOD spinal motor neurons as indicated by the decrease of ubiquitinated proteins upon administration of AZ191 in cultures and reverse of phenotype back to normal levels. The most important finding is that pharmacological inhibition of DIR1B reduces endogenous SOD1 protein expression in spinal motor neurons back to normal levels upon the administration of the AZ inhibitor. And it is the most important finding because the aggregation and accumulation of SOD1 is a hallmark of the motor neuron loss and severity of this disease. Last, pharmacological inhibition of DIR1B improves mitochondrial dysfunction of SOD1 spinal motor neurons by restoring the mitochondrial membrane potential that is indicative of the mitochondrial proper function back to normal levels, as you can see from this experiment. To conclude, DIR1B expression is upregulated in the embryonic spinal cord of SOD1 mice, while Sony Hedgehog signaling is impaired and is downregulated. Spinal cord of SOD1 mice exhibits reduced numbers of ventral progenitors, P3, PMN, P2, as well as postmitotic motor neurons. The important finding is that these symptoms, these findings are present already from embryonic developmental stages, and it's important to think that developmental stages include molecular alterations that that are observed lately at severe developmental stage postnatally. Collectively, DIR1B consists a potent pharmacological target in SOD1 mediated ALS and remains to be evaluated its pharmacological inhibition in vivo. At this point, thank you all of you for your attention. I'd like to thank my supervisor, Maria Gaetano, Rebecca Matsas, our collaborators, and of course, our students. Thank you.
Thank you, Nico, for this talk. Questions from the audience? So again, we have a sort of widespread sort of pathway that affects specifically a very small sort of group of neurons in this devastating disease. How do you think that this can be a pharmacological target? I mean, can it be given preclinically, for example, before the initiation of the disease or after?
Yes, this is a matter of experimentation. We have planned to perform the in vivo experiments by administration of AZ191 molecule in mice, and we want to see if we have any halting of symptoms. We plan to inject the inhibitor at the postnatal day 19. That is the first stage, stage of that the phenotype is severe in mice, and we wanted to observe the mice until the day, postnatal day 140. That is the terminal day of their life. The symptoms are very severe, and we have to check all these markers that we have checked in vitro cultures in this in vivo model. I believe that we are far from to use this molecule in clinical trials, but we have observed that is very beneficial until now in our very specific model we have.
Thank you.
Thank you, Nikos.
Thank you.
And we quickly move on with our last speaker, Stathis Athanasiou, who is actually a final year biology student at the University of Athens, and he comes from Professor Palkaras' laboratory at the Medical School of Athens. The title of his talk is MCU and MPTP mediate cytosolic calcium overload and neuronal dysfunction in C. elegans models of autosomal dominant optical atrophy.
So thank you for the introduction. As you heard, I am Stathis Athanasiou. I am working along with Fivos Borboulis and Professor Konstantinos Palkaras at the Medical School of Athens. And I'm going to show you how we are investigating blindness in an organism that does not have eyes. So let me explain. We are studying ADOA, or autosomal dominant optic atrophy. ADOA is a neurodegenerative disease causing the degeneration of the optic nerve specifically. This causes, of course, vision loss that is progressive and begins in childhood and develops over the following decades. ADOA is a rare disease affecting about 200,000 patients all around the world, and up to now there is no cure or any disease-modifying treatment. So the fundamental cause of ADOA are mutations in the OPA1 gene. They are point mutations and they have a dominant negative effect. OPA1 normally regulates inner mitochondrial membrane dynamics, and it does so by orchestrating mitochondrial fusion and the structure of the mitochondrial cristae. So, what about the molecular mechanism of ADOA? Until recently, not many things were known. It is a rare disease, not being, it hasn't been studied a lot, but in the past 5 years, two very important publications came up. it was found that ADOA is the result of excessive mitophagy, which is triggered by elevated cytosolic calcium levels. So in more detail, in ADOA, there is an accumulation of calcium ions inside the cytosol. This causes the activation of calcineurin and AMPK, and those two signaling molecules activate excessive mitophagy. This, of course, causes the degradation of mitochondria and leads to a lower number of mitochondria in the axons of the neurons. Now, most neurons are actually okay with this, and they can survive a lower mitochondrial number. However, the neurons of the optic nerve have extremely high energy demands by far. So when the mitochondrial numbers drop, these neurons die. Now, all this chain of events is initially triggered by the OPA1 mutation. However, up to now, it remains unknown how the OPA1 mutation leads to elevated cytosolic calcium levels. So, elevated cytosolic calcium is actually driving the ADOA pathogenesis, but we still don't know what causes it. And this is exactly the question we're trying to answer. What causes elevated cytosolic calcium in ADOA? Where is the extra calcium coming from, and what molecules are implicated? In order to answer these questions, we're using C. elegans. C. elegans is a microscopic nematode worm reaching only 1 millimeter in length. It lives in the soil and feeds on bacteria. But why are we using worms? You see, C. elegans has some very convenient properties. It has a very short life cycle, only 3.5 days. It can self-fertilize as a hermaphrodite. It is very easy to maintain in the lab. There are no bioethical restrictions with it, and up to now, it's the only organism to have its entire connectome fully mapped. Also, C. elegans is completely transparent, giving some excellent opportunities for fluorescent microscopy in vivo. So, because of those characteristics, C. elegans experiments with C. elegans are very fast, easy, and cheap, and this is why we like to use it. So, in order to model the ADOA disease in C. elegans, we're using the EAT3 gene. EAT3 is the nematode ortholog of the human OPA1 gene, so we're using a strain with a mutation in the EAT3 gene to replicate the phenotypes of mutant OPA1, which is causing ADOA. To measure cytosolic calcium levels, we're using GCAMP, a fluorescent calcium sensor expressed in muscle cells. And what we observe is that the EAT3 mutation indeed causes elevated cytosolic calcium levels, successfully replicating this specific phenotype of ADOA in neurons. And you can see visibly very higher intensity in the fluorescence because of the extra calcium. So now that we do observe elevated calcium levels, we can study what causes that. To do this, we use the specific gene knockdown by RNA interference. One of the genes we targeted was the MCU. MCU is a calcium transporter in the inner mitochondrial membrane, and it's responsible for importing calcium into the mitochondria. So, interestingly, we found that the MCU is essential for the cytosolic calcium elevation. In EAT3 mutants who have elevated cytosolic calcium, suppression of the MCU causes a drop in calcium levels, and the calcium levels return back to normal. This is also visible from the images because the clear difference between wild-type animals and EAT3 mutants completely disappears when we suppress the MCU, and now EAT3 animals look like normal worms. So, it appears that the import of calcium into mitochondria is responsible for the elevation of calcium in the cytosol. At first, this might seem a bit counterintuitive, but what it probably means is that calcium ions inside the mitochondria get released out into the cytosol. This is why we decided to focus on calcium export from mitochondria, which happens predominantly through the NCLX, LETMI1, and the MPTP complex. So starting from the NCLX and LETMI1, we found that those two calcium transporters are not implicated in the mechanism of cytosolic calcium elevation, since when we suppressed both of those genes, calcium levels remained high. Now let's take a look at the MPTP complex. The MPTP forms a non-selective pore in the inner mitochondrial membrane, and it's responsible, in the inner mitochondrial membrane, and it forms under stress conditions. It's considered a pathological pore. However, some physiological roles have been argued as well recently. However, when activated, the MPTP complex makes mitochondria permeable to all small molecules. So when it's activated, it's also releasing calcium ions out into the cytosol. The MPTP is believed to be comprised of three basic subunits, the A and T, cyclophilin D, and the ATP synthase. We tried the A and T and cyclophilin D, and we found that both of those two subunits are essential for the cytosolic calcium elevation. In E3 mutants with elevated cytosolic calcium levels, suppression of the A and T makes calcium levels return back to normal, and the very significant difference of the cytosolic calcium elevation completely disappears. Something similar happens with cyclophilin D as well. When we suppress cyclophilin D, calcium levels decrease, and now wild-type animals and E3 mutants have no difference. So both of those two subunits are necessary for the elevation of calcium. Moving on to another subunit of the MPTP, the ATP synthase. Surprisingly here, we don't see the same as the previous two subunits, and it appears that the ATP synthase is not necessary for the cytosolic calcium elevation. So what is causing cytosolic calcium elevation? It appears to be mediated by the MCU, which imports calcium into the mitochondria, and by specific subunits of the MPTP complex, which releases calcium from mitochondria. Also, these data seem to imply the existence of an MPTP complex that is independent of the ATP synthase. So, based on our results, we can start to outline the first proposed mechanistic model for cytosolic calcium elevation in ADOA, or OPA1 related dysfunction in general. So, we propose that during OPA1 dysfunction, large amounts of calcium from the ER get imported into the mitochondria through the MCU. From there, mitochondrial calcium gets released out into the cytosol because of MPTP activation. This, of course, causes cytosolic calcium elevation, which, as we mentioned before, triggers calcineurin and AMPK, activates excessive mitophagy, and leads to cell death. So the idea is that calcium from the ER passes right through the mitochondria and ends up accumulating in the cytosol. In order to test this mechanism, we decided to use RU360, an inhibitor of the MCU, and cyclosporin A, an inhibitor of the MPTP complex. In our next experiments, we're using a humanized model of ADOA, humanized because it's expressing the human mutant OPA1 gene that's actually found in ADOA patients. And in our model, it's expressed specifically in the GABAergic neurons. The phenotype we studied was the defecation cycle. Defecation in C. elegans is a strictly regulated periodic phenomenon happening about every 50 seconds in young, healthy adults. And now you would be absolutely right to ask, why am I telling you about the defecation of worms? This is because it's regulated by two specific GABAergic neurons. So any dysfunction or even death of those neurons is going to be reflected as an impairment in the defecation cycle, something we can easily observe. So the defecation cycle is practically a marker for neuronal dysfunction. And indeed, we observe that expression of mutant OPA1 causes an impairment in the defecation cycle. The defecation cycle lasts longer and it's much more variable, indicating neuronal dysfunction. Now, RU360, an inhibitor of the MCU, can prevent some of the neuron dysfunction caused by mutant OPA1, showing that without the function of the MCU, mutant OPA1 cannot further impair the defecation cycle and the function of the neurons. Cyclosporin A, an inhibitor of the MPTP complex, can fully restore the function of neurons expressing mutant OPA1. We see that OPA1 expression causes an impaired defecation cycle, and cyclosporin A can make the cycle return back to normal, effectively rescuing the function of neurons expressing mutant OPA1 and showing that the MPTP complex is responsible for this. So we saw that the MCU and the MPTP complex mediate the cytosolic calcium elevation and the neuronal dysfunction. And inhibition of those two molecules by RU360 and cyclosporin A can prevent those phenotypes of ADOA. Now, of course, the mechanism we're presenting here comes from experiments only in C. elegans. So, can those findings also be translated into human patients? Of course, we don't know. We need to test this on more complex models. An idea could be to administer cyclosporin A in mice who have an OPA1 mutation and then assess those mice for phenotypes of ADOA, like vision loss. A validation like this one, which I just said, could maybe establish this proposed mechanism as the cause behind ADOA and highlight the MCU and the MPTP complex as potential therapeutic targets for ADOA, a disease that up to now has no cure. I would like to thank my supervisors, Professor Konstantinos Palikaras and Fivos Borbolis for conceptualizing this project, and the rest of the members of the lab, also the organizing committee for having me here, and of course all of you for your attention.
Thank you, Stathis. Maybe one or two short questions. Yes, please.
Thank you for your talk. It's really interesting. I have two tiny questions. The first one is, have you measured, because of this mechanism, have you measured the endoplasmic reticulum mitochondria contact sites? Because if you are assuming that calcium is going from the reticle to the mitochondria, then there will be interesting to look. And the other one, have you any idea of how the mutation in OPA1 could be impacting in MCU or MPTP?
Okay, both great questions, thank you. The endoplasmic reticulum mitochondria contact sites is not something we have tested, but it has been documented that in OPA1 mutations, these contacts are closer. So this is where the exchange of calcium is happening. So this supports this model. And also, this is something we have thought of testing also in C. elegans or other models. Now, what you asked about OPA1 function, dysfunction affecting those molecules, this is not something we have studied, and this is probably the next question to complete the whole chain of events. An idea could be that I have read somewhere, but there's no experimental evidence for it, that maybe because OPA1 is responsible for the structure of the cristae, maybe MCU is normally trapped inside this cristae, but when OPA1 is dysfunctioning, those cristae disappear, and now more MCU is available in the membrane and can import more calcium. So this causes some accumulation, and it's released from the MPTP, which is known to be activated when we have an overload of mitochondrial calcium.
Thank you.
Really fascinating, because I was having the same question. Just a little curiosity. In your model, are you able to see any morphological changement with the OPA1 dysfunction in your mitochondria in C. elegans that perhaps you can see also in human patients?
It has been documented that the EAT3 mutation, which is the ortholog, shows pretty much the same features in the cellular level. Mitochondria are much more fragmented because OPA1 mediates mitochondrial fusion, and there's also a lower ATP production because of a more general mitochondrial dysfunction, and also increased ROS production, something that could also indicate maybe the opening of the MPTP complex.
Okay, thanks.
Okay, thank you very much, Stathis. And with this, we conclude the session. Congratulations to all speakers for their presentations, and good luck with your projects. Thank you all for joining us. And we move with the plenary. Thank you.
Okay, so we come to the point that it's the last but not least. Actually, I should say the last and the best. A few months ago, Elias and Athena approached me and they asked me for a suggestion for a keynote speaker, just for the one to close the event. And the keynote speaker, since this is about young investigators, he or she had to accommodate some important features so that they can inspire the audience. Also, it's quite important to combine two important features. So not just being an academic, but also to have some other interests that would go hand in hand, especially when it comes down to somebody who, in fact, does research that impacts the patients. And such a rarity, it's very difficult to accommodate, but of course, it's all within the interest of Professor Achilles Gravanis. So Professor Gravanis is a full professor of pharmacology at the University of Crete in the medical school, perhaps the most senior professor at the university, affiliated researcher at the Institute of Molecular Biology and Biotechnology, fourth. And most importantly, he is the co-founder of BioNature. In terms of research interest, he is one of the gurus of neurotrophins. That's basically the interest of BioNature and his research interest as well. And he's affiliated research professor at Northeastern University in Boston, where he actually shares his time, commuting back and forth. In terms of metrics, because that's also something quite important for young investigators to consider, he has more than 270 papers, an H-index of 48, more than 10,000 citations. And as I said, he includes this important rarity, an academic and a co-founder of a startup. So I don't want to waste more of your time, just to invite Professor Gravanis, my dear Achilles, to the podium, and we look forward to your talk. Thanks so much.
So, dear Rebecca, once again here with you. Thank you very much. I deeply thank the young investigators of the Pasteur Institute for this very onerous and kind invitation, and I do mean it. I consider this invitation having special, aspects. Another reason I'm really moved is because this is my first talk after my stepping down. I don't like the word we are using in Greece, syntaxi. And so I will speak as the CEO of our brand new, actually, spin-off company in Greece, Renewable Therapeutics. Today I'm going to speak for the young fellows, for the young colleagues here, and to present the 20 years actually history of our group in Crete. So don't pay much attention to the data. I'm going to show you data, but mostly to the concepts we discussed and how we approached basic questions, and finally how we use these basic questions to translational pharmacology to find out if we can help patients. And this is very important because it shows that if you want to do translational science, you should be a good basic scientist. So if you want to do, to find things that will be very helpful for patients, and of course have a value, a commercial value, you should have really prototype data or models, because in this country, and not only in this country, in other countries also, people think that translational science can come out of the blue, which is not true. And if you want to be really competitive internationally, you have to have two major, I think, properties. First of all, to tackle very difficult questions, because there are out there many people much better funded by us, who handle specific questions, so they are stronger ones. So you have to take the risk to tackle questions that most of the people out there won't do it. So if you have some chances there, you will be really very effective and very competitive internationally. So as Yorgos, thank you very much, Yorgos, for your kind words. 20 years ago, we started with BioNature in Crete, and BioNature tries to develop novel synthetic compounds with neuroprotective, neurogenic, and anti-neuroinflammatory actions. We named them microneurotrophins, and you realize why that was so. We have licensed actually one of our compounds, and in the next year, one of our compounds will be in clinical trials, not in Greece, of course, in Europe and in the US. And allow me to be very actually proud of it, because it's the first small molecule designed, synthesized, and preclinically evaluated in this country that goes into clinical trials in humans in a very actually important disease, diabetic retinopathy. You know that there are almost 170 million diabetics out there. 40% of them will develop vision problems, and 10% of them, they will lose their vision. So, and we don't have any treatment to help long-term these patients. The second spin-off company started a few months ago. We were facing lots of actually challenges to convince our academic institutions to give us the way to develop, to found this company. Its name is Renewrocell Therapeutics, and we want to develop a very efficient and complicated therapeutics for paralysis in patients with spinal cord injury. This is our first target. The second target is to apply this technology with scaffolds, 3D scaffolds hosting neural stem cells for the damage of optic nerve, and of course later on for stroke. The company BioNature was started 90 years ago by Ilias Kastanas and me. We were funded after some years by a very important company in the US, Emergo, $1 billion company. And the company was founded at the University of Crete, that has a percentage of the shares. And this is the website of the company for going there and find out more details. So we have two ways. I'm a pharmacologist by training. I have done pharmacy and medicine, and I never practiced both of them. And I've done my PhD in pharmacology actually in Paris with a prominent guy. The guys that come from France, they should know him, Etienne Beaulieu, president of the US Academy, member of the US Academy of Science, and president of the Medical Academy of France, Lasker Prize winner. We lost him a few months ago, and I'm so proud of having him as a mentor. I won't tell you how many years back, anyway. So, so there are two ways of developing new therapeutics. One is to define a specific therapeutic target and design ways to control it, block it, or induce it. And this is the way most of the drugs now are developing, because we have also other tools to help us. Which one? The in silico work, which really, propagates and facilitates and accelerates the way we're doing now new medicines. The other way is by chance. And this is the story of BioNature. So Yannis Charalabopoulos, 20 years ago, was working on his PhD thesis in our lab. And his research question was to define the way by which a very important steroid, dihydroepiandrosterone, is the most abundant steroid in humans. We have it in our body more than estrogens, progestins, glucocorticoids, androgens whatsoever. This small molecule, this steroid, is also produced besides the adrenal glands within the brain, human brain, by neurons and glia cells. So, and we know now with clinical and experimental data that it is involved in the degeneration of the brain during aging. So if you measure DHEA in people in their 50s, you will find almost 10% of what they are having in their brain and peripherally in their 30s. And if you go further, these levels diminish further. So we know now that DHEA is a molecule, endogenous hormone. We name them. This is one of the discoveries of my mentor, Etienne Bollet, and he named these steroids as neurosteroids because they are coming from the neural tissue. So Yanis tangled the question how this small steroid protects neurons. So, and he published his data 20 years ago, and when we were writing the thesis, we were shocked by the fact that this small molecule does not bind to an intracellular receptor, as we know happens with all steroid hormones, but is recognized with high affinity, KD of nanomolar levels on the membrane, and activates a cascade, actually, of events that go by activating the different kinases, transcription factors, and these transcription factors control the pro-apoptotic and the anti-apoptotic BCL2 genes. So when Yanis was writing the thesis, I was shocked by the similarities of the neuroprotective effect of this steroid with the most important neuroprotectant we have in our brain, which is nerve growth factor. It was discovered when I was born by Levi Montalcini and Cohen. No coincidence, okay? I found this, this work later on in my life. And so the next question we were facing was, okay, how this is happening? So Iakovos Lazaridis came in the lab from the UK, and his thesis was exactly to find out if DHEA mimics NGF because it affects, binds, or regulates NGF receptors. And that was true. We published this data, and then a third one came in the lab, Sifis Pediaditakis, with the obvious question, okay, this is a particularity of DHEA only for NGF receptors, or this small molecule can bind to all neurotrophin receptors, and Sifis published a paper after some years showing that indeed DHEA has high affinity, nanomolar levels for neurotrophin receptors. At that moment, we advanced the theory that based on biochemical and pharmacological data that most probably DHEA was the ancestor neurotrophic factor early in evolution. With species that don't have any brain, they don't have any tempospatial need for controlling the development and the shaping of the brain. Neural cells are endothelia, where mostly endothelial cells you can find around. So you needed a small molecule, highly lipophilic, going everywhere, stay for a long time, and of course steroids have this property. So this is another actually example that when you're doing basic science, you can find things that are helpful and useful for developing drugs. So we named, Rebecca, this paper From Evolution to Pharmacology, which is ambitious enough, but I think it's justified. So let me remind you some of the properties of neurotrophins. We have NGF, BDNF, brain-derived neurotrophin, NT3, neurotrophin 3. Each one of them are recognized on the membrane of neurons, and not only, they are recognized by other cells also, immune cells, for instance, and other epithelial cells in different systems. NGF is bound with track A, BDNF by track B, and NT3 by track C. All of these three receptors are supposed to be pro-survival receptors. If the neurotrophin, specific neurotrophin, bind to this receptor, it induces signaling factors supporting neuronal inhibiting neuronal death, I would say. There is another receptor with low affinity, a pan-neurotrophin receptor, the P75NTR membrane receptor, which binds with much lower affinity all of the neurotrophins, but not only that, it binds also the pro-neurotrophin molecules. Neurotrophins are the result, the cleavage, the enzymatic cleavage of coming from pro-neurotrophin, big molecules. They are cleaved, and you have finally the active neurotrophins. They are the end products that have all these properties summarized in this slide. P75ATR belongs to the TNF receptors of the family and supposed to be a pro-death receptor. So imagine how these are working in the brain, specifically in the developing brain. You have a system, a multifaceted system of neurotrophins. The mature neurotrophins support survival. The pre-neurotrophins induce death. Then you have the equilibrium of all these systems, expression of pro-neurotrophins plus mature neurotrophins, expression on different neurotrophin receptors, a specific loci, and the dynamics of these two molecules, the interplay of these molecules, define which neurons will die and which neurons will survive and evolve. And this is very important for the development, brain development. I'm talking in an institution that has lots of experience on that for decades. It's extremely important how all this interplay will work to have a normal and mature brain. So this system is involved not only in supporting the survival of neurons and other cells like glia cells and microglia, but also can induce, under specific conditions, the shaping of the brain by inducing apoptosis through the P75NTR receptor. These very important, actually, polypeptidic agents, endogenous polypeptidic agents, can regulate not only the shaping of the brain, but also the functionality of the brain. They control the myelination, they control synapses, they control differentiation of cells that facilitate the formation of myelin sheaths. They have a very central role. What is very important also is that we need neurotrophins from the first time we're coming in life to the end of our life. And if something happens and these molecules decline, then we have really problems of brain functionality. So all of these are involved, huge numbers of papers in neurodegenerative diseases. So NGF is involved in Alzheimer's. There were many, actually, people working 15, 20 years ago trying to develop peptidics for the treatment of Alzheimer's. One of them was George Yankopoulos, who didn't succeed and changed, actually, field 25 years ago. And I got an email 7 years ago when we published that. He was surprised that a small lab out of the blue from Crete finally had small molecules that could mimic, as you will see, this neurotrophin. And so we said, okay, we have a basic knowledge on neurotrophins. We have these neurotrophins cannot be used pharmacologically, big molecules, they cannot pass the blood-brain barrier. They are destroyed in the stomach. So what is the role of a pharmacologist? To mimic nature in an extent that pharmacodynamically and pharmacokinetically you can find something that will mimic something endogenous, so you can give it to supplement or to mimic the endogenous factor. It's exactly what we have done. Let me remind you, most of the biological endpoints of almost all neurodegenerative diseases. First of all, a very, actually, not so happy statement. We don't know the etiology of whatsoever neurodegenerative disease so far. We don't know. We have tons of papers, lots of work, But if you ask someone, what is the etiology of multiple sclerosis? We don't have any answer. We only have some changes in the neurobiological changes, but the etiology of how these are happening, we don't have any answer. So the neurodegenerative diseases have a common, actually, pattern of symptomatology based on their neurobiological pattern. First of all, in all of them, you have loss of neurons from apoptosis, from autophagy, from neuroinflammation. You have neuroinflammation. You have also demyelination. Demyelination, we were thinking about demyelination only for demyelinating disease like multiple sclerosis. Now it's not true. Now we know that most of the neurodegenerative disease, including Parkinson's disease and Alzheimer's, have problems in their myelin. And the new comer in this whole scheme was the new knowledge about neurostem cells and the potential even the aged brain have to produce brand new neurons by neurostem cells we have in almost all loci in our brain. So this is the new pattern. So this is a pattern to design not systematic, we don't know the etiology, but symptomatic treatments targeting on all these neurobiological endpoints. So our idea was to find dynamic molecules. When I was doing my PhD thesis in Paris, the dogma was the magic bullet. One molecule, one receptor, so you have efficacy, you don't have toxicity, but dear friends, I don't know any disease that is so targeted only on one, dependent on one gene or one pathway. So most of the diseases we are facing, specifically neurodegenerative diseases, are multidynamic diseases, involve many genes that have the redundancy. If you block one pathway, another one will take over. So the idea was to develop multifaceted small molecules mimicking the multifaceted endogenous neurotrophins, targeting apoptosis, demyelination, oligodendrocytes, neuroinflammation, glia, and if possible, through this way, we would stop the natural history of the diseases. In addition to that, if we had small molecules to boost neurostem cell proliferation, differentiation, and even migration, we could complement these diseases by having a chance to reverse some of the symptoms because of the fresh new tissue supplementing the suffering one. So that was our goal. You have here the DHEA molecule. Yannis has done a very wonderful work showing that the pharmacophore, that the anti-apoptotic and the neuroinflammatory component of the molecule was here on the 3-hydroxy group. The 70 carbon group is the group that is used by the steroidogenesis enzymes to produce, because you should remember that DHEA is up to the steroidogenesis. Cholesterol, pregnenolone, DHEA, and then you have androgens, estrogens, and all their metabolites. So we went to a good friend of mine for the last 20 years, Theodora Kalogeropoulou. She works as a very talented organic chemist in the National Foundation of Research here in Athens. Theodora, with her group, scrambled all this region and this region without changing the pharmacophore for neuroprotection and anti-neuroinflammation. Theodora and her team produced a very large chemical library of different 17 carbon derivatives of DHEA. Sivis Pediaditakis took the initiative to find out if some of these derivatives can mimic DHEA on neurotrophin receptors. He published the results a few years ago, And we know now that the exact location of the binding on the receptor of NGF, TrkA receptor of these small molecules, KD nanomolar, 1 to 5 nanomolar, very strong association with the receptor, binding to the receptor. And with the tedious work, Sivas Fidelitakis published the molecular mechanism by which the small molecule BNN27 acts and activates neurotrophin receptor, TrkA receptor. And we found a very interesting, this is pharmacology. We found a very interesting, actually, a difference between the binding of a small molecule on NGF receptor compared to the binding of the endogenous NGF. So, and this is summarized in this slide. When TrkA receptor binds to BNN27, internalizes into the cytoplasm, and normally the receptor bound to the endogenous ligand NGF goes towards the late endosomes, and there it is lysed by entering into endoplasmic dictyon, the plasmic network. So when you have TrkA receptor bound to the small molecule, you have the induction and activation of specific proteins which block the movement of the complex towards the late endosome, and by this way the complex returns very fast to the membrane. So not only you mimic some of the effects of NGF, but also you sensitize the neuron, because the neuron does not lose NGF receptors, but the membrane is enriched by the receptor, not de novo synthesized, by all the receptor that comes again on the membrane. This is very important, because if you think to mimic something that goes down, is replenished in a disease, so if you can, and so you have the endogenous ligand very low, so if you find a way to sensitize your neurons to the very low concentrations of the endogenous protectant, in addition to mimic the endogenous protectant, you can also increase the sensitivity to the low levels so you have two actions that are more effective, finally. So to cut a long story short, we have a very important chemical library of multidynamic microneurotrophins. Tassos Economou, the late Tassos Economou, a very good friend and biologist in Crete. When I was discussing collaboration with him, he was doing proteomics and genomics. He proposed this name, neurotrophins, mikres neurotrophins, microneurotrophins. So Tassos proposes this name. So we have NGF receptors with strong neuroprotective actions, with strong strong inhibitors of microglia activation, and some of them, they can boost neural stem cell proliferation. And we are talking about the same molecule, not different molecules. One molecule, as you will see, has these properties. BioNature got along with professionals in US. So we had drug advisors, SAGE group, and SAGE group, fortunately, proposed to us not to go on very complicated neurodegenerative disease like Alzheimer's or Parkinson, but to target a disease that is very neurodegenerative disease, but that is very, it's much easier to develop drugs, and that was diabetic retinopathy. 170 million diabetics, 40% of them, they have vision problems, 10% of them, they will lose their vision. So we had the possibility to test one of these compounds on this neurodegenerative disease, and we collaborated with Kiki Thermos, professor of pharmacology nearby my office in Crete. Kiki is a very, she retired also. Kiki is a very prominent pharmacologist for eye pharmacology, knows very well all these systems, and she have done with his team a very tedious work, and we found that indeed BNN27 can inhibit various types of damage of the retina through diabetes. We have used a very convenient, actually, animal model, the streptozotocin rats, which develop in a few weeks problems with their vision and their retina. So the work is already published, and IBAN has done now a very good work on that. I won't go into details. The paper is out. The second one as a target was Alzheimer's. That was our first target, but the guys that know how to develop drugs said this is very risky. People all over the world lost lots of money because of non-success in developing Alzheimer's therapeutics. And this is also the case even today. We have now monoclonal antibodies against beta amyloid. I will talk on that a little bit later, but questionable. They work in patients early in the disease. Some of them don't have even we don't have the capacity to say that they are indeed Alzheimer's patients. So the use of these monoclonals in the late disease doesn't work. So I would say with the reading and the discussion with people like Roubakis, who was one of the prominent persons proposing the theory of beta amyloid toxicity in Alzheimer's, that most probably beta amyloid is an epiphenomenon of the disease. So if it's an epiphenomenon, you can use it as a very good biomarker, but it's not a therapeutic target. So, but people are taking this drug because of the failure so many years. There is a proposal that a measurement that the pharmaceutical companies only lost almost $30 billion in their failure to develop a therapeutic for the disease. So very prominent problem in public health. Many people work on the field. So we have used a humanized model for Alzheimer's, the 5xFAD mice. They have specific, the most prominent actually, mutations related to human mutations related to the disease. The disease simulates some of the problems and symptoms of the diseases. It doesn't, mimic 100% of the disease, but it's useful for drug development. So 6 months after their birth, these animals with these 5 mutations, they develop, they lose the capacity of memory, on specific memory. They accumulate beta amyloid in their cortex. They have some of the aspects of human Alzheimer's. So the group published a few months ago the results of BNN27 on this model. And to cut a long story short, we have found that if we give to these animals for 60 days as a pellet to not stress them the compound, then 60 days afterwards we do a behavioral test, many actual behavioral tests, not only one. I present here, the paper describes all of them, and after we perfuse and we get the tissue to measure the different biological endpoints. BNN shows a very important downregulation of plaque formation, almost 50%. This is very difficult to find it in the literature. We were very skeptical when we found all this data, and we repeated many times this series of experiments. We have done comparative proteomics of animals, wild-type animals, or 5xFAD animals treated with the compound, and we found that indeed the compound can rescue some of the proteins involved, very known actually proteins involved in the disease, and the paper describes in details all this work. Now we're discussing with 2 big pharmas, with Sage Group in the US, to find out if there is an interest to go into the clinical development of this compound. Another work that has been done by Ioana Zota, she finished her thesis a few months ago, she is now in Fleming, is to define in this animal model of Alzheimer's disease the demyelination process and the function of the oligodendrocyte precursor cells. It is very recently published in glia, and indeed in these animals we have a very strong diminution of demyelination and the function of oligodendrocyte precursor cells is decreasing. The levels of demyelination, as I mentioned, are minimal, lesser in this animal 6 months later. We have reproduced some of the effects of the myelination platform in vitro. So we exposed in oligodendrocyte precursor cultures to BDNN, and we defined proliferation and survival in absence and presence of the neurotoxic beta amyloid, and BDNF can reverse this toxicity. Again, we went to our good friend Theodora Kalogeropoulou, and we tried to develop, to synthesize, to design and synthesize not TrkA mimetics, but now TrkB mimetics, the receptor for BDNF, and we presented this data of Despina Haru a few months ago. So we have a good library of TrkB mimetics, BDNF mimetics, small molecules again. One of them is tested in different models in vitro for reversing the beta amyloid toxicity. We tested if they have an effect on the proliferation and survival of mouse adult hippocampal neurostem cells in culture. Why so? Because these cells in hippocampus, they interplay with the glia cells and the remyelination processes. We have done also comparative genomics, ontology, enrichment analysis of the effect of this compound compared to the endogenous ligand. And indeed, we found very interesting data showing that this compound and BDNF can reverse some of the genes that are overexpressed in the disease. This is a way for validating a model to compare it with human material. I mean, you can get data from the databases. We have now data expression profiles for proteins and genes in various brain regions, normal people and patients with different with different neurodegenerative diseases, and you can compare your data, in vitro data, with these libraries and find out if indeed your system can mimic some of the effects and some of the aspects of the human disease in vivo. So now we have TrkA, NGF mimetics, TrkB, BDNF mimetics, and we want activators of these two receptors, and we propose, and we are starting a series of experiments now, the combination of these two small molecules for more effectively controlling apoptosis, demyelination, neuroinflammation, and if possible, boosting neurogenesis as a potential symptomatic therapy for Alzheimer's. This work was done by many groups. It's not only our group. We are collaborating with colleagues in Angeleto in Patra, with Domna Karaogeos in Crete, of course in Athens with Theodora Kalogeropoulou. So, and as I mentioned at the beginning, we are very happy that one of our compounds of the company is now licensed to Novaliq, behind Novalin is a big pharma. This is the way that big pharma work now. They create a small spin-offs. If this spin-off is effective, they get it. If it doesn't, they ignore their presence. Pfizer, BioNTech, okay? This is the way they are doing new therapeutics. And we are in discussions for Alzheimer's with two big, as I mentioned, two big pharmas in US. So this is the team. Yannis Karalabopoulos, now he's the professor of pharmacology. He went to Karolinska, and now he's a professor of pharmacology in Crete. Iakovos Lazaridis, he went again to Karolinska. Now he's faculty at the Department of Neuroscience in MIT. Vasilis Minas is in Oxford. Sifis Pegaditakis, he went to Harvard to emulate, then to Alchemab, and now he founded his own spin-off company in Boston for drug development for Parkinson's disease, a competitor actually. Paskalis Evstathopoulos, he went to Karolinska, to NIH, and now he is a senior scientist in Novo Nordisk in Copenhagen, trying to develop cell therapies for Parkinson's disease. They have wonderful data. Alexandra Kuryadaki, he went to UCL, and now he works in New York in a company, Intelligentsia, which develops algorithms, AI algorithms for drug development. Of course, Theodora, without her, we wouldn't be there. And it's a good friend and a very, very talented, actually, organic chemist. Now, I will finish fast with the neural cell therapeutic story. A spin-off of Forth and the University of Pitt Medical School. We're preparing a license agreement in Texas with Neurocords. Neurocords is a company of cell therapies, and we are almost done to sign the second license agreement of our group in the US. A comment here. We couldn't find money, dear friends, from Europe. All the money we found, being a European site, are coming from the US. This is very important, and this says lots of things for how we should act as Europeans the next years. Now we don't have competitors only in the US, we have competitors in Asia. China, North Korea, etc. Okay? So we have to be really, we have to think strongly how we are going to change mentality in Europe, because many good ideas are found in Europe, and they are developed and gain money in the US. So this is something for you, the young guys, the young European guys, to think differently. And now with the problems you were facing to go in the US because of Trump administration, many people leave the US. I know very well that. So you have to think European, and you have to think bold, and you have to think as scientists and developers with risk, taking risk. We focused in the problem of spinal cord injury, no therapy at all. Many actually patients worldly, young patients, because they are getting the condition by trauma. We don't have any therapies, only symptomatic and very limited effects of the symptomatic therapy. The only thing we can do is decompress the spinal cord, so the spinal cord does not further deteriorate, and diminish the inflammation of the spinal cord by giving different actually drugs. They don't work pretty well. The last few years, a big actually discussion started to use biomaterials that will cue the gap in the spinal cord, traumatic spinal cord, so they will give the possibility of the fresh, normal neuronal axis to communicate between each other and surpass the gap, so to re-establish interconnection, neural interconnection. It didn't work. One of these people that started this type of reasoning was Yanis Yanas in MIT and John Burke, a surgeon in Harvard, a trauma surgeon guy. They didn't succeed, and when I was in Boston, I was discussing with this wonderful person, Yanis Yanas, professor of MIT, father of tissue bioengineering, member of the US Academy of Science, member of the Hall of Fame of Inventors, entrepreneurial guy. He created Integra, $1 billion in NASDAQ, and with bioengineered artificial skin that is used all over the world. So we discussed with Yanas, and one of his fellow there, Dimitris Zeranis, who was doing his PhD thesis with him. He spent 10 years for his PhD thesis in there. He's very knowledgeable, in other words. So we started a collaboration to define ways to help these people. There are two ways of helping, three ways. One is by electronically stimulating the damaged spinal cord, so you surpass the vacuum by getting into the spinal cord electrodes, so you can bypass the vacuum and go from the brain to the periphery and vice versa. There are efforts to develop this type of microelectrodes. Clinically speaking, we don't have any success yet. The other way is what I mentioned with the story of Yanas and Burke, to find scaffolds that will fill up the gap, help the neuronal axis to work on them, interact, and by this way establish again neuronal transmission. The last one was by infusing neural stem cell suspensions of various origin, so you can create de novo with neurogenesis in situ, new fresh normal tissue which will interconnect. All of them didn't work. There are some results very recently, a few weeks ago, from Japan with neural stem cell suspension that seem to be promising. We have chosen with our engineers, and engineers have a very interesting property. They are problem solvers. They don't care about truth. They want, and they are trained to solve problems. So we went with engineers in MIT. We said, we have this problem, and we started to discuss the combination of all of this. 3D scaffold made by human proteins, friendly for the human body, which will host neural stem cells, and all this junk ex vivo will be regulated and treated with different factors protecting the neuroimplant. And once we are happy that this is functional, then transfer it to the gap, to the patient. And this is exactly the proposal, very elaborate and very complicated, actually, therapeutic approach. So we have now, we have done the different types of 3D scaffolds. We are hosting the scaffolds with neural stem cells. We treat them ex vivo with different factors protecting all this junk. And then the idea is to transfer it to humans. So we have done our proof of concept in animal models. We have used the dorsal column crash mouse model. Robin Franklin in Cambridge helped us a lot to learn how to operate these animals and how to evaluate them. Another group that helped all this effort the last years. And we have published these data a few years ago. Alexandra Kuryadaki was the first author, equally with Dimitris Zeranis and Karenina Canelli. You can go through and find out the specifics. To cut a long story short, this is the scaffold, scaffold mainly with human collagen, but has also other components. We co-cryopolymerize this globular human material with other components, so to give specific properties. How long they will stay in vitro or in vivo, how long, how they will stay there in the damaging region, so to give the cells to the trauma. So this is a neural stem cell component, a neurosphere that is cultured within the scaffold, and you can see that we can keep these cultures for 3 to 4 weeks ex vivo, and the neural stem cells, they differentiate, and you have neurons with neural axis, and they were very happy in this component. Alexandra has done with Dimitris tedious work to define exactly the composition of the scaffold. It took us 3.5 years to finish this work. Lots of effort and tries for developing the scaffolds. Then we had to test the scaffolds to be friendly not only for the neural stem cells, but the outcome of the differentiation of neural stems. In other words, sensory neurons and motor neurons. So we had another in parallel work to define if our scaffold was friendly also to this type of neurons, because that was what we want to do in vivo. Give them stem cells. The stem cells won't diffuse in the spinal cord, stay within the traumatic area with the help of the scaffold, and then give the chance of these neural stem cells to proliferate, to survive, very important, and to differentiate towards the neurons under the direction of the local factors, because the cells we're using different type of cells. We want to communicate between with the normal tissue, and this normal tissue will guide the differentiation of these cells to become finally the types of neurons are needed. We have done lots of experiments with different types of scaffolds. I won't go into details. These are the results with the transplantation of the scaffold to experimentally paralyzed animals. And as you see here, 12 weeks after the transplantation, animals walk very well. They sense very well, no statistical difference with the normal animals. And we have a very stringent actually test. We put the animals to walk on a horizontal ladder. Imagine yourself walking on an horizontal ladder. So you have to know exactly the position of your hands and feet. So this is a very tedious work, tedious test to define if the scaffolds were effective. And we know now exactly what was happening. The scaffold was GFP colored because of human collagen with GFP. So after the transplantation, we knew exactly the area of the setting of the neuroimplant, and we were able to follow up what is going in and what is going out. Now we have a very great number of data showing not only how these neural stem cells are differentiated, to which kind of neurons, what are happening with the neuronal axes that are developed, if the neural axis interact with the normal exogenous neuronal axis, and how this is happening. We have we define very well this interaction. Now we are moving from mouse neural stem cells to human stem cells, and we are setting up a series of experiments with different types of human neural stem cells, culture ex vivo in the neuroimplant, and then transfer to immunocompromised animals, which are surgically paralyzed on specific sides of their spine. And our new steps is to clinically develop in 2027 the neuroimplant to humans. These are the different stages. We have found the money for the proof of concept. We almost find the money for the JMP manufacturing in Boston and the IND preparation for FDA. and wish us good luck that in 2 years we will have our first try in 3 or 4 patients in Boston. We have defined a very strong collaboration with the Sporting Rehabilitation Hospital in Boston, and the new chairman of the institute, Professor Rosa Fonte. And in Europe, we discuss with the Hellenic Spine Society and the Euros Spine Society, a very talented and very experienced actually surgeon, spine surgeon. Sotiris Papastefano is the president here and the vice president here, and we're in Eurocourt. And the money we are now accumulating, we hope that in 2 years we will try this neuroimplant in paralyzed patients in Boston. And this is a team for the Neurocell, Yannis again, Dimitris Geranis from MIT, now assistant professor of Department of Material Science in Crete. He came back. Alexander Koryadaki in New York, Kaleli, and the new guys in the team, members now of Neurocell Therapeutics. Thank you very much for listening.
Thank you so much. Very inspiring talk, actually, demonstrating how you can move basic translational science, not just for the sake of it, just to address the patients, and if it is profitable as well, it's even better. Questions from the audience? Okay, Rebecca.
Well, first of all, thank you very much for this wonderful journey throughout your, I think, research and education career, and which eventually led to the development of these startup companies. So I do wish you luck with all this. I just wanted to ask you, is this compound that you're using for diabetic retinopathy, will it be developed as a supplement or as a drug?
Novalic has a very interesting eye drop solution without any water in there, nanoparticles. They have tested in various diseases for eye diseases. And we know now that with our compound we have prepared a common patent for formulation. So we don't have any medicines to help these people whatsoever. The only way to help them, Rebecca, is to control their diabetes. So to prolong the damage in the retina. Our idea is to prophylaxis, pharmacological prophylaxis, prevention. Okay? So the idea is to develop an eye drop solution containing our compound, BNN27. We have done pharmacokinetics in big animals, and we know very well now with iSol, the Novalic's eye drop solution, that the compound goes very deep into the eye. We have measured the concentrations of the compound into the retina and the optic nerve. So if everything happens well, because now the idea is to, and it's in process to test it in primates in the US, Caribbeans. If things work well, Novalic prepares itself to organize the first trial in Germany and in the US by the end of next year. Monotherapy, only one.
Yeah, but is it a drug? I mean, is it being developed as a drug? I mean, does it need all the filing and everything for drug development?
Of course.
Or is it going to be an off-the-shelf
it's a drug.
Supplement? It's a drug.
It's
okay.
not it's a drug.
It's a prescribed. It will be a prescribed
Absolutely.
drug. Right.
Yes.
Thank you.
Otherwise, they won't invest.
Okay. Well, I
want a
mean,
drug.
an off-the-shelf supplement can be
it's a
very,
drug.
very enriching as well and very profitable.
Right.
Fotini, our former president.
Well, thanks. It was really very nice to hear this story, which we all have followed along with you. And I wish you all the best now that you have stepped down, which is a different kind of life. But coming back to the drug for the diabetic retinopathy, I have a question. One of the main clinical problems in diabetic retinopathy is neoangiogenesis. So we have a growth phenomenon, an abnormal growth phenomenon. And your compound, at least in other systems, is a growth-promoting compound. So what is the experimental evidence that you have of the mechanism that this compound, which in other systems is growth-promoting, will it stop the neoangiogenesis? Will it act somewhere else? Or is it information that you cannot reveal? I mean, after all, it's a drug that's being developed. But can you tell us some things? I have this question.
It does not promote proliferation. It is a survival molecule. It blocks the loss of neurons and glia cells, and the only promoting effect it has is in neural stem cells of different types. So the idea is to protect, ganglial cells, Müller cells, and microglia, which is inflamming within the diabetic retina. So Novalik and the big guys behind, they are very happy to have a multifaceted small molecule that can control, in a certain extent, all these three phenomena. So even if you have a leakage by your neoangiogenesis stuff, the target is to control, to diminish the damaging effect of the neural tissue. Okay?
Thank you. Thank you.
And of course, this can be achieved by monoclonal antibodies against VEGF, which is in clinics for many years. So if they want to complement with the neoangiogenesis problem, they can propose to combine these small molecules with the VEGF monoclonals, actually.
Thank you very much and good luck.
Thank you.
Okay, we have a question over here.
Achillea, thank you very much for this exciting presentation, and good luck with all your efforts. How the cellular composition of the implant in the spinal cord changes over time. What goes in and what goes out? This is my first question. And the second question is, what is the time frame of the improvement of the animals that you saw? I saw in the your slides that it was probably 14 days or more.
12 weeks after the transplantation.
12 weeks. Okay. Did you test longer times?
Yes, we are testing longer times. We have all this data. We are not going to publish, Spiros, this data because we didn't patent yet the neuroimplant, because the first patent was a provisional patent. We want to have all the data as a PZT by the end of the provisional period, and we have data towards this direction, very interesting, and we have data also on the in vivo degradation of our scaffold and how the cells behave. But I won't state them in public unless we complement our patent.
Or sign an NDA. We have time for one more question, Athena.
Yeah.
One more question, or we can go on. Okay, Mimika.
Thank you very much, Achilles, for the great talk. I have actually two questions, but I can make only the one now.
All of you have many questions, and I know that you have many questions. Rebecca, all three of them.
Okay, so you showed us that there is an increase in neural stem cells neurogenesis in vitro following the microneurotrophin's addition. How about in vivo neurogenesis in the hippocampal region in animal models? Does it take place in the 5X FAD model, or?
We didn't test them in detail, but we have the data of the control animals in the 5xFAT model, and we have a small increase of the neural stem cells in the hippocampus of the control animals, and we have a much higher in the 5xFAT. Why, Emika? Because they are lesser. So the difference you can see is because these animals lose lots of their stemness in the hippocampus, so the difference in these animals is much bigger. But yes, we have also neurogenesis in the normal animals. This is not, I mean, it's not necessarily good, okay? But this is true. We have boosting, a small boosting of neural stem cells in the hippocampus of wild-type normal animals.
One last question here, front row.
Thanks a lot for the nice talk. Probably my question is really naive. It's regarding the second part of the presentation. So in particular about the spinal cord injury. So I don't know in your model at which level you are doing the injury, and based on that, can you guess what is the activity of the neural stem cell and how they are going to recover the ability to walk? Because I know
Yeah, very, very tedious, difficult question. So in pharmacology, sometimes we say the following. If you have a cat that catches mice, you don't care its color. So this is applicable also in our, so we have an end point, biological end point after the transplantation, and which it's without any discussion, statistically speaking, of course, okay? The question of Spiros is valid. I mean, we should know how long it will stay there. How long it will stay there. It will stay or regress. This is a very good question. So most probably, because of the ex vivo and in vitro data, neural stem cells differentiate, proliferated differentiate, this we know it, and we have different types of neurons within the scaffold. GABAergic, inhibitory and stimulatory, NMDA positive. So this is happening in coordination with the normal tissue sitting around. So we know that the neuronal axes interconnect. They go out of the green zone, the neuro implant, the scaffold, and interconnects, and we have synapses there, and vice versa. But which is the most important component that gives, the ability of these animals, restores some of the abilities of these animals to sense and walk? We haven't the slightest idea.
So I was, with this question, I wanted to ask, have you recorded the activity of some of the neural stem cells that are in place, like they are actually participating, with the motor component?
In vivo?
In vivo, yes.
No, only ex vivo. We have lots of data with the scaffold, and for 3 or 4 weeks, hosting different types of neural stem cells, and we measure calcium. If you go in the paper, you will see a wonderful video in the supplementary files of the paper, with calcium and with electrophysiological recording. We have electrodes within, and single cell recording, so you will find all this data there, ex vivo, not in vivo.
So I would assume you're going to have a very nice opportunity for recruitment, right?
Actually, we're seeking for young guys for the company. So.
Yeah. And actually, I couldn't agree more with your comment about these great ideas traveling in the US, because this is the place that they understand business, especially when it comes to startups. Because, to start discussing startup and to fund a startup, they have to first understand what this is and how much money it takes.
Of course.
So, if there are no other questions, I'm sure that Professor Gravanis will be available at the party afterwards if you want to talk to him. So thanks so much, Achilles, for coming over. It's really an honor to have you here.
It was an honor. I will keep this badge forever. This is my first badge as an entrepreneurial guy. So thank you so much. Thank you.
So, the floor is now to Athina or to Elias or Vasilis or all of you.
Thank you all for being here tonight. We will start with the award ceremony. So I will invite you again. We have 2 oral presentation awards, 2 poster awards, 1 flash talk award, and 1 image award. The oral presentations and the posters were selected from our scientific committee. Thank you very much for being here all these days, and you are very actively participating in the Congress, asking a lot of questions, specifically Professor Efthimiopoulos. But thank you very much, really. Our scientific committee, which is Professor Efthimiopoulos, Dr. Michailidis, Dr. Douni, Dr. Giannakakis, and Dr. Vekrelis, review more than 112 abstracts during these 3 days. So this is the results. Please. So we start with the Best Scientific Image Award. This award is given by the audience, and I would like to invite here Ekaterini Vourlia with the beautiful butterfly. The award is €200 from Plesio.
Okay, the award for best flash talk presentation, and this is given to Baby O'Hara, from the Pasteur Institute of Paris. So thank you.
Moving on to the awards for the best poster, I would like to invite Mary Margariti to the podium.
The next award for poster presentation will be to Marielena Pouliou. The poster awards are €300 each.
And for the Best Oral Presentation Award, we have 2 winners. The first one is Paschalis Theotokis. It's €500. And for the Lukeris Award, the last one, the winner is Sumia Abdelaki from Pasteur, from Paris Pasteur Institute, €500. Well done. I would like to thank all the everybody that presented here. It was amazing. It was very difficult for the scientists to decide which works to be awarded. And I would like to give the speech to Dr. Patanos.
Okay, I'll keep it short because the actual ending should it goes to the organizers. I want to thank you all for being here with us. It has been an amazing 3 days. Special thanks to the sponsors that have supported the event. Special thanks to the technical support of the Hellenic Pasteur Institute, Angeliki and her team, who made sure that everything is well arranged. I think she and her team deserves a very loud round of applause. And I must say here that thanks to their effort, the garden has been reopened. Yeah, what else? Actually, I really felt that those last 3 days were quite inspiring for you, just to dream and get exemplified by the speakers. So in few years, you could be in their positions, not just as an academic, but also as an entrepreneur. And it actually means that you should aim high. Don't take any obstacles in your career. You're going to have difficulties. This is to be taken for granted, but in the end, you will prevail if you should keep yourself very focused and don't care about difficulties. And actually, very many special thanks to you. I want to say that next year, God willing, we're going to have 2 new beautiful buildings, 4,500 square meters of laboratory and auxiliary space. It's going to be right next door, including 2 state-of-the-art additional amphitheaters. So I really hope that we're going to see you all next year at the 9th conference of the Young Investigators. So I want to thank you all again, and I pass the floor to the organizers.
We would also like to thank Professor Patrinos for your guidance and for allowing us to do this Congress exactly as we imagined it and giving us this freedom. I think it's very important for the young researchers to have the freedom and the floor to do what they want. This is the most difficult part. I would like to thank, of course, the rest of the members of the organizing committee, Ilias, Vasilis, Evgenia, and Stavros. Without them, this couldn't be possible. Of course, the scientific committee and the keynote speakers that were here with us. We would like also to thank Dr. Christina Ekonomopoulou for her overall support in the young scientists. Of course, the technical support team, and specifically Ms. Liberopoulou for taking care in every detail these 3 days. Our sponsors, and of course, the biggest thank you to our colleagues from the Pasteur in Paris. This is a joint conference. We did it together, and we are very happy that you were here. I would like to invite now Remy, Elea, and Raphael here, please. We have some small gifts from you because you visit our country and for our next collaborations, and to thank you for being here. If you
Well, as always, I improvise because I have nothing ready, but really thank you so much to have us here. I think that all of us were super happy to be here. We received a super warm welcome, so really thank you so much. It was so nice to visit Athens with you, to stay here with you, talking about science, and I really hope that we can continue to meet times. Maybe not only with science, also with some drinks, like after, so... And yeah, we all invite you in Paris if you want. And really thank you so much. I don't think that I should add something more, like really thank you from all of us. Thanks.
Now we have a dinner in the garden. I hope it's not raining, and a welcome party. Thank you very much for being here.